Assalamualaikum warahmatullahi wabarakatuh. A very good morning from Gorontalo, Indonesia. The Honorable Mrs. Arianti Anaya, MKM, as Director General of Health, First Ministry of Health, Republic of Indonesia.
The Honorable Director of Health, Poltekkes of Ministry of Health of Gorontalo, Mr. Muhammad Anas Anasiru, SKM Kemenkes.
The Honorable Vice Director of Health, Poltekkes of Ministry of Health of Gorontalo.
The Honorable Professor Masaki Kita, PhD from Nagoya University, Japan.
The Honorable Tyler Jafarisco, PharmD, PhD, from University of Houston College of Pharmacy, USA.
The Honorable Dr. Muhammad Azmal Shah from Hazara University, Mansehra, Pakistan.
The Honorable Dr. Siti Nur Fazila Musain from University Teknologi Mara in Malaysia.
The Honorable Professor Ikatan Adiana, MSc PhD from Institute of Technology Bandung.
The Honorable Professor Andi Dian Permana, MSI PhD APT from Faculty of Farmasi, Hasanuddin University, Makassar, Indonesia. The Honorable Mr. Salman SKM Kemenkes as Chairman of Pharmacy Department of Poltekkes of Ministry of Health of Gorontalo.
The Honorable our distinguished guests from Bank Syariah Indonesia, BPOM, KKP, IAI, PAFI, lecturer and stakeholder, and also honorable to our sponsor for this conference, the Honorable the Academic Community of Health Poltekkes of Ministry of Health of Gorontalo.
The Honorable all participants of international conference today.
Ladies and gentlemen from Gorontalo, Indonesia, on this lovely morning, we would like to welcome all delegates in the opening ceremony of Third International Conference in Pharmaceutical and Health Science 2023. It's being a great honor for me, Priska,
and Hadi,
to be here and welcome each and every one of you in this conference.
For your information, this conference supported by
CV Intraco,
Bank Syariah Indonesia,
INT Tour and Travel,
PT Maja Bintang Indonesia,
Mulia Sehati,
Apotik Fit Pharma,
CV Total Equipment,
PAFI Bone Bolango,
Advia Pharma,
Calon Dosen TV,
Kawan Medika,
Apotik Wan Setia Baru,
Warung Kita,
Prodia,
Rahida Cookies,
Farzana Printing,
and Pharmacy Alumni Health Poltekkes of Ministry of Health of Gorontalo. Ladies and gentlemen, over the next few days, this meeting we will provide you plenty of opportunities to discuss, share knowledge and insight.
First of all, let us pray and praise you Allah Subhanahu wa Ta'ala. Because of His blessing and mercy, we can come together without any obstacle here with healthy conditions in the seminar of welcoming ceremony of Third International Conference in Pharmaceutical and Health Science 2023, held by Pharmacy Department and Pharmacy Alumni, held by Poltekkes of Ministry of Health of Gorontalo. Second, let us greet and praise you our beloved Prophet Muhammad Sallallahu Alaihi Wasallam, who had brought to us the paths of light and left the darkness in this life.
Distinguished guests, ladies and gentlemen, on this nice occasion, let me deliver the structure of the event in the first and the second day. As follows, in the first day, opening ceremony, traditional dance by pharmacy student, national anthem Indonesia Raya, general report by head of committee, the welcoming speech, pray, photo session, closing, and plenary session.
In the second day, plenary session, parallel session for oral presentation and poster presentation, and the next closing ceremony.
Ladies and gentlemen, let's open this event by saying Basmalah. Next, perform all traditional dance by Farmasi student.
Next, National Anthem Indonesia Raya. All audience are kindly requested to stand up.
considered. Next, general report by head of committee. We would like to invite Ms. Hartati Eswam Farm APT.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Good morning, ladies and gentlemen. The Honorable Director General of Health Workforce, Ministry of Health Republic of Indonesia, Mrs. Arianti Anaya MKM, Director of Poltekkes Kemenkes Gorontalo, Mr. Muhammad Anas Anasiru SKM Kemenkes, Vice Director of Poltekkes Kemenkes Gorontalo, Head of Center and Head of Unit within the Poltekkes Kemenkes Gorontalo, Head of Department within the Poltekkes Kemenkes Gorontalo, Branch Manager of Bank Syariah Indonesia, Chairman of the Regional Management of the Indonesian Pharmacists Association, Gorontalo Province, Chairman of the Regional Management of the Association of Indonesian Pharmaceutical Experts, Gorontalo Province, Head of Gorontalo Port Health Office, Head of the Center for Drug and Food Control in Gorontalo, all invited speakers, all sponsors, and all of participants of this conference. First of all, I would like to welcome you to the third International Conference in Pharmaceutical and Health Science 2023, which is being held by the Department of Pharmacy, Poltekkes Kemenkes Gorontalo, collaborating with the Pharmacy Student Alumni Association. This conference is the third conference held by Department of Pharmacy collaborating with the Pharmacy Student Alumni Association with the theme Current Challenges and Innovation in the Field of Diabetes Mellitus. This conference also received support from various parties that are the Association of Indonesian Pharmacy Experts in Gorontalo, the Indonesian Pharmacists Association, and sponsorship. For publication of articles, we have collaborated with national journals accredited by SINTA. Activity purpose. The main purpose of this conference is to sharing information and developing knowledge related to effort to prevent diabetes mellitus, the latest developments in diabetes mellitus in the community, product formulation, natural ingredients, and development of diabetes mellitus drugs. Speakers. The speakers for this conference come from several different countries and institutions in some regions worldwide. Professor Masaki Kita, PhD, from Nagoya University, Japan. Tyler J. Verisco, from the PhD from University of Houston, USA. Dr. Muhammad Ajmal Shah, from Hazara University, Pakistan. Dr. Siti Nur Fazilah Musain, University Teknologi Mara, Malaysia. Professor Ikatut Adiana, Master of Science, PhD from Institut Teknologi Bandung, Indonesia. Professor Andi Dian Permana, Master of Science, PhD, Apoteker from Universitas Hasanuddin, Indonesia. Participants. The number of participants is 356 participants from Indonesia, including lecturers, practitioners, and general participants, with 53 participants in the oral presentation and 31 title of poster. Time and place. This international conference is held on October 18th and 19th, 2023 and conducted virtually using Zoom meeting and broadcast live in YouTube Farmasi, Poltekkes, Kemenkes, Gorontalo. The method of this conference are plenary section and parallel section. This event is financed from DIPA Poltekkes Kemenkes Gorontalo in 2023. Next, we request the director's willingness to give us a welcome speech and to be able to officially open this conference. That's the committee's report. Thank you and wassalamualaikum warahmatullahi wabarakatuh.
Next, welcoming speech from Director Health Polytechnic of Ministry of Health of Gorontalo. We would like to invite Mr. Muhammad Anas Anasiru SKM Kemenkes to deliver a speech.
Okay, thank you MC. Okay. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Good morning. Greetings to all who attended to international conference with the theme Current Challenges and Innovation in the Field of Diabetes Mellitus. I, as the Director of Health Polytechnic Gorontalo, I am very happy and proud to be involved in this activity. The Director General of Health Purpose, Ministry of Health Republic of Indonesia, Mrs. Arianti Ana MKM, thank you very much for your willingness to open this international seminar. Honorable mentions to our 6 guest speakers from Nagoya University, Japan, University of Houston, USA, Hazara University, Pakistan, University Teknologi Mara, Malaysia, Institut Teknologi Bandung, Indonesia, and Universitas Hasanuddin, Indonesia, the academic community of Health Polytechnic Gorontalo, and all participation of the conference. First of all, I would like to thank you all for attending this very important event. Your presence here means a lot, and I believe that we all share the same goal, which is to improve our understanding and knowledge of diabetes mellitus and find innovative solutions to challenges we are faced in this area. Ladies and gentlemen, as a dangerous health problem that is often found in Indonesia society, diabetes mellitus is one of the focus of the Ministry of Health of the Republic of Indonesia, which must receive treatment as early as possible to avoid various effects in the future. One of the steps public to get treatment is early as possible is to know and be sensitive to further things related to diabetes mellitus. Following up on this, the Pharmacy Department of the Health Polytechnic of the Ministry of Health of Gorontalo held an international conference with the theme Current Challenges and Innovation in the Field of Diabetes Mellitus. We hope that this event will provide new insights, spark creative ideas, and inspire us all to continue contribution to global efforts to treat diabetes mellitus. May this event run smoothly and give benefit for all of us. The honorable audience, I would also like to thank all the speakers, participants, and organizers who have worked hard to make this event early. With the hard work and cooperation of all of you, this conference would not have been possible. Thank you for attention, and may this international conference be a productive means for the exchange of ideas and encourage collaboration in the field of health. Finally, welcome to the International Conference, Challenges and Innovation in the Field of Diabetes Mellitus. Let us move forward together in facing these challenges and executing innovation that have benefit for the society. That is all, and thank you for your participation. Wassalamualaikum warahmatullahi wabarakatuh.
Thank you very much for the speech.
Excellencies, ladies and gentlemen, before we come to the main session, let us start this by praying first. It is going to be delivered by Ms. Saskia Hairunisa, MD Farm. Time is yours.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Ladies and gentlemen, let us bow our head for a moment, pray to God Allah Subhanahu wa Ta'ala, so that our international conference can be precious to all of us. I will lead this prayer based on the teaching of Islam, and those who are not Muslims, you are pleased to pray according to each of your beliefs. Oh Allah, today Wednesday, October 18, 2023, in this beautiful place, we gather here to bring about an third international conference in pharmaceutical and health science. The theme is current challenge and innovation in the fields of diabetes mellitus. Make this international conference as a useful science assembly, as a medium of sharing useful ideas, knowledge, and experience of scholars, researchers, and students of various disciplines. May the conference we organize today benefit to our lives, broaden our knowledge, shine our ideas, and lead us to be successful, productive person, which in turn will boost dignity of our nation. Oh Allah, quiet and bless our hearts and our minds with the light of your guidance. Impart your supreme wisdom upon our activities. Help us to speak our minds clearly. Help us to listen to each other, respect each other, love each other, so that we are included to the blessed person. Oh Allah, protect us from uniting the temptation, show us the right path and give us knowledge and strength to perform good things equally, so as and make it clear the bad things and give us knowledge and strength to avoid them. Oh Allah, you are the one who can fulfill our doa. Wabillahi taufiq wal hidayah. Wassalamualaikum warahmatullahi wabarakatuh.
Those are the agendas we have presented to you this morning as we have finished our opening ceremony. Before we start the plenary session, may I ask your preferred time to virtual photo session?
Start from first slide. Okay, second slide. Third slide. Fourth slide. Okay, finished. Finally, from the deepest of my heart, I do apologize for our mistakes in presenting this event.
Thank you very much for your kind intentions and thank you very much. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Next, let's enjoy the alumni vocalia performance from Giang Wulandari Tahala AMD Farm.
Raise a cup up for all my day ones. 2 middle fingers for the haters. Life's only getting greater. Straight up from nothing we go. Higher than the highest skyscraper. Now literally we measure. The proof is in the paper. We put the good in the good in the good life. We put the good in the good in the good.
Before we start the plenary session, may I ask your preferred time to photo session? Thank you. Excellencies, ladies and gentlemen, now we come to our main program, plenary session, who will be led by our moderator, Mr. Muhammad Arif Ismail M.A. Teso. And the keynote speech is Dr. Muhammad Ajmal Shah from Hazara University, Mansehra, Pakistan. Mr. Muhammad Arif, time is yours.
Okay. Let me audio. Right. Is the audio working? Hello. Dr. Muhammad Ajmal Shah, I can see you there. Is my audio
Yeah,
okay?
Assalamualaikum. Assalamualaikum and good morning.
Good morning. Right.
Waalaikumsalam warahmatullahi
Nice
wabarakatuh.
to hear from you. Okay. Right. A very good morning to ladies and gentlemen. Welcome again. The second time, I'd like to say, the third International Conference on Pharmaceutical and Health Sciences 2023. My name is Arif, and I'm your moderator for this very first plenary session. On behalf of the committee, we are delighted to state that we have a very good lineup of speakers today. But just before we get into the presentations, allow me to briefly introduce you to our first speaker today, Dr. Muhammad Ajmal Shah. I hope I pronounced your name correctly. Currently working as an assistant professor at the Department of Pharmacy, Hazara University, Mansehra, Pakistan. He holds a PhD degree in pharmaceutical sciences from Prince of Songkla University, Thailand, completed back in 2017. Dr. Shah is a registered pharmacist and academic researcher. Is an active member of the American Society of Pharmacognosy, Phytochemical Society of Europe, American Society of Microbiology, and Society for Medicinal Plant and Natural Product Research, Germany. His recent study explores bioactive multifunctional compound from nature. Now, ladies and gentlemen, I would like to invite Dr. Muhammad Ajmal Shah to deliver his presentation about green extraction and enrichment of berberine. Over to you. Dr. Muhammad Ajmal, can you hear me okay?
Yes, please. Just a moment.
Right. Okay, take your time. Over to you.
Can you see my screen?
Yes, I can see that very well.
Okay. So thank you so much for your introduction. Actually, this is a very early good morning from Pakistan. It's around 6 past 15. So I had to wake up early for this presentation. First of all, I would like to be very thankful for the organizer and the organizing committee who invited me on this auspicious occasion to discuss some of my research outcomes. And I would like to proceed further for my presentation. So the title of my talk for today is Green Extraction and Enrichment of Berberine, and it is a kind of innovative approach in phytomedicine. So my name is, as the moderator told, that my name is Muhammad Ajmal Shah, and I'm a pharmacist. And I have done my PhD in pharmaceutical sciences from Prince of Songkla University, Thailand. And currently I'm working as an assistant professor at Department of Pharmacy, Hazara University, Mansehra, Pakistan. So my presentation for today will cover some important aspects in phytomedicines. The first one is about the green extraction. So I will discuss that what is the green extraction and how we can take benefits from the principle and the procedure of green extraction in preparation of phytomedicines. I will discuss the principle of the green extraction, the procedures of the green extraction, and then I will also discuss about the role of the pharmaceutical excipients as an extraction solvent, that how we can use the bioconsumable and non-toxic pharmaceutical excipients are the solvents as an extraction vehicle for preparing various phytomedicine. And in the last, I will give an example of our recently developed berberine enriched extract from Berberis lycium root bark, and I will show you our results from our research projects regarding the bioequivalence study of the berberine rich extract, which we have prepared by green extraction process. And then we compared this green berberine rich extract, which is quite economical in comparison to commercially available berberine that is quite expensive. So let's go to the first slide, that is about the herbal extract. So nowadays the alternative medicines or the natural medicines have more attraction in our therapeutic systems, because we think that the herbal medicines are almost free of side effects because of their simple uh, nature, and they are from nature. So of course, most of the things which Almighty Allah have been uh, granted us have more benefits, and of course they are mostly in any area or in any region, they are mostly locally produced, so they are low cost, because like we don't have the duties for imports and something like that. We actually incorporate the local wisdom to prepare these kind of preparation and products. And due to their diverse phytochemical profile, they have a lot of um, biological activities, and most importantly, they show synergistic activity because of the complex and diverse phytochemical profile. But of course, they have some disadvantages too. Like if you are using the extract or the natural drugs, sometimes they have unsatisfied appearances. So there is a problem for patient compliance. And sometimes they don't have the desired stability and efficacy, which is, of course, needed for a pharmaceutical product. However, these problems can be overcome by different pharmaceutical strategies. So I will proceed further how we can prepare a herbal extract by green extraction. So before proceeding for the preparation of a herbal extract by using the green extraction procedure, we should know about the green extraction process. So the green extraction process actually that kind of extraction of phytochemical compounds that use renewables resources and alternative solvents. These two are very important points. Like the first one is uh, the raw material which will be used in the extraction process should be renewable. Like it should not be diminished from the globe. So of course we have to use that part of the plant which should be renewable, like leaf, like flower, like fruit, because it can be regrown in other seasons from the same plant. And another most important point is we have to use the alternative solvents. Alternative solvents means those solvents which are bioconsumable and which are non-toxic, and of course they are considered as a green solvent. Um, in this process, we have to ensure the safety and the quality of the extract, that we should have a standardized extract that should have a safe level for the consumption. And finally, the energy consumption during the extraction process should be as minimum as possible. So if we will reduce the energy consumption and the time consumption in the extraction process, so ultimately we will have the final cost of the product within the limit of the patient. Here are the 6 principles for green extraction. You can see the first one is the renewable resources, as I have already discussed in the previous slide, that we should select that part of the plant that could not damage the whole plant, that could not damage the whole plant from the globe. So, of course, some parts like leaves, flowers, fruits, and byproducts are waste of the vegetables and fruits. They can also be used as a source for the extraction. So the first thing we have to think smartly, that we should select that part of the plant which can be renewable, like the plant can give us in every season. And the second thing is, like, we can also use the byproducts, those products which we can get from the plants as a byproduct or as a waste. So we can use these waste and these byproducts for their incorporation into phytomedicine. So in this way, we can convert the waste into some beneficial product. The third point is about alternative solvents. So regarding the safety and regarding the word green, we have to think smartly about the green solvent. We have to use the alternative solvent. Instead of toxic organic solvent, we have to use the green solvent. The green solvent means we should be green for both the consumer and both and for the environment. So we have to use those solvents which are safe. For example, we can use the most safer one is the water, but of course there could be a problem of solubility, polarity. So sometimes we have to extract the non-polar compounds. So for those, we can use some oil. We can also use alcohol because it is a green solvent and it is a bioconsumable. And regarding the chemistry of alcohol, it can dissolve both polar and non-polar compounds. So, and the fourth important point is our process and protocol should be in such a way that there should be minimum steps for the extraction process. So in this way, we can reduce the energy consumption, and like we can reduce in the fifth point, we can reduce the unit operation. And the sixth and the most important point is the overall process or the material which we are using in the extraction process should be biodegradable. Because if they will not be biodegradable, so after completing our extraction process and whenever we will throw it away as a waste, and if they are not biodegradable, so they can be harmful for the environment. So we have to think about this too. So now we have to proceed that which should be the more suitable method for green extraction of phytomedicine or herbal medicine. So in our experience, the most simple and beneficial and economical method is microwave assisted extraction. However, there are many other extraction procedure which are considered to be the green, like the supercritical fluid extraction. In that method, you need a very sophisticated uh, equipment in which there should be in which the carbon dioxide is used as a solvent in its supercritical fluid state. Like the equipment has such a system that it exerts so much pressure that it converts the carbon dioxide into a liquid state, and then that liquid carbon dioxide is used as a solvent for extraction, but that is very expensive. Those are very expensive procedures and very expensive equipment. But for the lab scale and for the small scale, The most simple and the common method for green extraction is microwave assisted extraction. Why we select microwave assisted extraction? So before proceeding toward the microwave assisted extraction method, we should know the mechanism of the heating mechanism in the microwave assisted extraction. Actually, in microwave, if you think normally, whenever we are using microwave in our kitchen, so the same microwave we can use for the extraction process, and the phenomenon of the heating is different from the conventional heating. Like in the conventional heating, you have to give the heat from outside, but in the microwave, the heat is generated inside the equipment because of the ionic conduction and dipolar polarization due to the generation of microwave field. So actually when you turn on the microwave field is generated inside the equipment and it increases the kinetic energy of the molecule of the material which you have put in your pot and you want to heat it up. And finally, your container is cool while your food or something inside for the extraction is heating up, and it takes just like few seconds or few minutes for heating up. And heating is actually important for the extraction process. So here in this slide, you can see that how the conventional heating and microwave heating works. So in the conventional heating, which normally we do on the gas stove or on the fire, so the heat is come from outside into the inside. And in the microwave heating, you can see that the heat is generated within the material and it comes from outside, from inside to outside. So if we compare the conventional and microwave processes, so in the conventional heating, actually we don't want to uh, heat what is getting heated in the conventional system. But in the microwave, this is an ideal process. And actually, if I would like to explain the mechanism, like if we put some plant powder in the solvent. So first, what happened actually the molecule of the solvent, the dipolar moment of the molecule of the solvent is increased up, and due to the increased up in the moment, the kinetic energy is increasing, and ultimately it increase or generate the heat within the solvent system, and it ruptures the plant tissues, the plant powder tissues, and extract out maximum amount or concentration of the phytochemical. So this was the mechanism of the microwave heating or microwave extraction. It is actually low energy consumption if we further compare it with the conventional heating system. So in the conventional heating system, we need more energy and more time for the heating process, but in microwave heating, microwave extraction, we need less time and less energy. Now, in the next part of my presentation, I would like to discuss about the alternative solvents. So we have already discussed about the process, and we have explained that what are the benefits of the microwave extraction system or microwave heating as compared to the conventional one. And now we will discuss about the alternative solvents. So pharmaceutical excipient. I'm sure most of you are the pharmacist in the conference. So you know well that every pharmaceutical product has two parts. One is active part and another is non-active part. So the active part is actually the active pharmaceutical ingredient or the generic molecule of the drug, whereas the non-active part is normally known as a pharmaceutical excipient, which actually giving the shape to a specific dosage form. So what do you think? We can use the pharmaceutical excipient as a um, as a solvent for extraction? Yeah, of course, because these are bioconsumable, these are safe, and they can be used because of a wide range of polarity. They can be used in as in a solvent for the extraction. I would like to quote an example here um, for the incorporation of pharmaceutical excipient as a solvent for extraction in this study, which have been performed in our supervisor lab previously. So you may know this fruit, Garcinia mangostina, which is normally called as mangosteen. We don't have it in Pakistan, but in Thailand, Malaysia, Indonesia, and all other temperate zones, this fruit is very popular and very abundant in those regions, and it is very delicious too. So our colleagues, they worked on this fruit. Actually, uh, they use they uh, they use the byproduct of this fruit. Like the inside, the white part is the fruit which is edible, and the outer reddish shell is the waste product or the byproduct of this plant, which is normally to be throw away. So actually, they use the shell or the outer red covering of the fruit, which is normally a waste. They study it, its phytochemistry, and they uh, identify the compound in this shell, which is alpha mangosteen. You can see the structure of the compound in this slide, and they found that this compound is very potent antimicrobial, anti-inflammatory, and most importantly, anti-acne effect. You know, the acne vulgaris is a very popular organism, microorganism, bacteria that cause acne on the skin. So it has a very good anti-acne effect. And they have formulated several products and formulation from this uh, waste of the fruit based, and the therapeutic activity of all those products is based on the concentration and activity of the alpha mangosteen, which is a marker compound of this plant. So you can see that alpha mangosteen has been reported for antioxidant, for antimicrobial, for anti-inflammatory, and wound healing activities. In this slide, you can see the solvent for extraction of alpha mangosteen. In any extraction process, either you are performing by conventional extraction or classical extraction processes, or you are performing the green extraction process, the most important thing is the selection of the solvent. Because in extraction process, you have to select such a suitable solvent which should have the similar polarity is having by the phytochemical. So for this process, you have to screen your plant powder, or you have to extract your plant powder on a small scale in different organic solvents to check the polarity and to check the yield of extraction and to check the efficiency of your extraction process. So what they did, they actually extracted the alpha mangosteen from uh, Garcinia mangostina by succinate extraction with various solvent, and they found that dichloromethane, which is an organic solvent, has more alpha mangosteen content percent weight by weight in the dry extract as compared to ethanol. hexane, and water. So it means that alpha mangosteen is more non-polar compound, and that's why it dissolved more in the most non-polar solvent, that is dichloromethane. This is maybe there is a question can be arised in your mind that we are talking about the green extract or the green extraction, so why we are using the organic solvent? The reason is you have to do some pilot study for checking the polarity of the compounds which are present in your extract, so you have to know, and then you later on you can compare the polarity of these organic solvent with some available with some available pharmaceutical excipient, with the polarity of the pharmaceutical solvent. So first they have checked on the pilot scale, they have checked that what could be the most suitable solvent for the extraction of this compound, and later on they have compared the polarity of the some of the alternative solvents which we used as an excipient in the pharmaceutical preparation with that of the best solvent they have chosen, like the dichloromethane. So they found that some solvents uh, which should be non-toxic because they are alternative solvent and they are pharmaceutical excipient. So they found that the polarity of the pharmaceutical solvent or pharmaceutical excipient, like for example, you can see they have checked in mineral oil, isopropyl myristate, stearyl alcohol, cetyl alcohol, propylene glycol, and they found that isopropyl stearyl alcohol and cetyl alcohol, based on the uh, solubility in water, you can see in the third column solubility in water. So the solubility in water factor for the stearyl alcohol and cetyl alcohol is almost equivalent to that one of the dichloromethane, as the dichloromethane has 9.55 solubility in water factor, and similarly the stearyl alcohol and cetyl alcohol has 8.9 and 8.94 respectively, and also the isopropyl myristate is around 8. So based on the solubility in water factor, they found that these three alternative solvent are pharmaceutical excipient: isopropyl myristate, stearyl alcohol, and cetyl alcohol are comparable to the dichloromethane. So it means that the dichloromethane, which is a toxic organic solvent, can be replaced by the cetyl alcohol, stearyl alcohol, and isopropyl myristate. So further they proceed for the green extraction of Garcinia mangostina, and they use the cetyl and stearyl alcohol, which are normally the pharmaceutical excipients and used as a cream base in several pharmaceutical preparations. So they use that these cream base like the stearyl alcohol, cetyl alcohol, and isopropyl myristate for the extraction of the Garcinia mangostina, and they actually just did one step process. And so here they could reduce the unit operation. They just took the powder of the plant, the powder of the shells of the Garcinia mangostina fruit, and they melt the stearyl and cetyl alcohol, and they added the powder, and then they just simply extracted with the microwave extraction, which just took 1 or 2 minutes. And after that, they standardize these extract by using HPLC, because standardization is very important. Nowadays, maybe you have seen there are many natural and alternative products in the market, but there is a major concern of their quality and their efficacy. And the quality and efficacy of any product actually based on the concentration of the active pharmaceutical ingredient or active phytochemical in the product. So standardization for the herbal product or for the phytomedicine is very important. And the most reliable method for the standardization of the herbal product is high performance liquid chromatography, that is known as HPLC. So they, after their extraction of Garcinia mangostina shell powder in sterile alcohol and isopropyl myristate, they have tested these extract by HPLC for the quantitative analysis of alpha mangosteen, and they found that you can see dichloromethane is still on top, but the results in isopropyl myristate, the yield of extraction is more better than dichloromethane, and also the content of alpha mangosteen are more in isopropyl myristate in comparison to dichloromethane. And you can also see the in the last two row, the amount or the concentration of alpha mangosteen in sterile and cetyl alcohol, that is almost 1.05 and 1.07, and that is quite good, better than dichloromethane. So these results validate the hypothesis that you can check the extraction yield and the extraction of the phytochemical and the concentration of phytochemical by using normal classical organic solvent in the pilot study. Then you have to compare the polarity of the polarity of the compounds and phytochemicals found in your extract. Based on the polarity or maybe the water solubility content, you select some you have to select some alternative solvent and pharmaceutical excipient in comparison to that studied organic toxic solvent. And of course, then later on you can compare and you can proceed further. If you are going to commercialize your product, you can proceed with these pharmaceutical excipients because these are more safer than the toxic solvent. These are the structure of the acetyl and isopropyl alcohol. So you can also guess the polarity of the solvent or excipient from their structure. And this is the microwave extractor in the lab. You can see this principle of this microwave extractor is actually the similar to the household microwave, but the capacity is more larger. And these are some of the products which our colleagues have been prepared by using green extraction or microwave extraction for Garcinia mangostina. These are the HPLC chromatograph. So that was just an example, and I would like to share the knowledge that how we can modernize the herbal medicine, how we can modernize the phytomedicine. So this is the way. There are two important ways that the process means the extraction of our phytomedicine or the preparation of our phytomedicine should be green. And another point is our product, like any product which we prepare from our natural resources, if we are using in our alternative system of medicine, should be well standardized. Like we should know the exact phytochemical profile, and we should know the quantification of the active compound by using analytical techniques like HPLC. Another most important part and the last part of my presentation is how we can find the alternative way to pure phytochemical, because normally in phytochemistry when we are working with certain medicinal plants and phytochemical compounds, so phytochemical isolation is an important step whenever we are studying some natural sources and plants for their biological activities. We have to isolate and purify uh, the marker phytochemical compound which is responsible for the biological activity of the plant. And in most of the cases, the concentration and amount of the phytochemical is very less. So that's why we cannot commercialize that compound from the natural source because of the very low amount of the compound. But we can just only perform the in vitro studies, and sometimes even we don't have enough amount of the compound isolated that could be used for in vivo studies or in animal studies. So we have found a way to resolve this problem, and that is the preparation of active constituent enriched extract, or you may say active constituent enriched standardized extract. So we are using the same method, green protocols, for the preparation of such extract, and we hypothesized and actually we confirmed in several studies that our green enriched extract are bioequivalent to the pure uh, phytochemical, which are sometimes to be very hard to isolate, or sometimes they are very expensive in the commercial market. So here is an example of berberine. Maybe you know all berberine is an isoquinolone alkaloid, and it is found in many plants, including Berberis lycium. So based on the marker potential and the concentration, the major source of berberine is Berberis lycium. I don't know if it is available in Indonesia or not, but it is abundant in Pakistan here, mostly in our northern areas like in Himalayan region. We have this plant, and this is very this plant has a great potential for its medicinal uses due to this compound and some other compound which are present in this plant. So until now, this plant as well as the berberine which have been identified from this plant, and it is more abundant in the root. So it has been reported for its anti-diabetic, antimicrobial, anti-cancer, neuroprotective, wound healing effect, and for its immunomodulation activity. But the problem with berberine is there are two ways to get berberine. Either you have to isolate from this plant, or you have to buy the commercially available pure berberine. And in both cases, it is very expensive. Like for isolation, if you will isolate from this plant, you will have to use a large amount of toxic organic solvent. You will need uh, a long process and time taking process for the isolation, because in isolation normally we have to do some repeating column chromatography and other chromatographic technique for the purification and isolation of our final compound. So it is like the final cost is beyond the patient limit. And the commercially berberine is very expensive. I think few milligrams are only available in few $100s. So although this compound has a very good bioactivity potential, but due to its cost, it is impossible to be incorporated in a pharmaceutical product. So what we have done, we actually thought that how can we use the local resources, the Berberis lycium, to prepare a green berberine enriched extract, which have uh, same or which have bioequivalent potential to that of commercially available berberine. So based on our literature study, one of my PhD student is has worked on this project, and based on the literature search, we found different sources in the literature for berberine, and we have selected Barbarius lycium because of its berberine content. You can see that the berberine content is 13.39 in the extract, ethanol extract of the Barbarius lycium. So, and also this plant is very abundant in our area, so we have selected this plant for our study. This is the complete protocol or complete procedure that how we processed the Berberis lycium root bark powder for preparing our berberine rich extract. So you can see in the first image that here are the drying process. Here is the drying process. We have peel off the bark from the root, and then we washed, dried at certain temperature, and then we have proceeded for the microwave assisted extraction. Actually, there are two or three important points which you have to be keep in mind while you are doing your extraction by microwave. The first thing is the power of the microwave. So normally the household, and in this process we also use the household microwave, which normally we use uh, in our kitchen. So the frequency of the power and the power of the maximum power of our microwave was almost 1000 watt. And another important thing is you have to optimize the microwave extraction condition in a set of experiments, that what should be the best condition for extraction in microwave assisted extraction. So you have to check on different power, different timing, different amount of solvent, and different ratios of the powder with the solvent. So for us, we got these condition the most optimum for our extraction, that we used 300 gram of our powder in 1500 milliliter of ethyl alcohol, and the process was repeated 2 times. And we turn on the microwave for 6 minutes, and then we turn off, open the door to remove the fume of the alcohol, ethanol, and repeat it again for 6 minutes and then for 6 minutes. So we have repeated the extraction for 3 times, and we have put the powder in the solvent within the microwave for 6 minutes in each cycle, and we repeated this process for 3 times. And once we completed the 3 cycle of the extraction, we have filtered the extract, and then we have repeated the same process by adding the fresh ethanol. After getting the final pool of extract, we have filtered the extract, and then we have proceeded for the enrichment of our extract. So for this purpose, we use a special column that is called Dion HP20 column. It is a polymer, a hydrophobic kind of chromatographic column. And the flow rate, we keep 20 ml per minute. And the solvent which we used for elution was 30% to 70% ethanol in water. And finally, we got, after evaporation of the solvent, we got our berberine rich extract. Then we have checked our berberine rich extract. We have analyzed it for the quantification of the berberine, that how much berberine content are present in our rich extract. So in the crude extract means the extract that we got after filtration, that could be called a crude extract, because this extract have just undergone through the microwave extraction, and we haven't passed it through the column yet. So this is the crude extract in the beaker. So in the crude extract before enrichment, the amount of the berberine or the concentration of berberine was 32.42%, whereas the berberine rich extract, like after passing through the column, the Dion column, that actually enriched that column, removed the other material, that was a single step process, and that used a biodegradable resin, Dion, and water and alcohol as a eluting solvent, which are green, and that actually remove the other content, and in some fractions we got more amount of the berberine. So this rich extract gives us 58 means nearly 60% of the berberine. And also we have a very high yield of the extract. So we hypothesize that this berberine rich extract that contain 58.42% of berberine could be almost bioequivalent to the pure berberine which is available commercially, but that is more expensive. So we have studied this berberine rich extract in comparison to the available commercially available berberine for different phyto activities, for different biological activities. You can see in this slide, this is one of our study of this project, and we have compared our green berberine rich extract to pure commercially available berberine for its potent anti-inflammatory activities. We have performed this study in animals, in rats, and we found that our berberine rich extract has comparable bioequivalent effect to the commercially available uh, expensive berberine, and we have published this study in Journal of Food Biochemistry, which has almost 4 impact factor. In the next study, the same berberine rich extract, we have studied our green berberine rich extract in comparison to commercially available more expensive berberine for its neuroprotective potential. And we found that in all set of experiments, like in behavioral studies, because we have performed different kind of different types of experiment in this study, like we have done behavioral studies, we have done biochemical studies, and we have also done some molecular studies at gene level to check the effect of berberine rich extract as well as berberine on neuroinflammation. And we found that our berberine rich extract, which we have prepared by green process and that contain 60% of nearly 60% of berberine, is bioequivalent or comparable, has comparable effect to the commercially available berberine that is more expensive. So, and we have we have published this data in one of the popular Q1 journal, Environmental Science and Pollution Research, that has impact factor of 5.8. We have also studied the antimicrobial comparison of berberine with the berberine-rich extract. Before proceeding with this slide, I would like to tell you that berberine is very uh, potential compound for therapeutic use, and I have studied in the literature that there are some clinical trials still on this compound, but the problem with this compound is it is very expensive. So that's why we are trying to find out more economical and more cheaper way to find some alternative for the berberine. So the berberine was already reported in the literature for its antimicrobial activity. That's why we took it as a standard, and we compared our berberine rich extract that we have prepared by green process in a set of antimicrobial experiment. We have use two positive control, that is azithromycin and ciprofloxacin, and we have checked the activity, the antimicrobial activity of berberine-rich extract in comparison to berberine against several important microorganisms like Proteus vulgaris, uh, Pseudomonas aeruginosa, Staphylococcus aureus, pneumonia, MRSA, and Salmonella typhi, that is a causative agent of typhoid. And we got in the result, we got that our berberine rich extract has almost comparable and bioequivalent results to the berberine, commercially available berberine. Along with the antimicrobial activity, we have also performed the combination effect of our berberine-rich extract in comparison to berberine, along with the antibiotic to check the synergistic activity. And you may see in the results that in combination, the berberine-rich extract and berberine have almost bio have almost similar results and more potent than their individual effect. So if in the future, if there could be a product from the berberine-rich extract and you could use it along with the antibiotics, so it will give you a higher antimicrobial effect rather than using them as individual therapy. So it can reduce the dose of the antibiotics if we can use it along together with the antibiotic, and it can also overcome the problem of antimicrobial resistance. In another set of experiment, you can see in this slide, we have also checked the anti-cancer comparison of berberine and berberine-rich extract, because our claim was, and our claim is that the berberine-rich extract that we have prepared by green process is almost bioequivalent to the commercially available berberine. So previously, the berberine has been reported for anti-cancer activity. So we have also checked, and you can see in the results that the berberine and berberine-rich extract on human MDA-MB231 cell line have same anti-cancer effect. Here is the slide that shows the anti-cancer comparison of berberine and berberine-rich extract in another cell line, that is MDA-MB, and you can see from the graph that both berberine-rich extract and berberine have same inhibitory effect. We have also checked the cytotoxicity in the assay, and we found that the IC50 value of berberine and berberine-rich extract is almost equivalent, but in A549 cell line, the IC50 value of berberine-rich extract is more than the berberine, whereas in the MDA-MB cell line, the IC50 value of berberine is equivalent to berberine-rich extract. We have also checked because normally, traditionally, this plant is used for the wound healing purpose in our area. So, and this is the berberine is also reported before for the wound healing process. So we have also performed the wound healing potential of berberine-rich extract in cell migration assay. And from the results, you can see these are the results of BRE, berberine-rich extract, and these are the results of wound healing results of berberine. So in both the results, we have noticed that berberine-rich extract have almost bioequivalent wound healing potential is having by the berberine. In this slide, you can see the anti-diabetic, because the conference theme is anti-diabetic. So I have also included this data, which we have recently performed, but we did not publish it yet. So I will share some of the results of the anti-diabetic potential of berberine-rich extract in comparison to berberine. As I told you that berberine is previously reported for its various biological activities, including the anti-diabetic activity. So we also thought that the berberine-rich extract that has almost bioequivalent effect to the pure berberine, commercially available berberine. So in the same way, we also thought that it could have the same anti-diabetic effects. And we have performed this study In streptozotocin, nicotinamide induced diabetic rates, and we treated different groups in different doses, and we have compared our results with a positive control, that is metformin, and we found that the blood glucose level lowering effect of berberine rich extract in comparison to berberine is quite satisfactory, and they have almost same bioequivalent or hypoglycemic effect. In the graph B, you can see the serum insulin level. The black one is the normal control, the red one is the streptozotocin, like the diabetic rates, and the rest of the three bar, you can see the blue one, the pink one, and the brown one. These are the treated group. So in the last one, you can see that berberine enriched extract has almost similar effect to the, which is significantly similar. You can see both the effect of berberine and berberine rich extract are significantly similar and are almost bioequivalent to metformin, that is a standard anti-diabetic drug. And in the graph C, you can see the effect of berberine rich extract and berberine on the glycated hemoglobin. Glycated hemoglobin is an important indicator that indicate the level and intensity of the diabetes mellitus in diabetic patient. So you can see in diseased rates, the red one, streptozotocin, the glycated hemoglobin is very high, but the the treated one, and interestingly, you can see the one of berberine enriched extract treated rats have a very lower glycated hemoglobin value than those treated with berberine and metformin. So these results indicates the very potent anti-diabetic and bioequivalent anti-diabetic effect of berberine-rich extract, which have been prepared by green extraction method in comparison to the commercially available expensive berberine. These slides show the hypolipidemic potential of berberine-rich extract in comparison to pure commercially available berberine. In the first one, you can see the level of HDL, high density lipoprotein. Um, in streptozotocin induced diabetic rat, the HDL level is quite lower, and it is increased by metformin, berberine, and berberine enriched extract gradually. You can also see the effect of berberine rich extract, berberine in comparison to metformin on serum level of cholesterol. They have reduced this serum level of cholesterol at a significant level. You can also see the serum level of triglyceride, and interestingly, berberine rich extract have significantly reduced the serum level of triglyceride in comparison to berberine and metformin. And in the same way, you can also see the serum level of low density lipoprotein, which is interestingly and significantly reduced by the berberine rich extract in comparison to berberine, metformin. And finally, based on all these results, we have, before proceeding for its publication, we have proceeded for its intellectual property rights. I would like to suggest, if someone here is in this discussion is working on phytomedicine or natural medicine, whenever you have some interesting results regarding some of the extraction processes or biological activities of your project. So before publishing your data, you should proceed for uh, filing it as a patent to the intellectual property organization of your country or maybe internationally. So we had interesting results So that's why we were proceeded for filing a patent on our studies, and we have filed the patent on green preparation of berberine-rich extract and bioequivalent studies to be used as a phytopharmaceutical alternative of commercially available berberine to the intellectual property organization of government of Pakistan, and we got a patent filing number, and hopefully we will get this patent because it is now under review. And after getting the intellectual property, we are in contact with several pharmaceutical industries here locally, and we think that possibly we can commercialize our berberine rich extract in different formulations, like for wound healing process, for wound healing treatment, we can prepare some wound healing dresser from which can be enriched by the berberine rich extract because of its very potent wound healing activity, antimicrobial activity, and anti-inflammatory activity. We also think that we could, because the plant is edible, so we can prepare some oral formulations for their possible use in neuroprotection, diabetes, and some other disease. But before proceeding for the product formulation, we will and we are doing some safety assessment and toxicological studies like acute toxicity studies, subchronic toxicity studies of our berberine-rich extract to ensure its safety for its therapeutic use. So here are the concluding remarks that once, if you are working with the natural products, so the first and important thing is you have to know the marker and major compound or the marker and major phytochemical of your plant or extract. Then after that, if you want to proceed, and you should proceed with the green extraction, you have to find a suitable solvent according to the polarity of the compound which are present in your plant or in your herb. Then you have to find the suitable extraction technique. You can use the classical technique for on the pilot scale for preliminary screening, but for the commercial purpose or for the large scale, the microwave assisted extraction is the most recommended one. Of course, you have to optimize once you have decided that you are doing microwave assisted extraction of your herb or your plant. You have to optimize the extraction technique or the extraction protocol, and then you have to compare the polarity of your compound with the green bioconsumable solvent or the alternative solvent or pharmaceutical excipient. And finally, once if you have a success uh, to make to perform its green extraction by using the pharmaceutical excipient, you can go toward a product by the green process. So I would like to acknowledge the efforts of my student, my PhD student, Miss Iram. You can see in the picture. She is my PhD student, and she will defend her thesis soon. And my boss, with him I have done my PhD under his supervision, Associate Professor Park Company Chayopakaran. He supported us in this project based on his expertise and provision of the lab facilities for some of the activities, especially for the standardization and enrichment of the berberine rich extract. We would like to acknowledge his efforts and his services. At Faculty of Pharmaceutical Sciences, Prince of Songkla University, Hatia, Thailand. And I would also like to acknowledge for the travel grant provided by the Phytomedicine and Pharmaceutical Technology Excellence Center to my PhD student for performing some of her work at the Faculty of Pharmaceutical Sciences. I would like to acknowledge the Center of Excellence for Molecular Biology, University of Punjab, Pakistan. They provided us the facility for anti-cancer activity and cell migration assay. I would like to acknowledge the Department of Biotechnology and Bioinformatics, GZ University, Faisalabad. They also provided us a generous support during this project. And this was all about our project. And thank you for your kind attention.
Right. So that is the presentation from Dr. Muhammad Ajmal Shah. Very insightful one. And I guess there are some scholars in here who would like to come up with questions and maybe some of the technical terms that they would like to clarify from your presentation, because we notice you use some of the abbreviation, particular. Right. So if there's any question,
Yes.
feel free. You can type it on the chat box, or you may request to speak by raising your hand first. Okay, somebody's raising hand. So I'll just go to Mr. Israfil first. Yeah. Okay, Mr. Israfil, over to you. Can you hear me okay, Mr. Israfil?
Yeah, please.
Yeah. Please. Dia kok nggak sorry.
Okay, right.
If you have some, I think there is some problem about
Yeah, now it's working.
about some technical.
Okay.
You can write your question in the chat box too. No problem. I can read
I
it.
guess we can speak now.
Right.
Yeah. Okay.
Sorry. Okay. Is my voice sounds clear?
Yeah. Yeah.
Okay, right. Hello, Dr. Muhammad Ajmal Shah. Are you okay today?
Yeah. Yeah. Thank you.
Yeah. I'm Israfil. That contact you for this conference some times ago, and thank you for your willingness to be a keynote speaker at our conference. And I know that in Pakistan, especially in Hazara, it's still very early. If I'm not wrong, it's
like I had to wake up for the series, you know.
Yes, yeah, that's 6 p.m. 6 p.m.
Yeah.
That's right.
Yeah. Yeah.
So yeah, welcome to our conference, Dr. Ajmal.
Thank you. Thank you very much for your continuous networking and connecting with me. And you are very active guy. You know, you are a good organizer.
Right. I'd like to continue my question,
Okay.
Doctor. Okay, right. But yeah, I'm interested in your discussion about berberine as the one most potential alkaloid to treat many diseases. Some literature has also said that this compound is very potential. If I look in literature and then in your slide, that it used in a wound healing effect, anti-inflammatory, anti-diabetic, and several other studies that have you described in your slide. We know that berberine is the one of the alkaloid compound that requires special technique to extract it, as well as for fractionation and also isolation process. Could you share to us what is the most appropriate extraction technique for this compound, including the most optimal solvent to extract this compound, as well as the isolation technique? That's the first. And okay, and the second is if we know how about the yield value when you isolate it with this method, and then regarding the effect of berberine how far this compound have been tested in your laboratory. I mean, it have been clinical trial testing, or how? Maybe that's
Okay.
my question.
Okay, so first, I think your first question is about the isolation of berberine, right?
Yes, right.
Okay, so this is not a new compound. This is very old compound, and it has been extensively studied by several researchers in the past, and different researchers have isolated this compound in different ways. But as for us, our objective was not to isolate this compound because it is available commercially nowadays. You can purchase it from different suppliers like Sigma and some other suppliers. The problem is the cost of the compound. This compound is very expensive. You know, if you want to purchase it, it is very expensive. So our intention and our objective was to prepare berberine rich extract. Like the product which we prepared is not fully pure compound, but it is a semi-purified extract that contain almost 60% of the berberine, which we have quantified by HPLC. And we hypothesized and we think that this berberine rich extract that we have prepared by green extraction process is almost bioequivalent, means have the same activity and the same concentration if we compare biologically with the pure commercially available berberine that is quite expensive. So for our process, we use the solvent system because we work on green extraction. So we try to keep everything green, you know, like we always select the green solvent. We don't use the toxic solvent. So in this process, we have used ethanol and water, and from 30 to 70% aqueous ethanol, we found this range the most suitable range for the extraction. And in this range, we got the maximum yield. Like before us, it was just reported up to 13%, but in our attempt, we got 32% in the crude extract, and then we further enriched it by using a resin column up to 60%. So the prime objective was not isolation, but it was just to prepare the enriched extract. This is a new concept that how you can use the natural sources as a sustainable source for preparing the phytomedicine by green approaches. So I think your question may be clear now, right?
Okay, that's very clear, Dr. Achma. Yeah. So and the last, I'd like to say thank you very much for your willingness to be a keynote speaker at our conference today, and hope this collaboration can be continued in
And I
in
am
future.
very thankful to you too, because you have contacted me and you were in continuous communication with me for the everything. I think you are a very smart guy.
Okay, thank you very much, Dr. Muhammad Achmar Shah.
This one
Nice to meet you today.
Thank you. Thank you.
Yeah. Thank you very much, Mr. Statil. Now we're going to the next question. Somebody's raising hand. Pak Arlan, Mr. Arlan. Over to you, Pak Arlan. Go ahead, please. One, two. Wow.
Thank you. Okay. Good evening. Good morning. Mr. Azmal, Dr. Azmal. Thank you for the opportunity.
I think there is some technical problem for
Thank you for the
because I can't
opportunity,
hear.
Doctor. Yes. Very
Getting
interesting
the sound
material.
now.
Permission to ask Prof. Doctor. In my
Yeah.
last thesis, I used the acid-base extraction. Okay, acid-base extraction method to extract alkaloid compounds. I got an alkaloid level in 15%. Saw the professor presentation, the chocolation method using microwave heating is a good method to extraction alkaloid. My question is, the chocolation method with the heating using microwave extraction or acid-base extraction method is most suitable for extraction alkaloid, doctor? And my second question, does it not affect the characteristic of the sample we use, prof, doctor? And also in my thesis, I using acid-base extraction using heating to constant temperature, but using sonicator. My third question, how about sonicator, doctor? Is the principle the same as microwave heating? Thank you.
Okay. Many questions. Many questions in one question, huh?
That's a good one.
Okay, you can hear me?
Yes, yes.
Yes,
Your
very
audio.
clear.
Your first question was about the isolation of your alkaloid in your thesis, right?
Yes, I use the acid-base extraction using to extraction alkaloid, and I got
Okay.
the 15% alkaloid, and I saw to
Which method you used?
piperin. I use a piperin, the standard.
No, which method you used for extraction?
Acid-base extraction. Acid-base.
Mhm.
I use
And you
acid.
have also performed ultrasonication method, right? You can hear me?
With using a sonicator to heating.
Yeah, you know the sonication. Sonication is also a green method because normally you don't need more energy and more time for the extraction. So this is the prime objective of green extraction, that there should be less time for the extraction. The amount of the solvent should be used should be less, and the type of the solvent which you are using is should be less. Your important question was about the degradation of the compound by microwave, right? So if we are using microwave assisted extraction, so there could be a chance of degradation of our compound or not. So I would like to clear about this question that the extraction time in microwave is very short. Like in our study, you have seen that the exposure of the plant material to the microwave radiation is for very short time, like for for a few seconds or for a few minutes. So we think that due to the short exposure of the extract in the microwave apparatus, there is no problem because we have compared this extraction process with other classical extraction processes like maceration, like percolation, like suckslit extraction, like sonication, and we found that after the extraction, we have checked the content by using HPLC. We have quantified the compound by using HPLC, and we found that there is no variation and there is no differences in the concentration of the compound. So we believe that the microwave assisted extraction, due to a short exposure of time and little amount of solvent and a little amount of energy, and of course the overall processes like economical, cheap, and least time consuming. So in the future, if we will have some opportunity, or if I will visit there in the next year for the conference, if they will organized in person. So maybe we can conduct a workshop on microwave assisted extraction, that how we can perform these extraction for different bioactive compounds.
Thank you, doctor. Thank you for the answer.
Okay.
My question.
Right. Thank you very much for addressing the question, Dr. Ajmal. Let's move on to the next one. Okay, so the next one is from my colleague. Yeah. Miss Priska would like to speak directly
Okay.
to you. Over to you, Miss Priska. Go ahead. Everybody, over to you.
Yes. Okay, thank you, moderator. Thanks for the chance. It's a very awesome explanation and research. I just want to confirm about the CN or NC in your slide explain or STZ as sebagai abbreviation about CN or NC and STZ in your slide explain.
Just a moment. Let me check.
Yeah, I think that'll be best if you can refer to the slide again.
Yeah,
The one with
just a moment.
okay. Take your time.
This
Yeah.
this one.
Oh, the one with graphs with the N. I thought the NC, MCE, GBE, things like that. This one? This, yes.
This is this is the normal control. CN is normal control.
Normal control. Wow.
And STZ is the
Normal control. Okay, right. Najma, are you still with us? Yes. Hello, Dr. Ajma.
you can hear me now?
Now I can hear you very clear. Yeah, go ahead. So NC is the
CN is the normal control. Like
normal control.
the rates are not diabetic. We are just keeping them as a normal rate, and we are just feeding them the vehicle, the solvent in which we have dissolved our sample. You are getting my point? So these are just the normal rate, and they don't have diabetes, and we are just only giving them the solvent, the normal saline or the solvent which we used for dissolving our sample. The STZ means the diabetic rats. So we have induced the diabetes by streptozotocin. This is a major diabetic reagent that you used for the induction of disease, streptozotocin. So these are the diabetic rats. The STZ rats are the diabetic rats, and they are untreated. And the other three are the treated. They are also diabetic rat. The MET is metformin treated rats. BBR is berberine treated rats, and BEE is the rich extract that we have prepared, berberine enriched extract treated diabetic rats. I hope that the question is clear now, right?
Okay, thank you.
Right. Just seeking for some clarification. Okay, let's move on to the next one. A participant is also raising hands. Mr. Yabes Oral Kololiu. If you can hear me okay. Mr. Yabes, are you with us? Okay. Right. If it seems like Mr. Yabes is not quite ready to deliver the question, I've got a student with me, Dr. Ajmal, and he would like to deliver the
Yes.
question to you directly. Yes,
Yes,
go ahead
please.
with your question. Yes.
Yes.
Try to speak.
Okay. First of all, I want to say thank you for giving me the opportunity. And let me introduce myself to you. My name is Yabes Orotolio, and my question is, how do we know the ability or potential of natural ingredients as medicine? Again, my question is, how do we know the ability or potential ingredients as medicine?
How do we know the ability of
Yes. If you can repeat your question.
My question is, how do we know the ability or potential of natural ingredients as medicine?
The ability and
how do
potential
we
of
know?
natural ingredients of
Yes.
nature. That's what I got.
I still didn't get his question that what does it mean?
The question is actually about how do you know the potency of natural substance to be developed as the
Okay,
process?
that's fine. I got it. I got it. Um, in my opinion, there are two ways. Like if you are talking the potency based on the biological activity, or if you are talking about the potency based on the concentration of the compound in the natural medicine. So these are two different ways. If you want to know the potency, the biological potency of the natural medicine, you have to perform some bioassay that for which purpose this medicine is using, and you can check. Like for example, if some natural medicine is using for antimicrobial purpose, so you have to perform the antimicrobial assay, and based on your result as a MIC value, minimum inhibitory value, or zone of inhibition, you can conclude that how much your uh, how much your natural medicine is effective. On the other hand, if you want to know its chemical potency, so you have to check the extract or the herbal medicine, you have to extract it out, and then you have to check it by HPLC that how much is the amount of the compound in your extract or in your medicine. Hope you got the answer, right?
All right, thank you.
Okay, thank you very much for the question, and thank you very much for addressing addressing the question, Dr. Ajmal. And there's also another question. Let's move on. Are you willing to take the next one, Dr. Ajmal?
Yeah,
You
yeah,
okay with
sure,
it?
no problem. No
That
problem.
sounds really good.
Time is
No
yours.
problem.
You can ask many questions.
Okay, that sounds really good. Next question comes from my colleague, Mr. Usman. Mr. Usman is a lecturer in the Pharmacy Department, Gorontalo Health Polytechnic, and he would like to speak directly to you. Yeah, go ahead, Mr.
Okay.
Usman.
Okay.
Okay, the committee is allowing us to speak.
I'll check. Okay, thanks for the opportunity. I just want to ask and question why do you choose the solvent dichloromethanol, ethanol, hexane, and water to extract the alpha mangosteen compound?
This one, right?
Apa dia bilang?
Okay, thank you for your question. You are talking about this slide, right?
Yes, yes.
Okay. When I was discussing this slide, I have told you that whenever you are optimizing, you are optimizing the selection of the solvent. Like if you are proceeding for extraction of any herbal medicine or any plant powder, you have to optimize the selection of the solvent. So of course, for the selection of the solvent, you have to proceed with the classical organic solvent because we already know the polarity of the organic solvent. So you have to extract on a small scale, you have to extract your plant powder with the classical organic solvent to know that what is the polarity of the bioactive compound in your extract. So once you get the idea that which solvent is most suitable solvent for extraction of your compound, then based on the polarity of that solvent, you can go toward pharmaceutical excipient or any other alternative solvent or green solvent that has the same polarity and same characteristic is having by the organic solvent. So this is just a preliminary pilot study for knowing the polarity and the suitability of the solvent for extraction. But for commercial use or for large scale, you have to compare the polarity or the similarity of the toxic organic solvent with the green or bioconsumable one. You got my point?
Another option, sure, for the solvent.
Yeah, sure.
Okay, so
Sorry?
he said, so he's actually sitting next to me, Dr. Ajmal. He said he got your point, but then there's a follow-up question. He's wondering about, is there any other alternative of solvents apart from dichloromethan? It's difficult to pronounce.
Yeah, of course, you can see in this slide, based on the solubility in water factor, diochloromethane has almost the same polarity as having by the sterile alcohol, by the cetyl alcohol, if you want a cream base, like the solid one. But if you want to use the liquid one, you can see isopropyl myristate is the pharmaceutical excipient, and they can be used for the same polarity purpose as having by the dichloromethane.
Okay, right. Thank you, Dr. Ajmal. So they're trying to seek for some clarification because my colleagues, they are sitting next to me and they do the experiments, extraction on daily basis. We've got a laboratory very close to us next to these buildings. So that's why. Okay.
And I'm sure one thing. I'm sure that maybe some of your questions are not well addressed, or maybe you don't have enough time for you asking many things. And also maybe there is a technical problem due to the sound or something like that. So here are my contact details. You can see my contact details. I have shared my WhatsApp and I have shared my email. So you can ask me in WhatsApp, is more convenient. You can ask me in WhatsApp, about any of your problem regarding if you are working on extraction or if you are working in some similar area in which we are working. So you can talk to me about collaboration, or maybe if you have some technical problem, I can guide you, no problem.
Right. I think the contact would be very helpful. Thank you very much for that. Really appreciate it, Dr. Ajmal. Yeah. The next question comes from a student, and she's ready to deliver the question. Over to you.
Okay.
Okay.
First of all, thank you for the time and thank you for the opportunity that given to me. My name is Nashwa Ole Aju, and my question is: can the microwave assisted extraction method be replaced with another method?
Sorry, please repeat your question.
Okay. Can the microwave assisted extraction method be replaced with another method?
The
Yeah,
microwave.
sure, you can. You can, but the problem is, we are living in a developing country and our labs are not well equipped with sophisticated equipments. There are many extraction procedures, like as I have discussed, you can use supercritical fluid extraction technique that need a very sophisticated, high quality extraction equipment in which we use liquid carbon dioxide as a solvent for extraction. There are many other methods like ultrasonication and the other methods, but this one, everyone has a microwave in the kitchen. And believe me, you don't need a very sophisticated equipment for this extraction. You can just use your kitchen microwave for extraction on small scale. So, and most importantly, there are some benefits or other extraction methods, like you need a very little amount of the solvent, you need a very short time for extraction, you need a very little amount of energy, and you don't need a very sophisticated equipment. So all these benefits actually prioritize for us the use of microwave assisted extraction. I hope I explained well.
Yeah, okay, thank you.
Yeah. Thank you very much, Dr. Ajmal, for addressing the question. Yeah. That's question number, I think, fourth or fifth. Yeah. Very helpful one. And we've just had a little discussion, Dr. Ajmal, me and my colleagues around here. It seems like the one you just mentioned about collaboration, it seems like we've got plenty of spaces to collaborate in the future regarding the research about green stuff, green medicinal, things like that, pharmaceuticals. And also, they're wondering about another thing, which is, exchange program. Let's say if the lecturer in here to go there and visit, the laboratory, working on research, and the other way around, researchers from Pakistan, from your university, to come to Indonesia to share their expertise, and also students. Would that apply to, student case or things like that?
I am very open for collaboration. And maybe you have seen in my CV, I have many publications in a very early career stage. That is the reason that I have collaboration throughout the world. I have collaboration with different scientists from different countries. And I believe in science, collaboration is very important. Recently, I have visited Islamic University, Makassar last month. I have visited there and I have personally participated as a speaker in their conference. And along with the conference, we have also signed an MOU with UMI Makassar. So for exchange you need to have MOU with both between both of the universities or institutions. But for training, for short training, or for research collaboration, or for example, if you need our expertise, so of course you can invite me in as a visiting researcher or visiting professor if you want some seminar or some training or some hand-on workshop. So it is manageable. We can do it. I think it's not a big problem, and it is good for both of us.
And oh, thank you very much for that. And I forgot to address one thing. Some of the colleagues, my colleagues in here, they really looking forward to pursue PhD study. Just wondering, would that be, any opportunity from Pakistan universities or scholarship that they can pursue for? What do you think?
Currently in the university where I'm working, we don't have like PhD program now, because this is like a new program. Pharmacy is new program. Before this, I was working at Government College University, Faisalabad for 4 years. But there are some organization like Comstack. We have the head office of Comstack in Pakistan, and we have one international center for chemical and biological sciences at University of Karachi, and they are very generous. They have some scholarship for the international students and like that. But in our case, if some of the student can want to visit us, so they can visit if they have some funding from their own university or country, like for the traveling and for the living here. The living cost is not so much. So if someone want to come for 1 month, 2 months, 3 months, or for some activity, although if they don't want to come, they can send us their samples for some analysis, or if they want our expertise, so I can visit in the future to some universities over there to train your student, to give lectures, to train them in the lab or something like that.
Yes,
But of course, there are some scholarships. You have to search about it.
Okay, right. That sounds really good. Research opportunity seems very promising. The one you've just explained. They're really looking forward for that. We're going to work on, the matter. Right. So, Dr. Ajmal,
Thank you.
so since we've got hundreds of students with us in this auditorium, they would like to, wave to you, actually, to greet you. If the camera can point out to the students, please. So we've got hundreds of students. We've got 2 floors buildings. This is the second floor, and tens of other students. They are sitting upstairs. They're waving at you.
That's very
They're
nice.
following your presentation. They're following the presentations.
I hope they have enjoyed. I hope they have enjoyed my presentation.
They do follow your presentation. They do like your presentation, I guess. Okay, right. So I've just got information from the committee, Dr. Ajmal, that the committee has tried to contact, the person in charge who was in touch, who represent you from the university to ask for the materials. But seems like the committee hasn't got the material, so we would like to request you to send your material after this presentation, if you don't mind.
Yeah, sure, no problem.
Okay. Okay, that sounds really good. And for the last thing, Dr. Ajmal, the committee would like to present a certificate of appreciation to you personally, and the
thank you so
certificate
much.
should appear on the screen. There you go.
Thank you very much.
Certificate of appreciation to Dr. Muhammad.
Thank you very much.
Ajmal Shah.
Thank you very much.
My first name
Thank
is
you very
also
much.
Muhammad, Dr. Ajmal. My full name
Yeah.
is Muhammad. Right.
Yeah.
Okay.
Thank you very much. Thank you.
Thank you. The last thing, the committee would like to say thank you very much, Dr. Ajmal, for your contribution.
I am also very
to this
thankful for
conference.
the committee and for the organizer that they have provided me the opportunity So I hope that in the future we will have the same collaborative activities, and we will conduct some more practical, like in person, or maybe some workshops, some guest lectures, and
Itu dia.
we can work together, no problem.
Right. Thank you very much. Hopefully we can connect again in the near future. Thank you very
Hopefully,
much, Dr.
inshallah.
Najmal. Assalamualaikum.
Inshallah. Walaikum salam. Thank you. Take care.
Thank you, Dr. Ajmal.
So I can lift now?
Yeah, right.
Okay. Thank you.
Thank you. Take care.
Tunggu, mau thank you dulu. Udah? Aku mau thank you juga. Okay, thank you very much to our keynote speech, Dr.
Belum muncul.
Okay. Thank you very much to our keynote speech, Dr. Muhammad Azmal Shah, who has gives us knowledge. May it will be useful in our daily aiming. And thank you for Mr. Arif Ismail, MAT.
Raise a cup up for all my day ones. 2 middle fingers for the haters. Life's only getting greater. Straight up from nothing we do. Higher than the highest skyscraper. Now a little league we measure. The proof is in the paper. We put the good in the good in the good life. We put the good in the good in the good life. We put the Cikini kegondang dia, ku cari begini gara-gara dia. Cikampek, Tasikmalaya, hatiku capek bila kota sepi. Remember when we still want your love, your voice, and your bright eyes. We're together in this love. Promise we'll forever. And now you left me alone. You find someone better. You would play with her. And one thing I know, you break my heart, break my heart, make me so down in loneliness. You left me when I did not. You were my best friend, being my prince, but I was wrong. Baby, you changed a thing into. mine, don't stress your mind. We coming home tonight. Hey, mom, we're gonna be alright. Dry those eyes. We'll be back in the morning when the sun starts to rise.
We are now continuing our meeting to the next agenda. That is to hear the keynote speech will be delivered by Dr. Siti Nurfazila Musain from University Technology Mara in Malaysia. The keynote speech will be moderated by Mrs. Rahma Labajo, SST, Master of Public Health. Ladies and gentlemen, please welcome Dr. Siti Nurfazila Musain and Mrs. Rahma Labajo.
Hello. Thank you, Hadi. Can you hear my voice clearly? Assalamualaikum warahmatullahi wabarakatuh, and good morning, everyone. Welcome to the second plenary lecture of the third International Conference in Pharmaceutical and Health Sciences. And I am Rahma Labajo, and I will be serving as your moderator today. And before we begin, I have a few housekeeping rules. So for today's session, it's being recorded to be available for viewing post-conference. And if you have any questions for the presenter to answer after this session, please use the chat feature in this Zoom webinar. And if you have any technical questions, please reach out directly to me via chat box. The presenter today will have a total of 60 minutes for a presentation, and if the presenter completes the presentation before 60 minutes is up, they may have the time for question for attendees. So, ladies and gentlemen, we'll be hearing a presentation from Dr. Siti Nurfazila Musain from University Technology Mara, Malaysia. Hello, Dr. Siti. And on this very timely subject, but before we get started, I want to take a few minutes of your time to briefly introduce the second keynote speaker of this conference. As you can see on the screen, Dr. Siti Nurfazila. She has auditioned in 2006 to get a silver medal to international exposition on Sharia compliant idea invention, innovation and design. And also at the same year, she get a silver medal to international exposition on Sharia compliant innovation, innovation and design with the project Jeruk Tanpa Cuka. And in 2015, she get raised gathering potential to KIK Peringkat Zone Utara with the project e-host one-stop communication center for hospital training leader. And also she has publication chapter in book, article journals, and the working experience right now, she is working for University Technology and Mara, Penang branch, as senior lecturer at Faculty of Pharmacy. So please welcome Dr. Siti Nurfazila Musain.
Assalamualaikum warahmatullahi wabarakatuh, and a very good morning, everyone. So can I start with the presentation? So should I
You
start
may
with the
start.
presentation?
Yeah.
Thank you. So can you hear me, everyone?
Yeah. It's clear.
Okay. Assalamualaikum and a very good morning, everyone. First of all, I would like to thank Health Polytechnic Gorontalo for this opportunity to present a little bit of our work in the third international conference in pharmaceutical and health sciences. And as the chairperson has introduced, my name is Siti Fazila binti Mohsin, and I'm currently a senior lecturer in University Technology Mara, actually in Pulau Pinang, north of Malaysia. And again, thank you for this opportunity to present a little bit of our work, and perhaps this is an invitation for a future collaboration with those who are interested in the same area. So the presentation for today is physiological relevance of bilirubin in diabetes mellitus. So as you can see on the screen, this is my presentation outline. I will start with a little bit on the overview of bilirubin. Perhaps most of you are quite aware or quite familiar with what bilirubin is all about. And later I will continue with its health implications and supplement my presentation with some findings from our studies, from our group, and finally conclude my presentation for today. So this will be a quite simple and hopefully a concise presentation of bilirubin and its relevance to diabetes, and hopefully that will be enough for you to appreciate bilirubin, which is an old product or molecule, but becoming an increasingly interesting with the emergence of new findings nowadays. So in my presentation, I will highlight the role of oxidative stress that is the cause of many pathological disease, and particularly diabetes. And it's important to understand defense mechanism in cells. So in this line, it's quite important for us to know what is bilirubin and its role as an antioxidant. And hopefully this presentation will provide another point of view from which to approach the study and also the treatment of diseases such as diabetes with pertaining to the use of endogenous antioxidant. So let's start by reviewing a little bit on bilirubin. So bilirubin is an endogenously produced molecule, meaning it's created within the body as part of normal physiological processes, as we all know. So bilirubin is a product of breakdown of heme, as we know, which is a component of hemoglobin, and hemoglobin is a protein responsible for transporting oxygen in red blood cells. So its breakdown is actually a natural occurrence, as red blood cells has a finite lifespan, which is 120 days. So bilirubin, as we know, is not a foreign substance or toxin, is a natural byproduct of a vital process in the body. And so this immediately makes bilirubin a physiological molecule, and its presence is expected in a healthy system. So this is in contrast with our previous knowledge of bilirubin. So we usually relate bilirubin with its toxic effect, which is associated with liver dysfunction, kernicterus, jaundice, or neurological disorders, and also other metabolic diseases such as hypertension, atherosclerosis, and of course diabetes. So this presentation will be a new aspect of bilirubin and a new aspect on an old molecule. Okay, so if I can bring your attention to this circle down here. So this bilirubin is, as we know, is formed primarily in the spleen and further processed in the liver. So this emphasizes its role as a waste product elimination, as I've mentioned just now, product of RBC breakdown, and so it has an importance in maintaining the overall body health. So the first step in bilirubin metabolism is the breakdown of heme by a very important enzyme, which is heme oxygenase. So heme oxygenase is actually induced in a stress condition, for example, in diabetes. So this further produces biliverdin with the release of byproducts such as carbon monoxide and also iron. So biliverdin is further reduced when it is in the presence of radical species. It can be converted to bilirubin with the aid of biliverdin reductase. So this bilirubin, conjugated bilirubin, is transported to liver through blood and taken up by the liver hepatocytes, undergoes conjugation and becomes water soluble molecule. So this system, the heme oxygenation system, is a very important cycle, which I will elaborate later on in the presentation. And this system is also play a crucial role in the protective effect of bilirubin. So it's very important in maintaining bilirubin homeostasis and preventing the build-up of bilirubin in the body. So that's why bilirubin has always been associated with harmful effect until recent research on bilirubin suggests an entirely new function of bilirubin as a metabolic hormone. And we look into the cytoprotective effect of bilirubin later in the presentation. So bilirubin has a multifaceted impact on health. It is crucial, as I've mentioned, it's crucial for waste product elimination, it's crucial for liver function, and it has a potential protective effect on neurological disorders such as Parkinson, in cardiovascular health, atherosclerosis, reducing inflammation, reducing oxidative stress, cancer, and particularly for our benefit for today theme is diabetes. So monitoring bilirubin levels and understanding its role in health and disease can be a great importance in clinical practice and medical research. So as we all know, elevated levels of bilirubin can be indicative of liver dysfunction or obstruction of the bile ducts. Okay, so monitoring bilirubin level is very important in diagnosing liver diseases such as hepatitis, cirrhosis, and so on. So in line with our theme for today, I would like to emphasize its link to diabetes. So studies have shown that individuals with higher bilirubin levels in their blood may have a reduced risk of developing diabetes. So when I say increased level, it means within the normal level, but a higher normal level. Okay, so perhaps around 10 micromolar, which is still within a safe levels of bilirubin, but a little bit induced. So it has been also shown that there is a negative association between bilirubin and diabetic occurrence and also complications. So bilirubin's protective effect is actually all due to its anti-inflammatory and also insulin sensitizing effect. And this overall effect is thought to contribute to the protective effect of bilirubin. So there are studies available that can supplement or that can support the hypothesis that bilirubin has a positive effect on the diabetic occurrence and also diabetic complications. And some of it as I've mentioned at the bottom of the slide there. So we will look into the protective effect of bilirubin and its linked to diabetes later in the presentation. So now, these properties of bilirubin in the context of diabetes mellitus highlight its potential as a natural protective factor because it is a natural and endogenous compound in our body. So bilirubin can help counteract oxidative stress, inflammation, and obesity-related factor. Surprisingly, all of which play critical roles in the development and progression of diabetes. So as I've mentioned here, the antioxidant, anti-inflammatory, potential anti-adipogenic properties. They hold a potential particular relevance in the context of diabetes mellitus. So if you know or if you haven't known, so in diabetes, there is an increased production of reactive oxygen species, ROS, and that would link to an increased oxidative stress, which we know that oxidative stress damage cells, including the beta cells in the pancreas. So bilirubin is known as a potent endogenous antioxidant, and this can help to neutralize these harmful free radicals. So this protection is significant in safeguarding the pancreatic beta cells and also maintaining insulin production. As with the anti-inflammatory properties of bilirubin, so in diabetes there is an association with chronic low-grade inflammation with diabetes. So inflammatory molecules can interfere with insulin signaling, and also this can lead to insulin resistance. So bilirubin anti-inflammatory effects can help actually to mitigate this inflammation and also enhance insulin sensitivity. So there are studies that support these findings. Okay. Initially, it was found that bilirubin inhibited the expression of pro-inflammatory cytokines and also induce the levels of anti-inflammatory cytokines. So in atherosclerosis, in cardiovascular disease, and later on it was found that the same association was also seen in patients with diabetes. So bilirubin can actually inhibit the expression of pro-inflammatory cytokines and also exhibit or inhibit the anti-inflammatory cytokines. So this will help in the overall anti-inflammatory effect of bilirubin. In diabetes, of course, obesity is a major risk of diabetes. Fat cells are involved in the release of cytokines and also adipokines, which are inflammatory mediators. and this will contribute to insulin resistance, as I've mentioned just now. So if bilirubin indeed possess anti-adipogenic properties, it could potentially inhibit the formation of new fat cells and also help prevent obesity-related insulin resistance. So there are studies that has been done that shows that bilirubin has actually an anti-adipogenic properties, so it can reduce fat cells. So this is due to the elevation of PPAR alpha, which is a fat-burning nuclear receptor. So this overall effect, the anti-inflammatory, the antioxidant effect, and also the anti-adipogenic effect of bilirubin, overall effect can help certainly in the perhaps in the therapeutic target for diabetes management. So for my presentation for today, I would like to specifically focus on the role of bilirubin in inhibition of oxidative stress, which in line with, and I would like to supplement this fact by some findings from our group and our findings. So now, antioxidant properties of bilirubin. Okay, so I would like to bring your focus to one aspect of bilirubin cytoprotective effect, that is antioxidant, which I didn't highlight in the previous slide. So bilirubin acts as a powerful endogenous antioxidant. So in this way, actually, bilirubin has a dual effect. If it's high, it will become toxic to our body, but when it is induced a little bit, but we still within a normal range, it is actually a very potent endogenous antioxidant. So again, it helps neutralize harmful free radicals, reducing oxidative stress in the body, scavenge free radicals, and prevent oxidative damage. So this antioxidant function is essential for protecting cells, tissues, and organs from damage caused by oxidative stress, which is the highlight of diabetes as well, diabetes complications as well, which is associated, it's not just with diabetes, but with various chronic diseases. So serum bilirubin may serve as an important physiological modulator of oxidative stress and chronic inflammation in metabolic syndrome and diabetes. So actually, if you know about glutathione and also vitamin E, which is a very potent antioxidant before, actually bilirubin has been shown to be more effective at protecting lipids from oxidation than glutathione, which is a principal antioxidant in our body, and it is almost 30 times more potent toward the prevention of cholesterol oxidation compared to vitamin E analog. So it's actually a very newly found very potent antioxidant. And if you see at the bottom of my slide there, you can have a look at those references to have a little bit more understanding of the protective effect of bilirubin as an antioxidant. Okay, so and of course in diabetes, I've mentioned the hallmark of which consists elevated plasma glucose, hyperglycemia. Elevated hyperglycemia is consistently associated with an increased oxidative stress, and because bilirubin is a very powerful antioxidant, so it scavenges free radicals. So you must be, because I've mentioned this fact about bilirubin being a very powerful antioxidant multiple times now. So hopefully that will also interest you of how actually bilirubin do this antioxidant, possess this antioxidant property. So how does it do that? So if I can bring your attention to this diagram here. So this diagram is actually very important in the protective effect of bilirubin. So bilirubin is a very powerful antioxidant, scavenge radicals. So if you see here, heme, a product of heme is a product. The breakdown of heme will produce biliverdin by the action of heme oxygenase. So biliverdin further will be converted to bilirubin through the action of biliverdin reductase. So the findings by our group is that bilirubin, being the potent antioxidant, will be oxidized by ROS, broken down to metabolites, to water-soluble metabolites in the urine, and interestingly, when the excess of bilirubin will be converted back into biliverdin, which is a water-soluble compound. So this cycle plays a very important role in the protective effect of bilirubin. So if there is, when the body sense there is a increased ROS, for example, increased glucose level, heme or heme oxygenase system will be induced, and this will produce biliverdin. Biliverdin will be converted into bilirubin by the action of biliverdin reductase. So bilirubin will scavenge ROS, and that will be, after it scavenge ROS, and that will be excreted in the urine as a dipolar di or tripyrose. And that can be this tripyrose compound can be actually be detected in the urine, and it has been shown to be increased in an oxidative stress condition such as diabetes. So interestingly, now our group found that bilirubin, after it has done its job, it can be reconverted back to biliverdin. Why? Because bilirubin is a fat soluble, which increased level can be toxic to the body. But when it is converted back to biliverdin, biliverdin can be present in more abundant because biliverdin is water soluble. So this conversion of bilirubin to biliverdin is done by a bilirubin oxidase enzyme, which we found to be cytochrome 2A5, cytochrome P452A5 or 2A6. So our group has found CYP2A5 or CYP2A6 as a bilirubin oxidase. Okay, so this cycle is actually quite similar to a very well-known antioxidant cycle, which is the glutathione recycling mechanism. So in this way, our results have shown that CYP2A5 is in part of the heme oxygenase system. So this is a new finding by our group. So bilirubin is reduced to biliverdin by biliverdin reductase. Biliverdin, so if the body sense an oxidative stress or an increased ROS, biliverdin will be immediately converted to bilirubin by biliverdin reductase. Biliverdin reductase is an enzyme that can quickly convert biliverdin to bilirubin, and after bilirubin has done its job, it will be excreted in the urine. Any excess of bilirubin available will be converted back to biliverdin by CYP2A5, and this biliverdin will be just standby in case of another increased levels of ROS, then it will be converted back to bilirubin. So I hope that schematic presentation will highlight the importance of bilirubin, or you will be more appreciative of the role of bilirubin as an antioxidant. So in this way, bilirubin can be recycled as an antioxidant many times over. Although its cellular concentration is very small, it's in micromolar, but its effect and it's actually the concentration is much lower than glutathione, but the effect is much more than glutathione. In fact, I've mentioned just now, more than 20 or 30 times more than glutathione. So again, a little bit more on the antioxidant properties of bilirubin. So in a healthy condition, if you see the diagram over there, in healthy condition, liver produce glucose that is released to the systemic circulation. And this glucose will be used by other tissues, particularly during fasting, actually. So hepatic glucose production derives from glycogenolysis, and glucogenesis will contribute to a healthy blood glucose level in the body. Now, over time, if there is an increase in glucose production, such as in diabetes, there will be a high glucose production. So over time, this can result in liver damage, and this can contribute, of course, perhaps you have known, to the development of, for example, liver dysfunction, non-alcoholic fatty liver disease, and also more severe liver conditions such as carcinoma, for example. So it is very essential for individuals with diabetes to manage their blood sugar levels and other risk factors to minimize the risk of liver complication. So fortunately, we have seen that mild elevated of bilirubin can actually protect against diabetes occurrence and complications. And how does it do that? I want to explain a little bit, of course, from our studies pertaining to this effect of bilirubin in the liver. Okay, so diabetes can lead to oxidative stress and apoptosis in the liver, okay, through the combination of factors. So as I've mentioned just now, hyperglycemia or increased glucose level can lead to liver dysfunction, for example, any non-alcoholic fatty liver disease. So how does it do that? How does it contribute to non-alcoholic fatty liver disease? There are a few mechanisms that can be associated with that dysfunction. So among others, perhaps mitochondrial dysfunction and also endoplasmic reticulum stress. So others could be inflammation and also insulin resistance. But for the purpose of this presentation, I would like to highlight on the mitochondrial dysfunction, particularly mitochondrial oxidative stress and also endoplasmic reticulum stress. Okay, I would like to explain a little bit further on the mitochondrial dysfunction and ER stress, and pertaining or by the supplement of our findings from our studies. Okay, so we found that the system, the heme oxygenase C2A5 system that I've mentioned just now, is present in the endoplasmic reticulum, but it is also found in mitochondria, which is a very interesting point, because you know what, in mitochondria, the level of cytochrome 2A5 or cytochrome, the endogenous cytochrome in the mitochondria is known to be, for example, associated with endogenous metabolism, in the metabolism of lipid, for example, cytochrome P450, 14, 11, 12, and so on. But cytochrome P450 1 to 4 is actually more associated with stress or with a condition, some conditions that can induce it. It's not, it doesn't present constitutively. But however, with this cytochrome P452A5, it is present constitutively in the mitochondria. So that is a very important or very interesting finding from our study. So, so we found that in mitochondria, during physiological condition, bilirubin will scavenge ROS, and this will form dipyrrole. So as we know, mitochondria is the main producer of reactive oxygen species in the body. So in physiological condition, in a healthy condition, in a healthy liver, in a healthy individual, bilirubin definitely will scavenge ROS, and this will form dipyrrole. So perhaps, and from our findings, C2A5, which is normally not common cytochrome P450 to be associated with normal metabolism in the body, is present in the mitochondria. So as mitochondria is the main site of ROS formation, and this is actually a part of its function. So however, during heightened oxidative stress, such as hyperglycemia, and this can lead to mitochondrial dysfunction, as I've mentioned. So the C2A5 will be transported to this organelle and help to remove the ROS. So when this system is overburdened, then apoptosis will follow. So what I want to stress is that in a healthy condition, in a normal physiology condition, ROS mitochondria mitochondria is the main producer of ROS, and is the main site of producing ROS and also perhaps oxidative stress. But in a condition of increased oxidative stress, such as in diabetes or hyperglycemia, this ROS in the mitochondria will be more produced, and this will lead to mitochondrial dysfunction. And C2A5, which is, as I've mentioned, a bilirubin oxidase, will be transported to mitochondria, and this will help to mitigate the antioxidant properties of mitochondria, antioxidant properties of bilirubin do its job. So as I've mentioned just now, it will, once it senses there is an increased ROS, cytochrome P452A5 will be transported to mitochondria and do its job. Biliverdin will be converted to biliverdin will be converted to bilirubin. Bilirubin will scavenge ROS. The excess bilirubin will be converted back to biliverdin, and this cycle will go around and round to make sure that the level of ROS will be maintained at a safe level until the oxidative stress level is too much. Then the apoptosis will follow. So I hope you can follow the presentation. So it's quite interesting because you know what, I think mungkin Allah dah buat mitochondria is the main function is to produce energy. Is a source of energy, but it's also the site of death. It's the source of energy, but it's also the site of death in ourselves. Hidup dan mati bermula di mitochondria. So it's quite interesting to learn that actually. So in addition to mitochondria, in diabetes patient or in diabetes condition, increased glucose level can also disrupt endoplasmic reticulum stress. So leading to a condition called endoplasmic reticulum stress, which interestingly can further burden the stress in mitochondria. So in a condition of hyperglycemia, mitochondria can be stressed, endoplasmic reticulum can be stressed. So mitochondria will be in the presence of bilirubin or other antioxidant will be complement to each other, and they will try to mitigate the reactive oxygen species in the mitochondria. And in endoplasmic reticulum, there will be concurrently stressed with mitochondria. But if the endoplasmic reticulum stress is too much, then that stress can also be migrated, can also be transferred into mitochondria. So mitochondria dah lah, if I can speak in bahasa Melayu, so mitochondria itself, it has the stress, and also it will be supplemented by the stress from the endoplasmic reticulum. So you can imagine the burden of mitochondria. So to know that it's quite interesting to know that the function of cytochrome P452A5 in response to oxidative stress. So the findings from our group that shows that C2A5 is actually a bilirubin oxidase has helped in further understanding the role of bilirubin in help in maintaining the physiological ROS, and also in help to in the defense mechanism of cells to reduce the burden of oxidative stress in the body. There are many studies that have shown the protective effect of bilirubin. These are some of the, as I've mentioned down there in my slide, these are some of the studies that have shown the protective effect of bilirubin pertaining especially to mitochondria. Not many studies are available in pertaining to the mitochondrial dysfunction, pertaining to oxidative stress especially, and bilirubin. So further development of the studies in this area would be very beneficial for us. Okay, so I think I would like to, I have come to the end of my presentation. So in conclusion, bilirubin is an old molecule, but accumulating evidence suggests that bilirubin may be important in the physiological modulator of oxidative stress and chronic inflammation in metabolic syndrome and especially in diabetes. So studies should be done more to further explore the potential of bilirubin as a new biomarker and also as a therapeutic target of the management of diabetes. So using bilirubin as a biomarker for diabetes risk assessment has many advantages. Bilirubin is actually already a part of a routine blood panels, and it's relatively low cost. It means that it doesn't need a specialized testing is required, and it is widely available. So perhaps by developing bilirubin perhaps as a biomarker for diabetes during routine checkups, healthcare professionals can identify individuals at a higher risk of diabetes. Of course, we are still in the research stage. Further research or synchronized research needs to be done to actually distinguish or determine the levels of this biomarker so that it can be actually can be developed as a biomarker later on for diabetes. But I seriously believe in the potential of bilirubin in the as a target for or as a biomarker for developing diabetes. So early identification can lead to lifestyle intervention, and of course preventive measures can reduce the risk of developing diabetes. And further studies should be done to clearly define how we should use bilirubin actually in estimating the disease levels and the purpose of the treatment planning. And that is one aspect of future development with diabetes as a biomarker. As a therapeutic strategist, of course, many studies are done to provide more information on the usage of bilirubin as a therapeutic marker using heme oxygenase inducers, administration of heme oxygenase inducers, using biliverdin supplementation, using bilirubin supplementation. All these studies have shown positive effect on the diabetes complications and also occurrence. So and also perhaps on the development of inducers and natural inducers for bilirubin, it needs to be done as well. All this would be beneficial in the potential role of bilirubin as a treatment or in the management of bilirubin. So I think that's all for now. And I would like to thank again the committee for inviting me, and I hope my presentation highlight or increase your interest in the in the use of bilirubin, in the studies of bilirubin, in the research of bilirubin as a potential treatment, as a potential therapeutic treatment or biomarker in the future for the benefit of diabetes, definitely. And thank you so much for this opportunity. Thank you.
Thank you, Dr. Siti. And we'll move on the question and answer session. So we have some students to deliver the questions. Yes, please, Miss Ratati.
Okay, thank you, moderator. What a very interesting explanation, doctor. It's take my interest because especially about how mitochondria regulate life and death in quotation marks, and regarding the effects of bilirubin, which is very contradictory, I think. Based on explanation, doctor, please correct me if I'm wrong, that bilirubin plays a role in reducing the risk of diabetes. Am I right, doctor? All right. My first question is, does the mean that we can increase a person's bilirubin levels to prevent diabetes? And doesn't it cause a new negative effect on that person? And the second is, what is the ingredients in natural ingredients which can be used to increase or optimize bilirubin levels as a new biomarker in someone with diabetes mellitus? I think it's my question, doctor. Thank you.
Yeah. Okay, thank you for the question. Yes, diabetes or bilirubin, you must be aware. Tapi mungkin saya bicara juga dalam bahasa Melayu. To increase levels of bilirubin, but not toxic level. So increase a little bit level of bilirubin can actually have an effect, positive effect on diabetes. So it can actually show the occurrence and also the complications later on. Okay, so because we have seen that hypobilirubinemia in patient yang memang ada rendahnya bilirubin, bila kita banding bersama dengan bila kita bandingkan dengan individu yang mempunyai bilirubin lebih tinggi, sedikit hypobilirubin mungkin maksudnya berkurangan daripada 5 mikromolar, mungkin sebegitu. Kerana dalam actually tiada low range tu tidak tiada definisi untuk bilirubin. So the normal range is actually from 3 to 20 and toxic level is actually more than 150 micromole macam tu. So but with a reduced level in patients yang memang ada hypobilirubinemia, mereka mendapati risiko untuk mendapat diabetes itu lebih tinggi. sangat tinggi. So bila kita dapat meningkatkan sedikit bilirubin tetapi bukan pada tahap yang toksik, hanya kita, pada tahap physiological tetapi lebih tinggi sedikit, that would be a very potent antioxidant because dalam badan kita akan dapat regulate the cycle very nicely. Tetapi jika bilirubin terlalu tinggi, then our body will be overburdened, that it cannot manage the level of increased bilirubin, and perhaps later on it can be responsible for example, neurological disease and so on. Okay, so in fact memang ada perancangan dalam citation, dalam references untuk meningkatkan sedikit paras bilirubin, especially pada pesakit-pesakit yang mempunyai hypobilirubinemia, supaya mereka dapat mengurangkan risiko diabetes dalam pesakit-pesakit ini. So ini menunjukkan peningkatan bilirubin yang sedikit tetapi tidak terlalu banyak. So in this case, seperti yang tadi yang ditanya, sama ada peningkatan ini adakah mungkin later on akan mendapat komplikasi, akan memberikan pula komplikasi lain. Tetapi ya, jika peningkatan sedikit tetapi yang masih mampu ditampung oleh badan within a normal physiological range, then insya-Allah itu adalah targetnya, supaya ia memberikan efek antioksidan itu tetapi tanpa memberikan toxic effect kepada badan. Itu satu. Harap dapat memberikan. So seperti yang dinyatakan tadi, apakah bahan-bahan yang mungkin boleh meningkatkan kandungan di dalam badan? Ini agak sukar sepertinya. Mungkin ada this will actually potential interest. We know what actually natural products that can increase. So, but from the literature, perhaps for example, curcumin can also have been shown to increase and also has some positive impact on the bilirubin. And perhaps Indonesia with its biodiversity will also have some benefit on the research of bilirubin in pertaining to the natural products that can increase the levels of bilirubin perhaps later on. Perhaps that would be a very nice collaboration as well. So but a very good question nonetheless.
Okay, thank you, Doctor. It answered my question. Thanks.
Any other questions from the participants? Then raise your hand. For your information, Dr. Siti, the participants mostly the students from Pharmacy department.
Thank you for the information. So perhaps kalau tak ada soalan, mungkin saya ingin menyatakan sedikit tentang mungkin pengalaman saya lah menjalani PhD seperti sebelum ini, dan mungkin kemungkinannya para pelajar juga mungkin akan menjalani ataupun di masa hadapan ingin meneruskan pengajian ke bidang master dan juga sarjana dan juga doktor falsafah. So sebelum saya berjumpa dengan bilirubin ini adalah actually salah satu kajian saya menjalani kajian doktor falsafah di Queensland, Australia, dan it's interesting because sebelum saya bermula menjalankan kajian PhD, saya macam seperti yang ditanyakan tadi, pelik kerana bilirubin ini sepertinya Allah telah ciptakan sebelum ini kita hanya tahu tentang toxic effect-nya saja, penyakit kuning dalam baby. Itu sahaja. Tetapi saya percaya dalam hati saya, Allah tidak menciptakan anything tanpa sia-sia. So that is, even though without the all the citation, all the references, all these findings, kita dalam hati kita tahu tiada yang satu yang diciptakan sia-sia oleh Allah. So that is the motivation for me to understand what is actually bilirubin is all about. So because pada mula menjalankan kajian PhD, kita tidak tahu sama ada apakah saya akan mempunyai maklumat untuk disampaikan sebagai sebahagian besar daripada kajian saya sebagai PhD. Jika tiada maklumat dan saya tidak dapat bergraduat kan. Tapi percaya Allah tidak menjadikan sesuatu itu dengan sia-sia. So mungkin secara motivasinya mungkin pelajar-pelajar juga di hadapan ke depan nanti akan berusaha untuk menjalani kajian ataupun meneruskan pengajian dan ilmu Allah itu besar, banyak lagi yang kita tidak dapat ketahui. Bilirubin for example, ada banyak lagi yang kajian-kajian yang dilakukan especially untuk pada hari ini berkaitan dengan diabetes, which is a very important global burdens, mendatangkan banyak masalah kepada kita, especially kita di rantau Asia ini. Makanan pun makanan dengan banyak faktor yang mungkin mendorong kita untuk mendapat diabetes genetik and so on. So saya harapkan ini sedikit sebanyak dapat menimbulkan minat kepada para pelajar dan juga semua untuk berkolaborasi insyaAllah in the future dan juga mendalami lagi ilmu-ilmu tentang diabetes and also kompang-kompang yang mungkin membantulah dalam pesakit-pesakit menjalani rawatan insyaAllah. Itu sahaja. Terima kasih.
Terima kasih Dr. Siti. We have some questions from students. So Dr. Siti, maybe you can describe to us the work in your department in University of Marak.
Kurang jelas Ibu, mungkin boleh can you
Oh yeah.
minta maaf. Can you explain once again? Can you come again?
So can you tell us about your work in your department, mostly about diabetes or other non-communicable diseases, something like that? The research on your department, what is the most topic on the research on your department?
What is the interest of my department in the research? You mean that?
Yeah.
Okay. So I am attached to the Faculty of Pharmacy, University of Jimara in Pulau Pinang. So in my faculty, they have about 20 academic staff, something like that. So each one of us has our own interests, and perhaps like mine, I am interested in bilirubin, and also this is what I'm presenting today is about bilirubin pertaining to its antioxidant effect. So apart from its antioxidant effect, there is another properties of bilirubin which is as a psychological marker as well. So you can see the many function of bilirubin. So among other of my colleagues, there are also some of them are quite interested in the psychological or social studies, for example, pertaining to the patient. For example, the knowledge of self-prescription of diabetes, the self-management prescription of diabetes especially in the northern area, as a natural product as well, and also some pharmacotechnology, and also some education and also studies pertaining to education in pharmacy, the motivation and also what else, and also pharmaceutics, drug delivery and so on. So these are some of the general research interests of this academic staff. But if you, I would like to welcome any collaboration and from Gorontalo. Perhaps we can discuss more on this definitely, inshallah.
Thank you, Dr. Siti. I think my colleagues from the pharmacy department really interested in your research and also in your department. So we hope that we can have opportunity to study in your department. So we have some students to deliver their question. We have 4 students. So please, are you ready?
Assalamualaikum warahmatullahi wabarakatuh. Thank you for the time. Let me introduce myself. My name is Herman Kasim. My question is, what will happen when the bilirubin level is below normal? What will happen when the bilirubin level is below normal?
So, thank you for the question. So the question is, what will happen if the bilirubin level is
Yes.
above normal?
Yes.
Below.
Below normal.
Yes.
So in human, low bilirubin level is quite actually not that common. Although there are a condition known as, although there are conditions of hypobilirubinemia, as I've mentioned just now, and that is also associated, as I've mentioned, with increased, for example, increased incidence of metabolic diseases such as diabetes. In human, there is a condition, a genetic condition, we call it Crigler-Najjar syndrome. So that's in that patient with Crigler-Najjar syndrome, there is a decreased production of bilirubin because there is a decreased gluconation level of bilirubin, which is the metabolism. So it's not associated with any so far. If so far from the findings, patients with Crigler-Najjar syndrome has been some protective effect actually with bilirubin. Meaning, if we supplement bilirubin to this patient, we have shown that there is a protective effect of bilirubin. So in a condition of hypobilirubinemia, it's not as common. Usually it's due to some genetic effect actually, genetic conditions or congenital conditions. But there are conditions, we call it as hypobilirubinemia.
Okay, thank you.
Yeah, for the next question.
Okay, Assalamualaikum warahmatullahi wabarakatuh. Thank you for the time, and let me introduce myself. My name is Amanda Muzita Ramadhani Gobel. You can call me Amanda, and I have a question about how is bilirubin the result of the breakdown of hemoglobin related to diabetes? Thank you.
So thank you, Amanda. So the question is, how is bilirubin related to diabetes? Can you tell me the question one more time?
Yes, yeah. Hold on. Okay. How is bilirubin the result of the breakdown of hemoglobin related to diabetes?
Okay, so bilirubin, in my presentation, I have explained that, okay, bilirubin is a very potent antioxidant, endogenous antioxidant, which is actually an antioxidant that happens naturally within the body. Kita memang menghasilkan bilirubin di dalam badan. Okay, jika kandungannya terlalu tinggi, it can cause neurological damage, jaundice, and so on. So, bilirubin pada paras yang normal, pada paras fisiologikal, dapat memberikan kesan yang sangat positif kepada badan. Contohnya sebagai boleh mengurangkan keradangan, boleh mengurangkan oxidative stress, radical species dan sebagainya. So, di dalam penyakit diabetes mellitus, salah satu penyebab penyakit diabetes mellitus adalah disebabkan oleh kandungan paras gula yang sangat tinggi. Okey, bila kandungan paras gula tinggi, ini akan menyebabkan oxidative stress ataupun radikal-radikal bebas di dalam badan menjadi lebih banyak. So, inilah fungsi bilirubin di mana ia akan dapat membantu untuk mengurangkan paras radikal bebas di dalam badan. Kerana ia merupakan, seperti yang saya maklumkan tadi, bilirubin merupakan satu molekul yang mempunyai sifat anti-inflamasi, anti-keradangan, satu, sifat antioksidan yang sangat tinggi. So, inflammation is also the hallmark. Inflammation ataupun keradangan adalah salah satu ciri di dalam diabetes juga. So, bilirubin dapat mengurangkan keradangan, dapat mengurangkan apoptosis, iaitu sel death, kematian sel, dan ia juga dapat mengurangkan radikal-radikal bebas yang di dalam badan yang terhasil apabila diabetes terjadi di dalam badan. Adakah itu menjawab persoalannya antara hubung kait bilirubin dan juga diabetes?
Okay, thank you.
Please the next one.
Assalamualaikum warahmatullahi wabarakatuh. Thank you for the time. Let me introduce my name is Tasya Buloto, and you can call me Tasya, and I have a question about what is the mechanism of inhibition of oxidative stress on bilirubin? Thank you.
Okay, so what is the mechanism? How does bilirubin do its job? What is the mechanism of oxidative antioxidant of bilirubin? So, seperti yang saya maklumkan tadi, bilirubin terhasil daripada breakdown of heme. Okay, so heme akan di breakdown, akan di broken down into, heme itu adalah daripada sel darah merah. So akan di breakdown oleh enzim heme oxygenase kepada biliverdin. So biliverdin akan dapat ditukarkan kepada biliverdin melalui enzim biliverdin reductase. So heme oxygenase yang saya maksudkan tadi akan kadar konsentrasi heme oxygenase di dalam badan akan meningkat apabila badan mengetahui ada peningkatan oxidative stress di dalam badan. Okay, so bila ada kadar oxidative stress yang tinggi di dalam badan, heme oxygenase will be induced. So, induced dan akan menukarkan heme, iaitu kandungan daripada sel darah merah, kepada biliverdin. Biliverdin akan ditukarkan kepada bilirubin. Nah, bilirubin inilah yang akan memakan radikal-radikal bebas yang terhasil daripada oxidative stress. Oxidative stress ini terhasil daripada peningkatan, salah satunya disebabkan oleh peningkatan gula di dalam badan. So, bilirubin akan memakan radikal-radikal bebas yang terhasil akibat daripada peningkatan gula di dalam badan. So, apabila bilirubin ini telah memakan radikal-radikal bebas, ia akan di-excrete, akan dibuang daripada badan. Tetapi bilirubin yang tidak memakan radikal-radikal bebas itu akan ditukarkan semula kepada biliverdin. So biliverdin ini akan sentiasa dalam keadaan berjaga-jaga. Bila di mana akan ada lagi terjadi oxidative stress, radikal bebas terhasil, dia akan terus ditukarkan kepada bilirubin. Bilirubin akan memakan radikal bebas dan dia excrete daripada badan. Dan seperti itulah cycle dia akan berterusan. Dan jika radikal bebas ataupun oxidative stress dalam badan ada terlalu tinggi, ini sistem antioksidan di dalam badan tidak dapat menampung kandungan oxidative stress yang sangat tinggi, dan ini akan menyebabkan sel di dalam badan menjalani kematian, apoptosis. So ini apabila sistem defense dalam badan tu sudah terlalu lelah, tidak dapat memakan radikal-radikal bebas dan ia akan mati. Seperti itulah. So cycle yang saya sebutkan tadi, heme kepada bilirubin, itu adalah cycle yang sangat penting di dalam effect of bilirubin sebagai antioksidan. Faham tak?
Okay, thank you for the opportunity given to me. My name is Nabila Musliha Nasrun. So the question is, what if a baby with high bilirubin at birth is at risk of diabetes? Thank you.
Say that again. A baby with?
The question is, what if a baby with high bilirubin at birth is at risk of diabetes?
Okay, so diabetes is actually associated with, usually the main positive factor is lifestyle, right? Diet, the environment factors and so on. So in babies, in infant, usually they are more prone to get high bilirubin levels. This is because the mechanism that I've mentioned just now, the metabolism of bilirubin, is not yet fully developed in them. So the risk of increased bilirubin in babies will cause more on neurological effect. For example, jaundice, and if it's too much, it can damage the brain, cancerous. So in order for babies to develop diabetes, is a long shot, because most babies, diabetes is more of a environmental ataupun genetics. The causative effect is that genetics and environmental. But of course, in babies there are conditions yang kita katakan diabetes, and that is more on the macam perhaps type 1 diabetes, and also due to genetic. That is far-fetched from bilirubin lah, yang tu salah satu aspek yang perlu dilihat. Mungkin it triggers a question or an interest as well to see bilirubin in babies yang predisposed to diabetes, for example. Quite interesting. But bilirubin in babies, high bilirubin levels in babies are making them at risk of developing neurological disorder lah, for example, jaundice, brain damage, and so on, because the metabolism ataupun the metabolism pathway of bilirubin is not yet fully developed in them.
Okay, thanks for the opportunity given to me. So my name is Citra Maduyo, and my question is, why is the exogenous antioxidant bilirubin stronger than glutathione and vitamins? Thank you.
Okay, to answer why, I do not know actually how to approach that question. That's how the findings from the results actually. It's just that glutathione are present in the molar concentration, and we found that bilirubin can also do the job, but in micromolar concentration. So you can see that the difference there. So glutathione to do the same job as bilirubin, the concentration needs to be higher as compared to bilirubin, which is present in micro concentration. So in that sense, that's why I mentioned that bilirubin is actually 20 to 30 times more potent compared to glutathione, which is a principal antioxidant in the body. But you have to be cautious as well because bilirubin are more potent towards ROS that are different from the type of ROS that are produced that are attacked by glutathione, because glutathione is water soluble, bilirubin is fat soluble. So these two are present in different concentration. Bilirubin are present in micromole, which is much lower concentration compared to glutathione, but are doing the same job as glutathione. And number two, glutathione is water soluble and bilirubin is fat soluble. So this system complements each other. So Allah buat begitu kan, dia satu water soluble, satu fat soluble, but these two will work together to reduce the effect of radical bebas di dalam badan.
Okay, thank you, ma'am.
So that's all the questions from the students. So before we end the session, the committee would like to present the certificate of appreciation to Dr. Siti. The certificate will appear in your screen.
Thank you.
Yeah, so this the certificate is present to Dr. Siti Nur Fadila Muhtaye as the speaker at the third international conference in pharmaceutical and health sciences. So on behalf of the committee and also the Department of Pharmacy, Health Polytechnic of the Ministry of Health Gorontalo, we thank you for the insightful research and talk from Dr. Siti. Terima kasih, Dr. Siti.
Ya, sama-sama. Terima kasih atas jemputannya juga.
Okay, thank you very much to our keynote, Dr. Siti Nur Fazila Musain, and our moderator, Mrs. Rahmala Bajo. Next session for attendees, please break for pray and lunch until 1:30 p.m. Thank you.
was delicious.
Ladies and gentlemen, we are now continuing our meeting to the next agenda. That is to hear the keynote speech will be delivered by Professor Ikatan Adiana, MSI, PhD from Institut Teknologi Bandung and will be moderated by Rahmadwi Agustini, SST MCAP. Ladies and gentlemen, please welcome Professor Ikatan Adiana, MSI, PhD, and Rahmadwi Agustini, SST MCAP.
Okay, thank you so much, Mr. MC. Okay, Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam.
Good afternoon, ladies and gentlemen. Welcome to the third International Conferences in Pharmaceutical and Health Science 2023 by Pharmacy Department of Poltekkes, Kemenkes, Gorontalo with the theme Current Challenges and Innovation in the Field of Diabetes Mellitus. It is sure that I, Rahmadewi Agustini, will be the moderator for this session. Okay, the topic for today's session is update of diabetes mellitus. Diabetes mellitus is a chronic disease that affects millions of people worldwide. It is characterized by high blood sugar level that can lead to serious health complications if left untreated. Then we will discuss the latest updates on diabetes mellitus, including new treatments, prevention strategies, and also management technique. And for that, we have a speaker who is already with us. We have Professor Ikatan Adiana, MSI, PhD from Institut Teknologi Bandung. Okay, before Professor Ketut deliver his material, I'm going to read his profile. Okay, so Professor Ketut MSI PhD is from Institut Teknologi Bandung, Indonesia, and Professor Adiana has earned a PhD in Toyama Medical University, Japan, in 2001. He also holds a master degree from Institut Teknologi Bandung in 1996. And also, in terms of academic work experience, Adiana has working as a lab and also head of Material Toxicity and Safety Laboratory School of Farmasi, Institut Teknologi Bandung from 2007, and also from 2020 he is a member of Academic Senate of Institut Teknologi Bandung and also as a Dean of School of Farmasi. Okay, in addition to his academic and professional work experience, Professor Adiana has also published several books related to pharmacy and also traditional medicine. Okay, that was an exceptionally remarkable curriculum vitae of Professor Ketut Adiana. So now, without further ado, I would like to invite Professor Ikatan Adiana to present his talk on update of diabetes mellitus. Professor Ketut, the screen is yours.
Thank you very much, MC, moderator, Ms. Rahmadewi. My voice is clear enough? Ladies and
Yes,
gentlemen?
very clear, Prof.
Thank you very much. May I share my slide? Can you see my slide already? Hello?
Yes.
Already, yes. Thank you very much. Okay, again, thank you very much, moderator, for the opportunity and chance to me to share the topic about update of diabetes mellitus. Okay, as you already introduced about me, first of all, I want to say first, Assalamualaikum warahmatullahi wabarakatuh. Selamat siang, salam sehat dan sejahtera untuk kita semua, and good afternoon, everybody. That's already attends the third international conference in pharmaceutical and health science conducted by Poltekkes Gorontalo. Okay, as already introduced by the moderator, so now let me introduce a little bit about myself in other side. Yes, my name is Ikatan. Yes, of course, I was born in Bali. Now I'm working at School of Pharmacy, ITB, Bandung. Yes, about the Cinta score, the reasons is, yes, you can see from my slide. Its index Scopus around 26, and then Web of Science index 16, and its index Jujol is 30. So now I'm a Dean of School of Pharmacy ITB. My research interest in pharmacology of natural product. Of course, my research interest to discover the medicine from the product, specifically for treatment of metabolic syndrome such as obesity, diabetes mellitus, hypertension, dyslipidemic agent, arthritis, osteoporosis, aging, cancer, dementia, as well as stroke also. As you can contact me at ketut@itb.ic.id. Okay, before I start my topic, Mr. Rahmadewi, how long my time to present my topic?
Okay, Professor Ketut, you have for about 2 hours, included question and answer session.
Okay, thank you very much.
Yeah.
I think long enough. Okay, I can start slowly. Okay, so the presentation outline for my topic is we start from definition of diabetes mellitus, and then prevalence and classification, and also risk factor of diabetes mellitus, followed by symptom and complication, target therapy, new therapy, maybe also alternative therapy from natural product that we reach with that natural product resource. Yes, as you know, and then close by summary. Okay, we start. As all of you, I think, already understand and know about the diabetes mellitus. So, what is diabetes mellitus? So, I took two definition about diabetes mellitus. One from WHO. Yes, diabetes mellitus is a chronic metabolic disease characterized by elevated level of blood glucose. Yes, I think the key word here is excessive level of blood glucose, which leads over time to serious damage to heart, blood vessel, eyes, kidney, and also nerve. That means, yes, the logic consequence of diabetes mellitus, after a long time we suffer from diabetes mellitus, of course, can damage the vital organs. Yes, in WHO definition, that means stress from the elevated level of blood glucose, and also the complication of diabetes mellitus. So according to the Bureau, yes, one reference book that many students refer to this book. Yes, diabetes mellitus is a group of metabolic disorder of fat, carbohydrate, and protein metabolism result from defect in insulin secretion, insulin action, or sensitivity, or both. So from this definition, we can took at least two keywords. The first one, of course, about the diabetes mellitus is a metabolic disorder or metabolic syndrome. And the second keyword is the etiology of diabetes mellitus caused by insulin secretion, lack of insulin secretion, or insulin actions decrease, or sensitivity of insulin hormone is decreased, or both of them cause diabetes mellitus. So we can compare the definition of diabetes mellitus between WHO definition and also DVRO definition. I think the main substance of the definition is almost same. Okay. Okay. So how about the prevalence of diabetes mellitus? So as you already see in the slide, okay, I think the main conclusion is time by time the diabetes mellitus prevalence increase all over the world. So let me see at okay here. I think at North America and Caribbean, yes. I think there are around 46 million at 2017 prevalence of diabetes mellitus increased by 35%. So that means at 2045 become 62 million. So about in Europe also increased by 16%. At data from 2017, yes, around 58 million people become 67 million at 2043. So in Middle East and North Africa also increase, yes, amazingly around 110%. The data from 2017 around 39 million become 82 million at 2045. And then how about in Indonesia? So Indonesia, there is in the group of Western Pacific, I think here. Look at the color of the islands. Yes, increase also by 15%. Yes, the data from 2017 around 156 million become 183 million at 2045. Yes, also another country and another ocean also increase. That means all over the world, the prevalence of diabetes mellitus increase. So that means I think diabetes mellitus all become a problem all the country in the world, I think. And then from the resource of WHO, also Roland Berger, the prevalence of degenerative disease, yes, generally all kind of degenerative disease increase from 2019 to become 2045. Yes, how about diabetes mellitus? Look at this one. At 2019, the prevalence of diabetes mellitus around 3%. So at 2045, increased to become 4%. Yes, another disease also. Yes, I think the amazing increasing is Alzheimer and other dementias. Start from 3%, become 6%. Yes, 2 times increase. So that means we will face many problems about degenerative disease, including diabetes mellitus. Again, the specific data about diabetes mellitus in Indonesia. So, according to International Diabetes Federation at 2021, so the number of diabetes population in Indonesia, the classment number 5. Yes, around 19.47% a million, I think, the population suffer from diabetes mellitus. Yes, the number 1, of course, China, number 2, India, number 3, Pakistan, and number 4, United States of America, and then number 5, and Indonesia. So according to data from Riset Kesehatan Dasar, yes, at 2007, 2013, and 2018, you can see the distribution of diabetes mellitus population in, yes, all islands Indonesia, all province Indonesia. For example, here Jakarta, yes, at 2007 around 1.8 million to become 2.5 million at 2013 and 3.4 million at 2018. Yes, you can see here the data, all the province in Indonesia according to data from Riset Kesehatan Dasar at 2007 until 2018. So now we come to the classification of diabetes mellitus. According to the new reference, we have 4 classification of diabetes mellitus. Number 1 is type 1 diabetes mellitus. The abbreviations commonly DM T1. And the second one, type 2 diabetes mellitus. And number 3, or the specific type of diabetes mellitus. And number 4 is gestational diabetes mellitus or GDM. So here also you can see the etiology of the diabetes mellitus. So type 1 commonly, I think, mostly genetic factor, because beta cell destruction usually leading to absolute insulin deficiency. That means almost 90% the patients suffer from diabetes mellitus type 1, a lack of insulin secretion. So the main cause of diabetes mellitus type 1, yes, until now we don't know yet. That means idiopathic etiology. So how about type 2 diabetes mellitus? Yes, here we can see etiology is insulin resistant with relative insulin deficiency to a predominantly insulin secretory defect with insulin resistant. Yes, there are 2 characteristics of a type 2 diabetes mellitus. Maybe in a group of patients suffer from diabetes mellitus type 2, lack of insulin secretion. In another group, yes, insulin hormone little bit higher, but yes, again, but the sensitivity of hormone insulin is very, very decreased. Yes, commonly this etiology become a common etiology in type 2 diabetes mellitus. Yes, I think the main causes of this disease is obesity. As you know already, obesity also increased the prevalence in Indonesia and all over the world. Yes, and then from all classification of diabetes mellitus, 90% the population suffer from type 2 diabetes mellitus. Yes, and the classification number 3 is other specific type diabetes mellitus. There are many factors. One of them is genetic defect of beta cell functions. Another factor is a genetic defect in insulin action, or disease of the exocrine pancreas, or endocrinopathies. Maybe another etiology is drug or chemical induced. So we can see here there are several drugs that can induce diabetes mellitus. For example, here the common one is glucocorticoid. For example, the population suffer from asthma that treated with glucocorticoid for a long time. That means chronic asthma that treated by glucocorticoid, they can suffer from diabetes mellitus type 3. That means induced by drug. Yes, for example, here glucocorticoid. So that means we have to care and consider also when the patient possess a chronic asthma that treated with glucocorticoid. So we also consider about the increase of blood glucose level that for a long time can become diabetes mellitus. Another etiology for type 3 is infection, or also uncommon from immune mediated disease. Yes. So number 4 type of diabetes mellitus is gestational diabetes mellitus or GDM. This type of diabetes mellitus is suffer from the woman during a pregnant. Yes. The next one. Yes, of course, there are many or several risk factors for diabetes mellitus. So there are two kinds of risk factor. Number one is factor that we can fix. Yes, we can fix. That means we can improve or we can modify. Yes, another terminology, modified risk factor. For example, height and weight. That means body weight, physical activity, blood pressure, and also the cholesterol profile. Yes, we can change, we can modify this risk factor. Yes. So another risk factor is we cannot modify it anymore. That means unmodified risk factor. For example, here about the age. Yes, all the people, the age become older and older, of course. So that means age also the risk factor that we cannot modify. Another one is genetic. That means close relative with diabetics person. Yes. Another one is other medical condition. For example, a chronic asthma that treated for a long time from glucocorticoid, or gestational diabetes for pregnant women, and also high risk ethnic background. Yes. For example, in Indonesia, the Java ethnic people possess higher risk factor compared to another ethnic in Indonesia, for example, Padang or Sunda or Bali, for example. Yes, there are a risk factor for diabetes mellitus, unmodified risk factor, and also a modified risk factor. Okay, here I can show you a simple method for the screening for diabetes mellitus type 2, whether we have a risk for diabetes mellitus or not. I think this is a very, very beneficial for us to screen whether we has a risk factor for diabetes mellitus or not. Yes, I think there are 7 questions that we have to answer, and then from the answer, we can know whether we have a risk factor for diabetes mellitus or not. For example, I can use myself as an example. For example, how old are you? For me, my age now 55. That's I got 2 points. Yes, look at this one. My age around 55 years old. So I got 2 points. Okay. The question number 2, are you a man or woman? Yes, I'm a man. I got 1 point. 2 plus 1 point to become 3 already. 3 score. Okay. The question number 3, if you are a woman, okay, I can omit this question because I'm man. So I'm don't need to answer this question. Okay, we go to the question number 4. Yes. Do you have a mother, father, sister, or brother with diabetes mellitus? So my answer is yes. That means I got 1 point more. So that means already 4 score. So we go to the question number 5. Have you ever been diagnosed with high blood pressure? No. So I got 0 point here. And then number 6, are you physically active? Yes, because I'm already doing physical activity or sport 4 times per week. Yes, 4 times. That means I conclude myself is physically active. So that means I got 0 point. So we go to the last questions. Number 7, what is your weight category? You can see from this chart. Yes, my height, I'm already counted. Yesterday, my age around 5 feet, 5 lube. Yes, look at the weight. So my weight around 130. So that means my point is 0. So totally from the 7 questions, I got 4 score. So look at this one. If you score 5 or higher, that means we are have increased risk factor for diabetes mellitus. So that means I'm not yet. That means lower risk factor. So all of you can also answer the 7 question in this slide and then conclude by yourself whether you have a high risk for diabetes mellitus or not. Yo. Okay, we go to the next slide. So what's the main symptom of diabetes mellitus? Yes. So the blue color is commonly for type 1 diabetes mellitus, but the prevalence is, yes, almost rare, less than 10%, because the common prevalence of diabetes mellitus is type 2. Look at the black color. Yes. Okay. I think here you can see the right slide or on the red slide. Okay, look at the right, the left slide. Start from the organ central nervous system. Yes, polydipsia. That means you suffer from the thirsty. Polyphagia, that means from hunger. Also in the eyes organs, you have damage from your eyes, such as blurry vision, and then urinary system, polyuria, yes, excessive urination, and glycosuria. That means your urine contain glucose compound. Okay, if you look at the right slide, yes, I think a simple illustration, yes, at least there are around 8 symptoms for diabetes mellitus type 2. Yes, urinating often, that means polyuria. Always hungry, that means polyphagia. Extreme fatigue, yes, even though you don't do a physical activity, but easier to get a fatigue. Yes. And then the number 4 is blurry vision. Okay. And then tingling or numbness in your hand or your feet. Okay. Weight loss without any decrease of body weight program. Yes. Increased density, that means polydipsia, and also slow healing the wound. Yes, because there is a problem with your blood and also blood vessel and also your immune system. That's the main symptom of diabetes mellitus, especially type 2. And for type 1, there are lethargy, stupor, weight loss, osmolar breathing, or it means hyperventilation, smell of acetone because the metabolism of glucose and also fat is not normal. Okay, produce any kind of body ketone. That's why our smell is acetone smell. And also from the gastric organ, we suffer from nausea, vomiting, and also abdominal pain. Okay, if we don't treat the diabetes mellitus properly, so maybe we suffer from the complication of diabetes mellitus. Yes, for example, diabetic foot. Yes, for example, gangrene, diabetic ulcer. Okay, diabetic neuropathy, you loss of your sensation in our lower extreme, for example, in our feet. Peripheral arterial disease, yes, there is a microvascular complication, yes, and then we suffer from cataract because long time for blurred vision, and then, yes, for a simple thing, we getting diabetic retinopathy, diabetic glaucoma, and also diabetic nephropathy or chronic kidney disease, and also complication of macro and microvascular disease such as ischemic heart disease as well as stroke. Yes, I think that's the main problem of diabetes mellitus, such as a chronic complication and also acute complication. So how about the the guideline for treatment of diabetes mellitus type 2. Yes. So according to this guideline, we can see we start from non-pharmacologic treatment. Yes, it means healthy lifestyle behavior, diabetes self-management education and sport, the SMAS, and also social determination of health, SDOH. Yes. Okay, the goal of this treatment is cardiorenal risk reduction in the person high with risk patient with type 2 diabetes mellitus. Yes, we have to comprehensively the manage of cardiovascular risk. Yes. So for another goal is achievement and maintain of glycemic and weight management goal. So I think the guideline divided become two. Yes, for example, the person with high risk factor for atherosclerotic cardiovascular disease, yes, heart failure or chronic kidney disease, for example. There are specific goals for this one, and also the choice for the drug that we can use for treatment of diabetes mellitus. Okay, you can read from this guideline. And another goal is, for example, we want to maintain the blood glucose level. That means glycemic management. Yes, we can choose another drug of choice, or achievement and maintain of weight management goal. For example, diabetic type 2 person, that means also obesity person. Of course, the main goal is to achieve and maintain the weight management goal. Yes. Look at this one. There are variation of drug of choice we can choose for treatment for specific goal that we can to get. Okay, start from the first step of managed for diabetes mellitus type 2. We start from non-pharmacologic treatment. Yes, we can use nutrition approach. Yes, for example, here we can see effectiveness of nutrition therapy. Yes, so an individualized medical nutrition therapy program is needed to achieve treatment goal. Yes, look at the evidence rating. I think A is already a good evidence for nutrition approach for diabetes mellitus. Yes, so this treatment recommended for all people with type 2, type 1, pre-diabetes, and also GDM, gestational diabetes mellitus. Yes. And then energy balance for all patients with overweight and obesity. Yes, you can use method energy balance if the diabetic person is obesity one. Yes, lifestyle modification to achieve and maintain minimum weight loss of 5%, for example, here. Also, another approachment is eating pattern and macronutrient distribution. Yes, but the evidence rating is still mild, for example, here. And also, how about the topic about carbohydrate? So, carbohydrate intake should emphasize nutrition-based carbohydrate sources that are high in fiber. Yes, minimally processed. That means a fresh fiber, for example, vegetable you can choose. Yes, or fruit, whole grain, also diet product, like vegetable, for example, here. And also reducing overall carbohydrate intake for individual with diabetes. Yes, and so on. You can see in this slide how to modify our carbohydrate intake for diabetes mellitus person. Again, what about protein, for example, here? So for individual with type 2 diabetes mellitus, ingested protein appear to increase insulin response. I think it is the good one without increasing plasma glucose concentration. So one thing we have to consider, carbohydrate source high in protein should be avoided when trying to treat or prevent hypoglycemic condition. Yes, you must consider about this if you want to control your protein intake. Okay, how about dietary fat? Yes, an eating plan emphasizing element of Mediterranean style eating pattern rich in monounsaturated and polyunsaturated fat might be considered to improve glucose metabolism and lower cardiovascular disease risk. Yes, also another method we can choose here from dietary fat. How about alcohol? Yes, adults with diabetes who drink alcohol should do so in moderation, no more than 1 drink per day. Okay, because we know alcohol also can increase our blood glucose level. Yes, another consideration we have to care about, education people with diabetes about the signs, symptoms, and self-management of delayed hypoglycemic after drinking alcohol. Okay, the next one, how about sodium intake? Yes, as for the general population, people with diabetes and pre-diabetes should limit sodium consumption. Yes, less than 2,300 milligram per day. Yes, because can increase our blood pressure and also increase the blood glucose level. Yes. So that means we have to care about the sodium intake, not just about a carbohydrate, but also sodium intake. Yes. Another one is non-nutritive sweetener. The use of non-nutritive sweetener might have the potential to reduce overall calorie and carbohydrate intake if substitute for calorie sugar sweetener and without compensation by intake of additional calorie from other food source. That means if the person still want to eat, to drink the sweet one, yes, we have to change the sugar to become non-nutritive sweetener because can control our blood glucose level. Okay. So another non-pharmacologic treatment is about regular physical activity. Yes, you can see here, by doing regular physical activity such as regular stepping per day. Yes, for example, you can see here stepping, for example, only 500 steps per day. That's mean associated with 2 to 9% decrease risk of cardiovascular morbidity and also all cause of mortality. Yes, another physical activity is breaking up prolonged sitting. Yes, for example, here with short regular bout of slow walking or simple resistance exercise can improve glucose metabolism. Another physical activity is physical exercise or sufficient quantity and quality of sleep. For example, here, yes, sweating, density moderate to vigorous activity. For example, look at this one. Yes, the duration around more than 150 minutes per week. I think divided by 5. That means just at least 30 minutes per day. We doing regular physical activity can control our blood glucose level. Yes. Okay. Against 30 minutes per day with intensity mild to moderate physical activity can control our blood glucose level also. Yes. Another activity is, for example, here, physical function of frailty sarcopenia. Yes, also strengthening we can take. And also another important thing is sleep. Yes, from this table you can see, yes, sitting or breaking up prolonged sitting can decrease our glucose level, decrease our blood pressure, decrease our hemoglobin A1C, and also decrease the lipid, increase the physical function, decrease depression, and overall increase our quality of life. Also another our physical activity, and also the important one is about the quality and duration of sleep. Look at this one, the blue color, adequate sleep duration, good sleep quality, yes, decrease our blood glucose level, decrease our blood pressure, decrease our hemoglobin A1C, and also decrease the cholesterol level, yes, decrease depression, and overall increase the quality of life. Okay, that's why I am, as an example, I'm always doing full-time sport or physical activity per week, at least 1 hour. Yes, it is good to control our blood glucose level. So we go to the target therapy for diabetes mellitus type 2. Yes, for the class therapy for diabetes mellitus type 2, at least we have age group of conventional medicine. Number 1 is sulfonylurea. Yes, sulfonylurea here, for example, the name of the drug is albutamide and also glyphosate. So this group work to increase the insulin secretion in pancreas organ. So that means increase the metabolism of glucose in blood enter to the cell all of our body. So the group number 2 is biguanide. Biguanide is here. For example, the name of the drug is metformin. That's, I think, the most popular one. Yes, the mechanism of action biguanide is decrease hepatic output of glucose. Yes, that means can decrease blood glucose level. Number 3 is thiazolidinedione. Yes, the name of the drug is pioglitazone that still exists until now in the market. Yes, so the mechanism action of thiazolidinedione is almost similar with biguanide, plus here also work in the muscle organs, the decrease absorption of glucose from the blood into muscle here. That means increase the sensitivity of insulin hormone. Yes, increase the sensitivity of insulin hormone. That means also work in adipocyte organ. Okay, number 4 is DPP4 inhibitor. Yes, which one? Yes, this one. Dipeptidyl peptidase 4 inhibitor. Yes, until now, at least there are 2 kind of drug that also available in the market. The name is sitagliptin and tildagliptin. They work also in pancreas to increase the secretion of insulin hormone. The group drug number 5 is alpha-glucosidase inhibitor. Yes, which one? This one. I think the name of the drug that available in the market, there are 2, miglitol and also acarbose. Yes, inhibit the enzyme of alpha-glucosidase to break down carbohydrate to become glucose. That means work in intestine organ. Yes, by that mechanism of action can decrease the absorption of glucose and also decrease the blood glucose level. Yes. So the group drug number 6 is GLP-1 analog or glucagon-like peptide 1 analog. Yes, the name of the drug is exenatides and also liraglutide. Work also in the liver organs by decreasing hepatic output of glucose. I think the new one is SGLT2 inhibitor. Yes, sodium glucose transporter 2 inhibitor that work in kidney organ. So the mechanism of action is inhibit the reabsorption of glucose in kidney tubule. Yes, that means another word is increased glucose renal excretion. Yes, the name of the drug is dapagliflozin and also canagliflozin. Of course, and the last one is insulin hormone that already available in our body can maintain the baseline of insulin hormone in our body. Yes, there are many kind of marketed product that contain insulin hormone with variation of working capacity, for example. Okay, here we can choose a specific drug and also for specific cases or patient, yes, that we have to consider according to the expected outcome therapy. Yes, for example here, metformin, the efficacy is high. The hypoglycemic effect is no. White chain is neutral. Yes. How about the cardiovascular effect? Sorry, there are. Okay. Can I continue? Okay, thank you. I will continue my presentation. Yes. For the person that possess cardiovascular risk factor, I think choosing metformin also possess potential benefit for the atherosclerotic cardiovascular disease. Yes. How about the heart failure? There is no information about that. Cost, yes, of course, lower than another method. Yes. Using of this drug is orally. How about the renal effect of metformin? Okay, contraindicated with the specific percent that eGFR less than 3 milliliter per minute per 1.33 meter square. So additional considerations, we have to also read and care about the gastrointestinal side effect common, that means diarrhea or nausea. And also potential for vitamin B12 deficiency. So also available here another conventional medicine that we have to choose. For example, SGLT2 inhibitor, GLP-1 receptor antagonist, DPP4 inhibitor, thiazolidinediones group, sulfonylurea, specifically for the second generation, and also insulin hormones, human insulin or analog insulin. With several considerations, we have to understand what kind of specific effect we want, what kind of side effect we have to care about. Okay. So I'm also presented here new therapy. Yes. There are several products for treatment of specific diabetes mellitus with specific advantage. For example, here, yes, from this table we can see nanocarrier-based therapy of diabetes mellitus and also associated complication. Yes, for example, here, polymeric neutral protamine. Yes, the composition is contain metformin. Yes, loaded with polymeric neutral protamine. For diabetes mellitus. Yes, the advantage of this treatment is controlled drug release profile as compared to pure metformin. Lowering also blood glucose level. Yes, that's the advantage if we use this new treatment. Another product is phytonatural protamine that contains selenium, clemi, Droserifolia NP. Also for diabetes mellitus patient, the advantage is significant elevation in serum glucose concentration compared with selenium itself or NP itself. Also decrease in serum insulin. That means can inhibit or prevent hypoglycemic condition. Yes. For example, also here solid lipid nanoparticle that contain valsartan loaded with Yes, SLN for diabetic foot ulcer. Yes, the advantage is small particle size and sustained drug release that we can control the drug concentration in our blood. Yes. Another one is bovine serum albumin's natural protamine, for example here, that contain apatinib, hyaluronic acids, yes, for specific patient such as diabetic retinopathy, that is the problem with the eyes. Yes, the advantage is sustained drug release, no cytotoxic effect on rabbit corneal epithelial cell, and also improve retinal accumulation. Another new therapy is using stem cell therapy. Yes. From this slide, you can see the new agent we can take from the donor or the, for example, Kemenkes program, or from pig as a xenogenetic donor, and also from stem cell derived from beta cell. Yes. So these products injects to the diabetics recipient or diabetic person or patient. Okay. After that, yes, the patient specific, the pancreatic organ will try to produce this red cell pancreatics to secrete insulin hormone. Another new treatment for diabetes mellitus is, yes, I think FDA already approved novel dual targeted treatment for diabetes mellitus type 2. Yes, you can see here in the red box. Yes, there are 3 different doses of Mounjaro, contain 5 milligram, 10 milligram, and 15 milligram. Yes, already evaluated in 5 clinical study. So the efficacy of Mounjaro is compared to placebo. GLP-1 receptor agonist, that means comparator is semaglutide, and also 2 long-acting insulin analogs. So on average, the result, patient randomized to receive the maximum recommended dose of Mounjaro, 15 milligram, had a lowering of their hemoglobin A1C, HbA1c level by 1.6% more than placebo when used as a standalone of therapy, and 1.5% more than placebo when used in combination with long-acting insulin. Yes, in a trial comparing Mounjaro to other diabetes medication, patients who receive the maximum of recommended dose of Mounjaro have lowering of their HbA1c by 0.5% more than semaglutide as a GLP-1 receptor analog, and 0.9% more than insulin degludec, and 1.0% more than insulin glargine. Yes, that means the new treatment already approved by Federation Food and Drug Administration of United States of America. So, how about the alternative treatment for diabetes mellitus come from natural resources? Yes, here I can share to you about my result in School of Pharmacy ITB. So there is a heart supplement that contain chromium picolinate, yeah, combined with mangosteen extract, and already approved by Badan POM as a health supplement. Yes, to maintain the blood glucose level in type 2 diabetes mellitus. How about the scientific study about this product? Yes, we can see here in the table. Yes, the name, yes, the marketed name is glucagon. I think the final name is glucomap. as you can see here in the slide, glucomap. Yes, look at the fasting blood glucose level. Yes, at the 0 months. Yes, from baseline, almost similar. And the first month already decreased until the third month. Yes, look at this one. If we look at this result, yes, the patients treated with glucomap at third month, yes, there are around 48% the blood glucose level become normal. That means less than 106 milligram per deciliter compared to placebo, just 22% fasting blood glucose level less than 106 milligram per deciliter. Okay, in this slide, we can show you the story of health supplement that we research in our school. Yes, the marketed name start from glucagon to become glucofast, and I think the final name that already approved by Badan POM is glucomap. Yes, this product already available in online marketplace. Yes, the name is glucomap. Yes, the collaboration between School of Pharmacy ITB and PT Mepro Bandung. Yes, you can see here the health supplement already. Yes, for maintain the blood glucose level in type 2 diabetes mellitus. Another product that also already available in Indonesian market, the name is Intrasin, that already become phytopharmaca. That means phytopharmaca. Also, yes, completed by clinical study. The indication of this drug is combination therapy with anti-diabetics medicine orally in type 2 diabetes mellitus. Yes, the group is obat modern asli Indonesia. I think Intrasin contained with Langostreomia speciosa and Cinnamon burmani. Yes, in Indonesian language is ekstrak bungur dan ekstrak kayu manis. So how this drug work in our body? At least there are two scientific information about this drug. The first one is, yes, refer the phosphorylation of insulin receptor. Yes, for example, tyrosine receptor and on time. Increase translocation of GLUT4 for cytoplasmics to membrane. And the third one is upregulation of PTR gamma. So there is the new synthesis of GLUT4. The last one is decrease TNF alpha blood level. Yes. So Intrasin works by this mechanism of action to maintain our blood glucose level. So this drug also already available in the market as a phytopharmaca. Yes. Okay. Summary for this presentation is, number one is diabetes mellitus prevalence is going to increase. So we have to care about that, and then how to prevent to become worse, for example. And the second one is researcher continue to conduct research to find more effective diabetes mellitus therapy. And the last one is a need holistic approach for diabetes mellitus treatment. For example, stress management, because stress can increase our blood glucose level. And then doing regular physical activity. Yes, and then care about our diet. That means healthy food that we have to consume. And then next one, normal body weights. Yes. Next one is we can using or consume natural product to prevent the increase of blood glucose level, yes, from pre-diabetic to become established diabetes, for example. And then, of course, we can take conventional medicines, single drug combination, or combined with insulin hormone, for example. That's all. Thank you very much for your attention. And then the time and place I give back to the moderator. Thank you very much.
Okay, thank you very much for Professor Ketut for your very informative presentation. But before we continue to the question and answer session, we ask your permission to postpone for a while because our Director General of Workforce is still with us. Do you mind, Prof. Ketut?
Okay.
If we postpone
Please.
for a while.
I'm sorry I didn't read in the chat box. There is already.
Yeah. I'm sorry,
okay,
Professor.
thank you. It's okay.
Okay, thank you so much. Over to you, Mr. and Mrs. MC.
Ladies and gentlemen, next speech from Director General of Health Workforce Ministry of Health of Republic Indonesia, Mrs. Arianti Anaya, MKM, to deliver a speech.
Oh, oke. Ya, dah. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Good afternoon, greeting to all of us. The Honorable Director of Poltekkes Kementerian Kesehatan Gorontalo, with all the Civitas Akademika Kementerian Kesehatan Gorontalo, ladies and gentlemen, all the speaker, on behalf of the Ministry of the Ministry Health of the Republic Indonesia, I would like to extend my warm welcome to all of you for being here for the third International Conference in Pharmaceutical and Health Science. It is indeed a momentum occasion as we gather here to bring forth groundbreaking discussion, collaboration, and breakthrough in the field of the pharmaceutical and health science. To begin, I would like to extend my deep gratitude to each and every one of your presence here today. Your presence signified not only your dedication and commitment to the advising the field of the pharmaceutical and health science, but also your unwavering support for knowledge sharing and scientific development. I think we agree that your contribution is invaluable, and it is what makes this conference is resounding success, hopefully. Ladies and gentlemen, also I would like to to express my gratitude to all the participants, especially for the speaker, researcher, practitioner, expert who has gathered here today. Then I know we would like to work together, especially to against the or fight the diabetic. Yeah, as we know that that is one of the disease that is the root cause about another disease like stroke, heart disease, and something like and another disease that now increase in Indonesia. This year, the conference, I think that is the good theme, yeah, current challenge and innovation in the field of diabetes mellitus. That we know that diabetic is a complex and a multifaceted condition that pose significant challenge to individual, community, healthcare system, and economy. And we know because of the now how we eat our food, we do our work and forget the exercise. That is one thing that why diabetes become increased in Indonesia. Diabetes mellitus is a chronic disease, as we know. Then and by elevated blood sugar level has witnesses a dramatic increase, yeah, in recent years, I think. We would like to against the diabetics start from the beginning. We would like to make a promotion, prevention and promotion from the beginning in the Puskesmas, such as how we encourage the Puskesmas to do the rapid test to control our blood sugar level, so we can detect earlier every diabetic mellitus in every area in Indonesia. We are gathered here, scientists, clinical, and researchers are tackling the many challenges that arise from the epidemic. And as I said before, that from prevention strategy to the early detection treatment, and hope that it can be improve the quality of life for those affected by diabetes. This conference provides an unique platform for enhancing the latest breakthroughs, sharing best practice, and also fostering the collaboration among the professionals from various disciplines. Our aim is to foster an environment where experts can engage in stimulating discussion, explore new research avenue, and empower one another with evidence-based knowledge and practical solution. Over the next few days, we have a meeting, yeah, with the speaker, yeah, and then from exploring the latest technology advancement to discussion about the how we against the diabetes. And I think that is the good opportunity for us to discuss about the latest medical device for the prevention, also the pharmaceutical. Now there are so many pharmaceutical that is developed from the industrial who want to against the diabetic, yeah. And moreover, this conference is an opportunity to acknowledge and appreciate the theme of that Congress, yeah. And I hope that we can work together and And I believe by recognizing their dedication, we are not only express our gratitude, but also draw attention to the significant impact, especially for diabetic, and hope that we can intervention to the patient and to their family. I encourage each and every one of you to actively engage in the conference, the participate in the scientific session, attend poster presentation, and network with your peer. This is an I think this is there's an opportunity to connect with the professional, build a collaborative partnership, yeah, and will continue to inspiring that and support your professional growth. Once again, I would like to extend my warm welcome to all the dignitary, speaker, researcher, student, and also the delegate who has traveled from near and far to be a part of this conference. And let us seize this opportunity to embrace the exciting possibility that lie ahead, share our expertise, and collaborate to bring about the transformative change that will shape the trajectory of global healthcare. Ladies and gentlemen, in conclusion, I would like to extend my deep gratitude to all the participants for join us with the symposium. And together, let us pave the way for the healthier future, where the burden of the diabetic is reduced and innovation break new ground. Throughout this conference, we have the opportunity to engage in various discussion, presentation, and workshop related to the pharmaceutical, medical device, and health science. We will address current challenge and explore opportunity to improve healthcare delivery, enhance patient outcome, and contribute to the overall development of the pharmaceutical and health science profession. Thank you, and hope that we have a memorable experience in this international conference. Terima kasih. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam.
Waalaikumsalam.
Thank you very much for this speech.
Next, we are continue the plenary sessions. Please to Mrs. Dewi Agustini to moderate.
Okay, thank you so much for Mr. and also Mrs. MC. Hello, Professor Ikatan. Are you still with us?
Yes.
Okay, thank you for your kindness to wait for a while. Okay, we are going to the next session. This is a question and also answer session. If you have any question, you can type your question in the chat box, or you can raise your hand in Zoom meeting application. Okay, I think we have two participants who raise their hand, Prof. I'm going to the first participant, Miss Rahmatia Nyura.
Yeah. Okay.
Okay, you can give your question directly to Prof. Ketut.
Yes.
Bismillah. Thank you for the opportunity, moderator. Let me introduce myself. My name is Rahmatia Nyura. Professor, my
Yes.
question is about, can diabetes mellitus be caused by genetic heredity? And if so, how can we prevent or reduce the potential for diabetes mellitus
if we have genetic diabetes mellitus? Can be a healthy lifestyle help reduce its potential? Thank you.
Yes.
Okay, thank you so much for Miss Rahmatia Nyura for the question. I think you have a family who have diabetic mellitus. Okay, over to you, Professor Ketut. Maybe you can
Okay.
answer the question from
Yes.
Miss Rahmatia.
Thank you very much, Ms. Ramatia. I think very good question. So you asked about the genetic factor for diabetes mellitus. Yes, of course, there are. I think until now, as I understand, there is a close related between genetics and diabetes mellitus, and also other degenerative disease. Yes, you can see from the many scientific journal, any kind of disease, there is relate to genetic factor, directly or indirectly. For diabetes mellitus, I think there is a direct correlation between genetic factor and diabetes mellitus. Yes, I think you same with me. I'm also has genetic factor for diabetes mellitus. That's why I'm consciously control my modified risk factor. As I already explained, there are several modified risk factors that can prevent the manifestation of diabetes mellitus caused by genetic factor. Yes. For example, body weight. As long as I control my normal body weight. Until now, my body weight is 62 kg. That means, according to body mass index, BMI, my ring of BMI at 21 kg per meter square. That means a normal range for body weight index, body mass index. Yes. So that means our body weight in normal range. Okay, that's the second, the first factor. The second factor is regular physical activity or exercise or sport. So that's why every week I'm doing sport at least 4 times. Yes, and then the third one is diet. Okay, I try to reduce my carbohydrate intakes and increase the fiber intakes or vegetable. And then how to prove that I'm not yet getting diabetes mellitus? Yes, if I get chance to doing medical checkup, MCO, for example. So of course, one parameter I have to measure about blood glucose level, fasting or 2 hours after taking meal, isn't it? And then until now, still in normal range. That means less than 100 milligram per deciliter in fasting blood glucose level. So don't worry about genetic factor as long as modified risk factor you control toughly. Your body weight, your lifestyle, your regular physical activity, and then also about your diet. Okay, also the important thing is stress management, because stress can increase the glucocorticoid hormone, isn't it? As you know, glucocorticoid hormone working also can increase the blood glucose level. So that's why you also have to manage your stress. I hope your question can I'm answer for my statement. Okay.
Okay, thank you so much, Professor Ketut. So, Ms. Rahmatia, you
Yeah.
don't have to be sad if you have a family who get diabetes mellitus.
Yeah.
Because that is something that we cannot fix it, like Professor
Yes.
Ketut said before. Yeah, but we can modify the factors by doing some activity that can reduce the risk of the diabetes mellitus. Thank you so much,
Yes.
Professor Ketut, for your explanation. We are going to the next participant who raised his hand. We have Mr. Arlan Imran.
Yep. Thank you,
Okay,
Mrs. Rahma.
please you can ask
Thank you,
directly
Mrs.
to
Rahma.
Professor Ketut.
Thank you for the opportunity. Very interesting material. Permission to ask Prof. In the previous
Yes.
speaker, alkaloid and flavonoid can treat diabetes mellitus. And other side, alkaloid and flavonoid are secondary metabolites then can be found in all plants. My question is there a standard reference book or parameter book can be used to collect plant potential, dosage, mechanism, and update and integrate with herbal and pharmacopoeia herbal Indonesia? Because currently there are only few plants in the Indonesian herbal medicine formularium that are in the Indonesian herbal pharmacopoeia. Thank you, Prof.
Thank you, Mr. Alan, for your questions. I think natural product is also my main interest. Yes, so that's why I always doing research for natural product to try discover new medicine for treatment of degenerative disease. So from your question, I think for your information, there are many kind of alkaloid compound, and also there are many kind of flavonoid compound in our plant. So as long as I know, until now, there is no specific alkaloid chemical compound or flavonoid compound can claim their activity as a anti-diabetic agent. That's the main problem, I think. But as long as I know, also flavonoid is belong to group phenolic compound, isn't it? Yeah, as we know, phenolic compound possess antioxidant activity. Yes. As we also know from the pathogenesis of diabetes mellitus, yes, diabetes mellitus can generate from free radical. So, for a common sense, I think the plants that contain or rich with flavonoid compound can use as antioxidant agents, can prevent for the early step or the early process of diabetes mellitus. So that's why you can consume any kind of plants that contain or rich with the flavonoid. That means rich with antioxidants activity. So because free radical can cause many kind of degenerative disease. That means if you regularly consume the product that rich with flavonoids, that means also increase the antioxidant activity level that can prevent any kind of degenerative disease. So for information, until now, in the market, there is at least one product belong to phytopharmaca group that already presented in my presentation is enlacin that contain bungur extract, you know? Yes, bungur extract, if you search in the scientific journal, there are many journals that also state possess anti-diabetic properties. Also, kayu manis extract, cinnamon burmani. Another plants, based on my experience in research, for example, is what is called pati, momor bicarbonate. Yes, there are many scientific journal already published that claim for diabetic mellitus agent, and also do you know about jamblang? Yes, jamblang or jambolan also possess anti-diabetic agent. So don't worry about the natural product that rich available in Indonesia. There are many scientific journals already published that contain anti-diabetic agent. Yes, you can read from internet or from another media about the pharmacological activity. Okay, thank you very much, Mr. Alan.
Okay, thank you very
You,
much,
Professor. Yeah.
for your explanation. Okay, we still have the next question is from Miss Siti Romlah Yahya. Okay, you can ask directly to Prof. Ketut.
Thank you very much for the opportunity. Let me introduce myself. My name is Siti Romlah Yahya, and I will take this opportunity to ask to the expert directly. That is my lecturer at Bandung Institute of Technology. Thank you for joining us, Professor.
Thank you.
So my question is, if a patient of diabetes mellitus receives any conventional treatment regularly, is it okay to consume natural products at the same time? Thank you.
Okay. So thank you very much, Ms. Siti. Thank you very much also for your invitation to share my knowledge in this event, I think. Okay. So your question is, I think the common question for everyone, it's okay, the combination between conventional medicine and natural product agent, isn't it? Okay, yes. It's, I think, a very, very good question. Why? Yes, of course, there are many Indonesian people with their tradition using natural product, isn't it? And then some of them also taking conventional medicine. Of course, also there is a case in diabetes mellitus patient, of course. Okay, so the main question is it okay or is it safe if we taking together between conventional medicine and natural products agent? Yes, I think this is our main role as a apotheker or nurse or physician, isn't it? So I think better we find more information about the about the proposed mechanism of action of natural product. Of course, from conventional medicine, we already know about the mechanism of action, as already explained in my PowerPoint. There are at least 5 group of conventional medicine that work as an anti-diabetic agent, isn't it? Such as sulfonylurea, biguanide, DPP inhibitor, and also diazolidinedione, acarbose, and also SGLT2. So we know which side they work and then what kind of mechanism of action. So as a general knowledge, if the mechanism of action is different, yes, commonly we can combine it. Because from the guideline also, if we use 2 conventional medicine, that means must be different mechanism of action. For example, metformin combined with sulfonylurea or diazolidinedione combined with sulfonylurea or acarbose, for example. Yes. So that means if we can predict the mechanism of action of natural product in which parts they work, and then we know conventional medicine we want to take also possess a different mechanism of action, of course, we can combine both of them, isn't it? And then the second one, so we can roughly monitor about the blood glucose level. That's, I think, the easier parameter, isn't it? If still normal, that means there is no problem with that combination. If to lower the blood glucose level, that means we have to adjust the dose or the stop another one, for example. Or if the blood glucose level stay higher, maybe the combination is not right. We have to change or we have to modify, for example. So based on my experience in the laboratorium, yes, I'm already combined between mangosteen extract with metformin. There is no problem with that both combination. Yes, so we can combine using mangosteen extract with metformin, for example. So I think that a common generic answer from me about your question. Thank you very much, Miss Siti. Success for you all.
Okay, thank you very much, Professor Ketut. Related to the question of Miss Siti Romlah before,
Yes.
Ketut, can I ask something to you?
Okay.
If we,
With my pleasure.
yeah, after we count the risk factor like you show before in
Yeah.
your slide, and let's say
Yes.
our number is 4 or 5, and then
Yeah.
you show also the nature medicine like glucomap,
Yeah.
can we consume it for a daily basis if we don't get that diabetes mellitus, only have a risk?
Yes.
Okay, thank you.
So I think, yes, thank you very much. That's also my concern. Yes, the main position of natural product is to prevent the pre-diabetic person to become established diabetic person. Yes, for example, if the blood glucose level around 110 to 126, isn't it? Still pre-diabetic person. If the blood glucose level in fasting condition already more than 126 milligram per deciliter, already become established diabetes mellitus system. From my research, I think natural product position is how to prevent from pre-condition to become established condition. So that's why better we choose natural product first before we decide to choose conventional medicine. So according my result from the diabetic research, so I think it's good we use natural product medicine first to prevent the serious side effect from the conventional medicine. So that's, I think, the main position of natural product medicine, and also safer than conventional medicine. That means less with side effect, and then also, of course, the cost cheaper than conventional medicine. Okay, and then we can use for long time to maintain our blood glucose level.
So
Maybe
much.
my answer. Yes, thank you very much.
Yeah, thank you so much, Prof. Ketut, for your explanation. Okay, I think we still have a question from participant who raised her hand. We have Miss Nur Nikma Ismail. Okay, for Miss Nur Nikma, please open your mic and give your question directly to Professor Ketut.
Okay, thank you for the opportunity given to me. Let me introduce myself. My name is Nurnikma Putri Ismail, and my question is, based on the questionnaire for screening type 2 diabetes mellitus, how can the
Yes.
age and gender contribute to risk factors for type 2 diabetes? Thank you.
Okay, thank you very much. I think a very nice question from Ms. Nur. I'm really glad in this event because all the participants almost active to ask the question. Okay, there is one question about the risk factor for gender and age, isn't it? Yes. I think in common answer about the age, yes. By increasing the age, I think all our organ system function is decreased, isn't it? Yes, as long as we know all the organ system function decrease when our age increase. Yes, when our age increase. That means we will face many problems with any kind of disease, especially degenerative disease, including diabetes mellitus. Yes, you can see many scientific journals. Age already become a risk factor for all any kind of degenerative disease, not just diabetes mellitus. For example, asthma, kidney disease, osteoarthritis, especially osteoporosis and any kind. So we have to access. That's why age already belong to non-modified risk factor, isn't it? So we must account age. If our age already more than 55 years old, that's mean age already become a risk factor of kind degenerative diseases. Yes, we have to accept it without any objection. Okay. And the second one about the gender. Yes, as you know, men and women possess hormonal profile, isn't it? Different. Yes, that also the main problem, and also the characteristic of physical bodies of that people. Yes, so that's why from the screening methods, yes, men possess higher potency for diabetes mellitus compared to women. Yes, might be caused by the diet profile of men. That yes, from the volume of most much more than women. And next one is maybe the composition of diet. Yes, rich with carbohydrate and also rich with fat, for example. And the second one might be about the body composition. That also the main problem with diabetes mellitus, because as you know, the population of diabetic mellitus is diabetes mellitus type 2 caused by obesity. Obesity caused by the composition of our body, how much the fat, how much the muscle, how much the what's the water, and then something else. So that's why gender also become the unmodified risk factor. Age also become unmodified risk factor for any kind of degenerative disease. So according to the philosophical talking, yes, degenerative disease basically caused by our habit, not caused by external factor, caused by our habit. So that's why we must realize that. So that's why I am toughly control the modified risk factors such as body weight, stress management, and then regular physical activity and also diet. I think I hope answer your question, Ms. Nur. Thank you very much.
Thank you very much, Professor Ketut. I have a additional question again related to the question from Dornikma. Just like you mentioned before on your presentation, you said that Javanese ethnic possess higher risk to get diabetes mellitus. As a Javanese person, I'm start worry about it, Professor. Can you explain a little for me about this factor?
Thank you, Ms. Rahmadewi. Okay, I think the first thing we have to do to be honest to ourselves, isn't it? So look at about the history background of your family, especially for your diet pattern. Yes, commonly the Japanese foods, the taste is what? Sweet, isn't it? Yes, for example, gudeg. Yes, gudeg is sweet, isn't it? Yes, gudeg is sweet. Yes, and then for the pattern of diet in Sundanese people is vegetable. It's called lalaban. Yes, for Padang people almost rich with santan, isn't it? Coconut.
Yes.
Yeah, so that's why for Padang people, the common degenerative disease is hypercholesterol because the diet rich with santan or coconut. So for Sundanese people, rich with fiber, that's why the skin is also glowing, isn't it? Because rich with lalapan, for example. So the Japanese people, the pattern of diet is mostly sweet. So that's why there is a genetic for that people, because according to mass media reports, yes, more than 70% the member of DPRD in Jawa Tengah, yes, suffer from diabetes mellitus. Yes, because the diet pattern of that person. So that's why we must realize that story. So according to the research also, in Japan, Japanese person, the diet pattern is very, very healthy. Yes, very, very healthy. So when the Japan people move to America and adopt the lifestyle of the American, the prevalence of diabetes mellitus in the immigrated Japanese people to America increase. Because why? Because they adopt the the diet pattern, and the lifestyle is the wrong lifestyle for their genetic. So that's why the prevalence of diabetes mellitus in migrated Japanese people increase. So that's already published in the scientific journal, I think. So that's why I state that Japanese people possess high risk diabetes mellitus compared to another ethnic because of the history of their diet pattern. Thank you very much, Mr. Madewi. Yes.
Okay.
You can
Thank you
change
very
it if you change your yes, your diet.
Okay, I will do. So
Yeah.
I think Gorontalo people is healthier than Japanese people because Gorontalo
Yes.
people really likes spicy food.
Yeah.
Yeah,
So
I
that's
think
mean
chili
they are okay.
contain a lot of vitamin C.
Yes.
Okay, I think we have one question in the chat box, Prof.
Yes.
Okay.
From Fitriah Ayu
Yes, from
Magfirah.
Fitriayu. Yes.
Okay, I read her questions. Diabetes sufferer sometimes experience complications such as hypoglycemic condition. How do we control this? Is it necessary to stop using diabetes medication? Of course, I think the first step we have to do, we have to stop the medication first, and then we check our blood glucose level. Yes. Okay. If lower than 80 or 70 milligram per deciliter, that means we get from the hypoglycemic condition. Yes. Okay. So we have to evaluate again our treatment or our therapy. Better we adjust the dose, adjust the duration, or change the medication. Okay, and then also about our lifestyle. Yes, maybe we doing physical activity too hard with low of carbohydrate consumption, for example, and also taking medication, especially insulin, for example, or sulfonylurea group, for example, because the side effect of insulin hormone and sulfonylurea medicine is hypoglycemic condition, especially if we combine with hard physical activity or low carbohydrate intake. So better we get a discussion with the pharmacist or apotheker or with clinician to control our recently condition. Thank you very much, Ms. Pitria, for your question.
Okay, thank you very much, Professor Ketut. Okay, Prof. Ketut, your material is very interesting for me. I have so many questions
Thank you
for
very
you.
much. Yeah.
Okay, you mentioned before on your presentation that the number of prevalency of Alzheimer is getting increased. It is more than 2 times.
Yeah.
Related to the diabetes mellitus, is there any correlation between diabetes mellitus and also Alzheimer?
Exactly, there is close related with diabetes mellitus. Why? Because the main food for our brain is glucose, and our brain cannot make glucose by itself. So the brain getting glucose from the liver. So if our cell brain lack with glucose, of course, our brain cell will be die, isn't it? So we're getting Alzheimer. So that's why, as you know, one of conventional medicine, it's called nasal insulin hormone. The indication for treatment of Alzheimer, not for treatment of diabetes mellitus. Why? Because nasal treatment, insulin hormone will easier to absorb to the brain cell. So that's why nasal insulin hormone or products, the indication for the treatment of Alzheimer. I think I can answer your question. Thank you, Ms. Ramadevi. Yeah.
Okay, thank you very much, Professor Ketut. Okay, all of participants, are you still have any question? We still have a few minutes before the session end. Okay, I think all of the participants here and also in Zoom meeting is clear enough about your presentation, Professor Ketut. Okay, ladies and gentlemen, we have come to the end of the question and also answer session with Professor Ketut Adiana. Thank you all for your attending in the session on update of diabetes mellitus. I hope you found it informative and also engaging. Okay, this is the certificate of appreciation, Prof. Ketut, from committee and also from Poltekkes Kemenkes Gorontalo.
Yes.
Please kindly
Salam.
receive it even though in Zoom meeting.
Okay, thank you very much.
Yeah, thank you very much, Professor Ketut. And we hope that the information shared today will inspire for further research, collaboration, and innovative approach in terms of diabetes mellitus. Thank you once again for Professor Ketut Adiana, to all of
Thank you very much.
Yeah, thank you for being part of this international conference in pharmaceutical and health science 2023. We wish you success and fruitful discussion in the upcoming session. Thank you.
Okay, thank you very much. Very glad to participate in this event. Yeah, success for you all. Okay, I leave. It's okay?
Yes.
Thank you very much.
Thank you very much, Ketut, Professor Ketut, and also all the participants. Thank you. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam. Thank you very much to our keynote speech, Professor Ketut Adiana, MSI, PhD, and our moderator, Rahma Dewi Agustini, SST, MCAB. We are now continuing our meeting to the next agenda, that is to hear last keynote speech for today, which will be delivered by Professor Andi Dian Permana, MSI, PhD, APT, from Faculty of Farmasi, Hasanuddin University, Makassar, Indonesia, will be moderated by Mr. Fadli Hussain, SSE, MSE.
Okay, thank you, Mr. MC. Maybe we're waiting for Professor Dian. Professor Dian is still break for prayer, so maybe we will start in 3:30. Thank you.
every day. Okay, I got the keys to the universe, so stay with me. 'Cause I got the keys, baby. Don't wanna wake up one day.
Check. Okay, good afternoon, Professor Dian. Professor Dian, can you hear me?
Yes, I can hear you.
Okay, thank you. Okay, thank you, Professor Dian, to joining with us. Okay, ladies and gentlemen, for all participants for this conference, please introduce for this session. We have speaker, Professor Andi Dian Permana, MSc, PhD, apoteker. First of all, I want to say thank you, Professor Dian, for joining our conference for today. First, I will read your CV before. Professor Andi Dian Permana, the speaker for today, with the title of the presentation is Microneedle Delivery System for Diabetes Treatment. So, Professor Andi Dian Permana is a professor in Transdermal and Dermal Drug Delivery System, Department of Pharmaceutical Technology, Faculty of Pharmacy, Hasanuddin University. His academic qualification, bachelor degree of Bachelor of Science, Hasanuddin University, Indonesia, in 2006 until 2010. Pharmacist professional degree in Hasanuddin University, Indonesia, 2010 until 2011. His master degree in pharmaceutical science is from Hasanuddin University, Indonesia, 2012 until 2014. Professional license or PIL 1968 under the Animal Scientific Procedure Act 1890 on December 2017. His PhD in pharmacy is from Queen's University Belfast in 2016 until 2019. His thesis title is Development of Anthelmintic and Antibiotic Microneedle Delivery System for Treatment of Lymphatic Filariasis. His contact, you can contact Professor Dian, andi.dian.permana@farmasi.unhas.ac.id. His research experience, more than 5 in 2000, start from 2012 until 2014. Graduate researcher from Hasanuddin University, Indonesia. Master student in the field of development and characterization of nanovesicle carrier of natural products for transdermal delivery. In 2014, he is a visiting researcher for Sakura Science Program in University of Toyama, Japan. And then his PhD degree is graduate from Queen's University Belfast, UK. And his postdoctoral researcher in 2021 to Department of Biosciences, Chemical Biology and Biological Therapeutic in University of Salzburg. And 2023, he is a visiting researcher, Department of Applied Chemistry in Faculty of Engineering, Kyushu University, Japan, 2023. His teaching experience is supervised the final research project of final year and undergraduate student for BSc Pharmaceutical Science. He is also supervised the master thesis of master student in Pharmaceutical Science, and also his student supervision in Hasanuddin University from 2012 until 2016 and January 2020. His university teaching in Hasanuddin University 2012 until 2016, January 2020 until now. Professor Dian also have secured funding from 2015 until now, research and development of medical sciences from Ministry of Health, and then funded by Tanoto Research Foundation, and also National Competitive Program and College Assignment, postgraduate research, master thesis, and also from Ministry of Education, and former is from Brain Research Grant. His oral presentation is about nanoparticle and microneedling, and also a lot of poster presentation. Professor Dian, when I read it, all of this one, we need more time. Maybe I can describe to you, Professor Dian is basic or specifically for nanoparticle and microneedling. Okay, that's all for curriculum vitae of Professor Dian. Now, for this nice occasion, we will hear the great presentation from Professor Andi Dian Permana with the title of Microneedle Delivery System for Diabetes Treatment. So, Professor Dian, we have approximately 2 hours for your delivering your presentation, including discussion and ask and question and ask session. So, for the presentation, we please Professor Dian to deliver the presentation. Thank you.
Thank you very much, Mr. Fadli. Everyone hear my voice now? Hello? Can you hear me clearly? Or
Okay.
do you have any problem with the voice? Or
Yeah, your voice crystal
okay,
clear.
thank you.
Please.
Okay, hello everyone. Hello, Mr. Fadli. It's finally happy to see you here after several times you come to Makassar, but we haven't met yet even one time. And finally we meet here through online. So thank you everyone. Thank you very much for Poltekkes Kemenkes Gorontalo for inviting me to deliver a topic regarding the themes of the conference. So sorry, because my voice is not normal. I have a flu now. So yeah, let me share the screen. Hopefully it won't take that long because, I hope that we can have a discussion during the session. So, okay, can you see my screen? Or it's okay, right? Okay, I think it's
Okay,
fine.
it's okay.
So, what I'm gonna be talking about the use of macroneedles in terms of diabetes treatment, because the topic of the conference is about diabetes. So we're gonna talk about the possibility and the application of microneedles to treat diabetes treatment, even though the microneedle is not reaching the market yet, but a lot of researchers around the world already developed this kind of the system to treat or to overcome several problems of diabetes treatment. So my name is Dian. I know that the moderator already introduced me. So my name is Dian. I'm from the Faculty of Pharmacy, Hasanuddin University. So basically, I'm working on drug delivery system. I'm from Department of Pharmaceutical Technology. So before going forward to my explanation, I just want to show the audience about my research activities, just to show the people that I'm doing several development in drug delivery systems. So maybe if any one of you wants to do some collaborations, you can contact me after the conference actually. So basically, I'm working on several routes of drug administration. So the main topic of my research is about the skin delivery system. Oh, skin should be there. Sorry, skin should be here. So my main activities is about skin drug delivery systems, and in this kind of systems, we developed a lot of transdermal patch formulation, and now during the past 4 years, we developed like dissolving microneedle formulations, both for local and systemic applications. So we're not only targeting local effects, but also systemic effects using these systems. And I'm also keen on developing particle formulations, including nanoparticle and microparticle formulations. And both of these systems we included into transdermal and topical drug delivery systems. And some of my study, we developed skin infection model, but we don't directly apply the infection models into the animals. But first, we develop ex vivo infection models in the skin, for example, and we'll use bacterial and fungi strains to provide the infection of the skin. And also, we developed a dermatokinetic study to show the profile of the transdermal or dermal delivery drug in the skin. And we are doing in vivo pharmacokinetic study following administration of several formulations applied to the skin. And we are also working on ocular drug delivery systems. So I'm not only working on skin, but also ocular drug delivery system and vagina drug delivery system. So I'm also working on vagina drug delivery system, not vagina, but vagina drug delivery system, okay? So we're working on how to deliver the drug to the systemic and local activities through the vagina tissue, and also the ocular tissue, of course. And also oral drug delivery systems. It's a common drug delivery system, but I'm trying to also develop some systems using this route. And also the most important part of this kind of development is analytical and bioanalytical drugs that topics, because in terms of why I'm trying to explain this, because especially in diabetes treatment, so we use a lot of drugs, and in order to show that this system will be working on diabetes, we have to show that we are able to control the drug release, for example. And in order to show that we are able to control the drug release, we have to show the profiles of the drug using HPLC, for example, or other treatment or other instruments. So this is the main topics that I'm working now. And before showing you guys about what kind of systems or drug delivery system that we can use to treat the diabetes. So according to the topic, I'm gonna be talking about the drug delivery system for diabetes treatment. So I know it's not something new. I know it's well known by a lot of people, especially in Poltekkes, Kemenkes, and everyone watching this video. So you know that, everyone knows that diabetes is a chronic condition, chronic disease, which is caused by absolute lack of insulin and relative lack of insulin, and this is as a result of impaired insulin secretion and action. So this is there are a lot of people try to define what is diabetes, but this is the main idea what is diabetes. So the pharmacological is I'm not trying to explain the pharmacological effect of this or something like that, but I'm just trying to show the surface knowledge about the diabetes before going to the drug delivery system of this. So let me try to hide something here because I'm okay. All right. So this kind of disease can contribute the development of some complications. Like, that diabetes can be complicated, can be really complicated because it's not only single disease, but a lot of diseases can also be attacking people because people have diabetes. Like, oh, sorry. like cardiovascular, retinopathy, nephropathy, neuropathy, and high risk of cancer. So this is one of the problems of diabetes, because it's not only affecting the insulin secretion, for example, but also it can affect a lot of part of the body, like cardiovascular and also cancer. I'm not going deep with this. And according to the population, it has been reported that globally in 2019, according to the report, there are around 9.3 of the total population in the world, they have diabetes. So it's not a small number. It's really high number, like 463 million people in the world have this diabetes. And it is predicted, so it is already predicted that around it will be increasing. So the increase will be until 10.9% of the total population will be 700 million people around the world will have this diabetes. So it is also reported that 1 in 2 people living with diabetes do not know that they have diabetes. So please, guys, according to the sentence, please check your glucose blood level, because around 50% of these people, they don't know that they have diabetes. So please check your glucose. It's just introduction. So according to the pathophysiology, most of cases of diabetes can assign to type 2 or type 1 diabetes. So we have this kind of type of diabetes. I'm not going to this in detail, but just to say again that type 1 diabetes usually results from autoimmune destruction of pancreatic beta cells, but the type 2 diabetes is characterized by impaired insulin secretion and resistance of the insulin action. So this is the type of diabetes. And with respect to the pharmacological treatment, so basically there are nowadays, especially in Indonesia, we use a lot of kind of things, a lot of kind of treatment that people do to treat the diabetes. So according to some reports, so in terms of the pharmacological treatment, so the main idea, the main drug or the main compound that people use is using the insulin. And it is used along with diet, because it is crucial to the survival of individual with type 1 diabetes. And also, it also plays a major role in the therapy of the people with type 2 diabetes. So this is one of the most effective drug at the moment for the diabetes treatment. And also, we have seen here a lot of this is not something new. Like nowadays, a lot of people already use also, in addition to insulin, so people also use sulfonylureas, so it can enhance the release of insulin from pancreatic islets. And biguanides, this is the most common also. People use metformin, buformin, so it can reduce the hepatic glucose production. So they also use pemafibrate and also acarbose as a part of alpha glucosidase inhibitors. So there are a lot of oral treatment of diabetes at the moment. However, so we are going to the main problems, because why drug delivery system is really important, because the use of conventional drug delivery system is not effective anymore. So that drug delivery system plays really important role to control the release of the drug, to reduce the side effects, to improve the efficacy of some medication, including the diabetes mellitus. So, although in clinic, the oral administration of the chemical compound using drugs for the treatment of diabetes, it's not. It is effective. People say it's effective, but people people find a lot of problems when they use oral oral anti-diabetic drugs. They found when they use this kind of drugs, they will face severe hypoglycemia, and also they will weight gain. And it has been also reported that nowadays the oral anti-diabetic drugs have lower therapeutic efficacy, and also it is also reported that it is an effective dosage regimen, and they have been shown that they will show low potency and altered side effects due to the drug metabolism. So some of drugs you might know that in oral administration, the drug will be metabolized in the hipper, so it will increase some problems like low potency because the concentration of the drug will be decreased due to the metabolism, but also sometimes like the metabolism of drug can form a new compound that can increase the side effects of the drug. So it is these two things that will be faced if we consume the drug by the oral administration, especially when it is metabolized into the hyper activities. So it is also reported there is lack of target specificity. So I mean, I will show you some example later, but for example, why several anti-diabetics oral anti-diabetic drug can show lack of target specificity, because like it is important to control the glucose in the blood, the glucose level in the blood. So if we don't control the concentration of the drug, the concentration of the glucose in the blood also will be uncontrolled. So it is really important to improve the specificity of the delivery of the drug into the body, because it can control the blood glucose level. And the main important problem also is because of the characteristics and the physicochemical properties of the drug. They will have problems in terms of solubility and permeability. So you know that if the drug have the problem with the solubility, it will have the problem with the bioavailability. So, for example, if the bioavailability is low, people need to take a high dose of the drug, meaning that if they take a higher dose of the drug, for example, so it will have a higher possibility to show the side effects of the body. So this is the main problem if they have the low solubility. And not only that, it also shows some problems in terms of permeability problems. So it can be soluble in the body, but the problem is if not permeate to the cell, so it cannot reach the target. So it is the main problem of the anti-diabetics. So that's why, because of all of these problems, because of all of these drawbacks of giving people oral anti-diabetic drugs. So nowadays, a lot of researchers in terms of a drug delivery system develop some systems. So it's not, I mean, there are a lot of drug delivery systems that people use to improve the efficacy of the diabetes treatment. But in this study, in this presentation, what I'm going to be showing you guys is the use of macroneedle delivery system. So this is one of the systems that people use to improve the efficacy of the diabetes treatment. So, sorry, let me have a drink first. Okay, so this is one of the system that people use, that people try to develop to to avoid a problem in terms of the bioavailability in the application of oral drug delivery system of anti-diabetics. So what is microneedles actually? So I know that some of you may know this kind of systems, but let's try to forget about this information before. Let's assume that this is something new, okay? So this is one of new platforms for drug delivery. So it can consist of micro-sized needles, which have different diameters and length with several types of materials. So if you see these examples, so this is the examples of one of microneedles. So it's actually like a patch. If you guys are familiar with the transdermal patch, it is something like a patch, like a plaster, like a wound healing plaster that contain a needle on the surface of that part of the patch actually. So this is one of the benefits of this kind of drug delivery system, because patch have some problem with the permeability, because the drug is stay in the surface of the skin, and then it can cause some irritation because it can contact the skin in the long term. But with the help of the needles here, So with the help of the needles, it can improve the permeability of the drug, and it can avoid the long-term contact of the drug in the skin. So it can also avoid the possibility of irritation of the skin, because the drug is inside the needles, and once we apply the drug in the skin, and once the needle dissolves, we can remove the patch, and the needle will stay in the skin with the drug. So the drug will be delivered to the systemic part of the body if we want to deliver them systemically, or the drug will stay in the skin if you want to deliver them locally. So it's like something that really interested to develop, because it is really, I can say it's a really new systems in Indonesia, because I remember when I did my PhD, one of the reasons why I chose that university is because there was no researcher in Indonesia using this kind of system. So I'm not trying to showing off or or be arrogant with this, but this is the systems. Like when I finished my PhD, I was the first Indonesian who used this system for drug delivery system. But now we found a lot of Indonesian researchers using this kind of drug delivery platform to improve the efficacy of different drugs, which I'm really proud of, because I can collaborate with a lot of people now, because I'm not the only one using the system to develop a drug delivery system. So in conclusion, we can see that this is the combination of transdermal patch and injection. But injection it can cause pain, but it cannot cause any pain, because the needle is attached with the surface of the patch, and the needle size is really small. It doesn't reach a pain nerve, so it doesn't cause any pain. So this is the main idea of this. So what is, well, actually we can see a lot of types of microneedles. So here we can see like solid microneedles, hollow microneedles, coated microneedles, dissolving hydrogel forming microneedles. So sorry. So here is the illustration of the use of the microneedles. So if we have a look in the first type of microneedles here, so it is only used to make a hole, to create a hole in the skin. So this is not actually a real drug delivery systems, because the solid microneedles is only used to create a hole in the skin, and then we apply the real drug delivery system. So it can be used to help the penetration of the drug. It doesn't contain any drug at all. So here it is only used to create a hole in the skin, and then it is removed, and then we apply the skin patch, for example, and we also can apply transdermal cream, gel, or something like that. So if you are aware with the the derma roller, guys, so if you go to the skin treatment, so sometimes they use a derma roller on your face, and then they apply a serum to improve the efficacy of a vitamin or any skin product to improve the efficacy of this. So it is actually one of the old. It is not a new. It is the really old type of microneedles, because it doesn't contain any drug. It's just used to create a hole in the skin and then apply to help the penetration of the drug in the skin. So okay, this is the first type of the solid microneedle. The second type of microneedle is hollow microneedles. So hollow microneedles is like the similar types of normal injection, but it is really small. So this is similar with the solid microneedles actually, but it contains a small hollow in the middle part of the needle. So it contains hollow something like this, and then the drug will be placed in the top of the formulation, and then the drug will be permeate using this through the hollow, and then we will remove the formulation, and then the drug will stay in the skin. So it also doesn't contain a drug. It is only it is only a media to facilitate the penetration of the drug through the skin, through the hollow of the needles. Okay. The third part of the microneedles is a coated microneedle. So the coated microneedle, it's not, I mean, like it is also only like a tools, because in this part of needles, so it is similar with the solid microneedles actually. Oh. Okay. If someone say something, okay. Can the host help you to? Okay, it's mute now. Okay, so where was I? Forgot. coated microneedle. So in terms of the application of coated microneedles, so we can see here that it is similar to solid microneedle. So this is needles. So this is needles, but the needle will be like we will prepare a drug solution. So the drug solution, like for example, we have drug solution here, we have the needle here, and the needle we will do some deep coating. So we will pour the needle to the drug solution, and then take the needles, and then just wait until the drug will be dry in the surface of the needle, and then we will apply the drug in the we will apply the needle in the skin, and then after several times we remove the needles, and then the drug will permeate through the skin. So basically, I can say that the type of microneedle we can divide into two parts. So the first part is the first three types of the microneedles. It is solid microneedles, hollow, and coated, because it doesn't contain any drug. It is only used to facilitate the penetration of the drug using the hollow that they created. Okay. And then the second microneedle is real. I mean, I can say it's a real microneedle, because it is developed like recently compared to those three microneedle types. So dissolving microneedle. So this is the microneedles that Ibu Sulfi Ayu will prepare. So this is dissolving microneedle. So dissolving microneedle is one of the needles that is containing, it will contain a drug actually. So different from other part of the needle, different from the other types of microneedle. In this type of needles, the drug will be incorporated into the needle. So actually, the needle is like the needle will be containing the drug. So it is made by water-soluble polymer for the dissolving microneedle. We can also use long-acting polymer to create long-acting microneedle. So this is the needle is made for the polymer and the drug. And then once we apply the needle in the skin, because the name is dissolving microneedle, and the drug will be dissolved with the help of the polymers, and the drug will be permeate to the skin. It is the the short explanation about the dissolving microneedle. So the last part of microneedle is the hydrogel forming microneedle. This is the newest. This is the youngest generation of microneedle system. It is actually this is actually developed by my professor, Professor Ryan Donnelly. He created this type of microneedle because of some limitations of dissolving microneedles. So this is one of the type of dissolving polymeric microneedles. So dissolving and hydrogel forming, we can say it's a polymeric microneedles, because the needle is made from the polymeric part. So here the needle is made from the polymer that can swell when we apply to the skin. So in this kind of microneedles, we will have two parts. The first part is the needle. The second part is the drug reservoir. So the purple one is a drug reservoir. So the drug reservoir can be formulated like a tablet, like a patch, or any other form. So the application of the system is like we will apply the microneedle on the top of the skin, and then we will apply the drug reservoir on the top of the microneedle, right? So when we apply the microneedle on the skin, they will absorb the interstitial fluid on the skin. They will swell, and then the solution or the liquid will solubilize the drug on the top of the formulation, and then the drug on the top of formulation will go permeate to the hydrogel system, and then the drug will permeate to the skin. So this is the main idea of the mechanism of action of hydrogel forming microneedle. Why it is created? Because if we only put the drug on the needle, the limitations will be the drug with high dose will have some problems, right? So if we want to use this for the drug with high dose, it's not enough to put the drug only with the needle, right? So it is really challenging. So if you make the needle longer to improve the drug loading, you will kill the patient if we make the really long needle, right? So this is the limitation of the system, and that's why with this system, with the fifth system, which is the hydrogel forming systems, we can load the drug as high as possible because we put the drug on the top of the needles, right? So this is the example, the types of the five types of the microneedle that is developed to deliver the drug to the skin. Okay. So this is the examples. This is the real figures of the needles. So if you have a look here, the needle is really small, and if you realize, I put some rulers on the beside the needles. It is a rulers. It's one. This is like 5 millimeters. This is 1 centimeters, right? It's really short. It's really small, and the needle is really small. So this is one of the example of the needle, and sorry, and sorry. I think I lost my presentation. Okay, this is also the pictures of the needles. So if you can see here from microscope, the needle is really small. It's 0.5 millimeters, 500 micrometers. It's really small, and we can compare the pictures on the scanning electron microscopes. So we can clearly see that the needle is really small, and it's also sharp, so it can permeate the skin, right? And this is the examples of another type of needle. So if you have a look here, so here is the the examples of the needles. This is also the examples of this of needles. This is a metal part, so it is a solid microneedle because it is made from metal, so it can create the hole in skin and then help the permeation of the drug to the skin. So this is the examples of hollow microneedle. So here you can see like there's a hollow, right? You can see here there's a hollow. So if we apply drug, if we apply to the skin, it can help facilitate the drug to the skin. So maybe some of you will ask, is that painful? Will we have a problem with the pain if we apply to the skin? The answer is yes, but not really. The painful is really less. So I can say because I was getting involved in one of the trial of the clinical study of microneedles when I was doing my PhD, and then I used microneedle on my skin, and then I really didn't feel any pain at all. Like when you apply the microneedle, you will not have any problem with the painful because the application of this will not reach the nerve of pain in the dermis layer because it will create a hole only until the upper part of the dermis, while the nerve of the pain is located in the down part of the dermis. So the application of this will not reach the pain nerve. Okay, so no worries, don't worry, you will not have any problem with the pain with this kind of application. So like a derma roller, right? You will see like a bleeding or something, but the ladies enjoy it, right? Because it can make them beautiful, but it's not like something like painful. So fabrications of the microneedles. Okay, maybe I'm just too excited to explain to you all, and now I'm thirsty. So please give some time to drink something.
Have a rest.
No, I'm not taking a rest because I will sleep if you ask me.
Oh my God. You're on fire.
So please. All right. So so how to prepare the microneedles? So basically, we will have several types, several methods to prepare the microneedles. So here we will have methods. The first method is really simple. It is not. Let me. Oh yeah, sorry. this is not this is the first method that people use to create the microneedle, but it's so complicated. It's not like easy. It's just like complicated. So if you see here, so we put the drug solution into the base plate, and then like if you see the illustration, when we like we will combine two part of base plate, and then when we make them separated, we will apply some air here. So the dry when we remove the part, the air will help to facilitate the drying of the needles, and then when it's dry, it will form the needles. It is really old methods actually. It's 2013, and it's not like it is not used anymore actually. For I mean, it is used, but not many people use this anymore because of the complicated system, the complicated methods of this for to prepare the microneedles. So now what people are trying to do is these methods. So this is two-step casting methods of the needles. So okay, let me remove some part. Okay. Right. So here we will have mold. We will have silicone molds that we prepare for preparing the microneedles. So here we will like prepare. We will dissolve the drug with the polymers. So in order to prepare the microneedle, we will have to make a drug solution. But it's not only drug solution, but we have to combine this drug of solution with some polymers, which is soluble in water if you want to make dissolving microneedles. If you want to make implantable microneedle, we will have to dissolve the drug with the polymer that can contain polymer with the insoluble part. So here we will have the molds. So this is actually the mold that we will use to prepare the microneedles. So here we will have a drug solution. We will just need to put the drug solution on the top of the needles, and then we will centrifuge. So sometimes how to fill the drug, how to fill the mold cavity with the needles with the drug, we will centrifuge them. So we will put the drug on the top of the molds, and then we will centrifuge the molds. So when we centrifuge the mold, the drug will go to the cavity of the molds, and then the molds will be, the drug will be in the cavity, right? So and then we will put the base plate, like the polymer without any drug. The yellow part is the polymer without any drug, right? And then we will solidify them. We will try to wait until 2 or 3 days, and then we will just peel as simple as that, and then we will form the microneedles. So this is the like this is really simple, but it is challenging because it's not easy to form the hard needles so we can control the drug properly. Okay, this is really simple way, but it can form needle that can permeate the skin really well. Okay, so but when we want to develop a needle, so you have to make sure that we have a good penetration and good mechanical properties. So here, so if you can see here, guys, the examples of the needles, they prepare like a 3 part, or the black one, the pink one, and the white one, with the same mechanism. So it is not like complicated as it looks. So first we have to make sure that the needles that we created have a good penetration properties. And also, like, the problem is we have to make sure that when we apply the microneedle on the skin, it is not like a banding like this. So we have to make sure that it can permeate through the skin. So we have to perform the study, mechanical properties and penetration tests to make sure that the needles are strong enough and the needles are able to penetrate the skin. Okay, this is the main two. We have a lot of characteristics, we have a lot of characterizations, but the most important characterizations that people should do when they develop microneedles is like this. Okay, Okay, so finally, we are arriving with the main part of my talk, which is the use of micros for diabetes treatment. It's not that long. It's not that it's not that complicated. It's not that long because I already spent 75% of my presentation with the interaction. Because I mean, why I explain that, because it is not something like it is not common in Indonesia. So I need to explain the basic knowledge of microneedle first before going through the explanation of why you use microneedle for diabetes treatment, right? So here first, one of the study. So when I was contacted, when I was invited to talk about this diabetes treatment, I was like, okay, I have one study. So I have one study. We use microneedles for diabetes treatment, where we use this kind of things to deliver metformin as a model drug. So in this study, we developed glucose response microparticle. So what is glucose response microparticle, guys? So this is microparticles that can release the drug only when the glucose levels is high. So this is it can, like, when, if you remember, one of the problems of oral anti-diabetic drugs is lack of specificity, lack of selectivity. So if we want to improve the selectivity of the drug, we have to choose the appropriate drug delivery system. So in this study, we used metformin as a model drug here. And first, we use microparticle system. But in these systems, we use glucose response microparticle. So what is glucose response microparticle? So this is the particle that, for example, like we coat the drug with the particles, and the polymer will keep the drug if the glucose levels is low. So when the glucose levels is high, so the polymers will release the drug, right? So when the polymers release the drug, it can reduce the glucose blood levels, and when the glucose blood levels is low, the drug will stay again in the polymers. This is the idea of this system, right? So this is one of the benefits of the system is that we can control the drug to avoid the hypoglycemic, right? So we can be sure that the drug is not released if the normal condition is there, right? So here we use gelatin, and we also use PBA, phenylboronic acid, actually. So we developed 5 kind of formulations, and then we try to compare the characterization of this. And as you can see here, we were able to produce like a circle shape and various shapes of the particles with the micro size. So this is the characterizations. We perform FTIR to show the form of the microparticles, and then we also perform DSC and then XRD to do some physical characterization, because it's really important to make sure that the characterizations of the drug is still there, right? And this is the main idea of this. So let me show you something. Okay. So here is the proof of concept study. So this is the main idea of this, because here we use 4 conditions. So if you have a look this with this, sorry. If you have a look here, so A is media without any glucose. B, C, and D is the media, release media, and we add some glucose to the medium. So if you have a look at the results, if you have a look the release of them, so the green one is the microparticles, the black one is microparticles but without any glucose response materials, and the blue one is the pure metformin. So what we show here is that um, you can see the release of the drug of the black one, right? So the green one. So the green one is the, like, if we don't have any polymer that can control the release of the drug with the presence of the glucose, we can see here, like, after 24 hours, the release of the drug is really, really small. This is the condition of normal condition. This is condition of normal people. So the glucose is not that high. And if we increase the concentration of the glucose, the release of the drug will also be increased. So it means that we were able to control the release of the drug with the presence of the glucose response material. And here, if you have a look in the E, it's the pulsatile release. So with the presence here, glucose response, the blue one here, the blue one is the low concentration, and the green one is the high concentration. You can see here, like, with the use of the microneedle, the use of microparticle with glucose response material, we were able to control the release of the drug. You can see here, like, if the concentration of the glucose is high, the release drug is high. When it decrease, the release of the drug is also decreased. So you can see here, like, a really fluctuative release because the condition of the body is like that. So if the level of glucose is high, so the drug will go down, and if the concentration is high, the drug will go high. So this is the main idea of this. And what we do was, we finally incorporated the microparticle into the microneedles. So here we prepare the microneedles using polyphenol pyrrolidone and polyphenol alcohol. So we use these formulations to prepare the microneedles. So we combine several types of the polymers, we compare the several concentration of the polymers just to optimize the formulation. So this is the results. So we were able to finally show that the needles were sharp and really small, and they have really good mechanical properties, and they have a really good insertion properties, meaning that we were able to insert the skin really well, and the recovery of the drug inside the formulation is not affected by the formulation, meaning that all the drug is there. We couldn't find any drug reduced in terms of the formulation process. And if you have a look in figure number 7 here, so A, B, C, D is the same study that we have done in this part, but this is microneedle. So we can see that the permeation of the drug through the skin, if we apply microneedles in the media containing high glucose, the drug will permeate really well. So we can see here that the drug is there, right? The green one is the green one is the formulation with our particles. So we can see here that it's there. And finally, we did some in vivo study. So here is in vivo study. So we perform in the rats with diabetic models using streptozotocin compound. Here we can see that the formulation that we have here is this. So we can reduce the drug, the blood glucose levels, and then we can maintain in the long period. So the concentration of, because of the selectivity, because we compare with normal normal treatment, like oral treatment of metformin. So because of lack of selectivity, the control formulation was not able to maintain the drug, the blood glucose levels. But here, if you can see here in our study, we can control the blood glucose levels until like 6 hours, and then it can increase, but not that high if we compare to this part. Why? Because we have like glucose response materials, so they can control the presence of the drug in the body. So when we have this in the body, so when the glucose levels is high, so the drug will be released. But when the glucose response is lower, the drug will stay in the part of the particle. This is the main idea of this. So this is one of the study that I'm performing using microneedle to control release of the drug on the diabetes treatment. So next, so we will have another study. Okay, okay, yeah. So this is one of the study using insulin. It's not my study, but I quote here. I asked for permission to show this presentation also, so no problem. So this is one of the study. They use insulin for transdermal delivery of insulin, yeah, for diabetes treatment. So here they prepare the formulation, and as you can see here, they have like really good penetration to the skin. They use subcutaneous part of the skin here, and they can see that, okay, the insulin is there, the insulin is inserted to the skin. So here, after several hours, they see, like, they compare the drug with the intramuscular insertion, and then they see like a subcutaneous injection, intramuscular microneedle insertion. As we can see here, that we can control the profile of the drug using microneedle, but subcutaneous is not like that. So the most important part of the study, as you can see here, like a similar study that I have done previously. So if you have see here, like this is similar with this. It's also like this. So if you can see here, like the use of the microneedles is really effective. It's not only to reduce the blood glucose levels in the lower part, but also maintain the low compared to other part. So it's showing that this is really promising part of controlling this. I remember one of my friend. So he was doing his PhD in Austria, and he was diagnosed with type 2 diabetes. And the doctor in Austria gave him insulin microneedle in the arm to control the release of, to control the blood glucose levels. So he doesn't have to be worried to to take the drug, for example, to control the blood glucose levels, because they use microneedles in the arm to control the insulin to the skin. So this is one of the other examples of the successful part of microneedles in terms of diabetes treatment. It is again the use of microneedles. It's again insulin, and in this part, they in the previous one they use PVA to prepare the microneedle. In this part, they use alginate and maltose. So it is different part of the polymers. So again, it is successfully shown that they were able to reduce the drug levels. And it is not only that, they also, like, this is the this is the concentration of the insulin in the plasma, just to show that, okay, we're not only reduce the blood glucose levels, but also we were able to deliver the insulin, something like that. So here, one of the part also, they use another part of microneedle drug delivery systems. So they use glucose oxidase compound. Here they use polymer. So it is similar to the study that I performed. Like they use phenylboronic acid here. Phenylboronic acid is the compound that can control the release of the drug based on the presence of the insulin. So this is the needles. So they apply into the pig model, and they were able to control the release of the drug based on the presence of the glucose because of the selectivity of the particles of the formulation. Okay, so I think I'm almost there because I will have only 1 hour talk, but I hope that we will have longer time for discussion. So this is one of the example again. So if you have a look, the needle is really, really simple to make. It is just the matter of mechanical properties is the challenging part of this formulation. So they use this ixenatide, one of the drug for diabetes treatment, especially for type 2. So they prepare the microdose and then they will see like the glucose levels is really, really controlled using this non-fasting diabetes rats, and they compare with subcutaneous injection of this, and then they show that it is really effective to replace the needles, something like that. Okay, it is one of the example again. So this is separable effervescent microneedle. So this is made by a researcher to create the effervescent layer. So they use this, and like they will put this needle to the skin, and then the separation will be based on the effervescent reaction, and they will remove the part of the needle, and then the needle will stay the skin, and then it can control the release of the metformin, and finally it can control the release of the blood glucose levels here. So if you see here, they are successfully control the release of the blood glucose levels of the of the rats. And this is again metformin in diabetic rats. It is like if you see like we only use like different part of the polymers because we want to produce as effective system as possible. So if you see like the needle will be like this, and then the needle will degrade, and then they will release drug, and then the glucose will be decreased. Okay, thank you very much. Finally, I am in the end of my presentation. So if you have any question, if you have anything to ask, we can have a discussion if you want to discuss something as well in terms of drug delivery system in general, or microneedle delivery system in particular, or the treatment of diabetes in particular also. So thank you very much for your attention. Let's have a discussion, and I hope the discussion will be great. Thank you.
Okay, thank you very much, Professor Dian. Okay, now we are going to discussion session. Please participant who have a question, please raise your hand. Okay, committee, please participant.
I think one of the participants already raised his hand.
Okay, please, Mr. Arlan Kaya Imran.
Thank you. Yes, Mr. Fadli. Thank you very much for the opportunity. Very interesting material, Prof. Andi Dian. In definition, microneedle is platform for drug delivery which have different diameter and type material. When administered oral, active substance include 2 BCS class, which BCS class 2, 3, and 4 must first correct their solubility and their permeability problem. To achieve BCS class 1, which has a good solubility and permeability. It's the same things done for active substance formulated in the microneedle preparation, Prof. And also, how about metabolism?
Okay.
About metabolism. And second, my question is, According to Professor, microneedle preparation are very suitable or very effective to decrease glucose, for which oral antidiabetic group active compound mechanism of action inhibitor alpha glucosidase or sulfonylurea or with intramuscular insulin. Thank you, Professor.
Okay,
That's my question.
so the first question is like if we talk about biopharmaceutical classification system in oral administration, we know that for part of that, but in this kind of in this system, I'm not saying we don't consider those kind of things, but it's different thing actually. We cannot make them similar because in this part we don't see any permeability because we already breach the permeability. So if the drug have a problem with the permeability, they will not have any problem in the microneedle because we help them to permeate to the skin. So even though the drug is not having a really good permeability, because we help them to permeate to the skin, so they will have any problem with the permeability. So it's not the same thing. But in the solubility, yes, it is important because when we apply the drug in the skin, it is important to make sure that the drug will go to the blood circulation. So if they have a really bad solubility, they will not reach the blood circulation. We have to improve the solubility of the drug if we want to make them into microneedles. So if we have a problem with the solubility of the drug, we will have to improve the solubility first, and then we will incorporate them inside the microneedles. So it's something like that. But we don't have to worry about the permeability because we already break the rules of the permeability by breaking the skin, by importing them to the skin. So this is one thing that we need to bear in mind if you want to develop microdrug delivery system. It is the first question. So can you repeat your second question, sir? I didn't get it, sorry.
Yes. Microneedle preparation is very effective to decrease glucose, for which antidiabetic group active compound. In diabetes mellitus, we have 4 groups.
I see.
Active compound.
I see.
Inhibitor alpha.
I see. Yeah. So basically, nowadays people are trying to avoid some problems of oral anti-diabetic drugs. So because a lot of reports showing that metformin, for example, they have a lot of side effects. So basically, people try to develop microneedle for metformin, for example. So to the best of my knowledge, not all drug delivery systems, not all type of anti-diabetics drugs is developed into the microneedles, but only the drugs with the problems. You know what I mean? Like metformin, for example, they have a lot of side effects. Insulin, we have a lot of problem with insulin. Until now, it's really difficult to drink insulin because the insulin is really sensitive with DPH. While the injection, it's not really effective because we have the people type blah, 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 blah. People will not follow the rules. People will put the insulin everywhere they want, something like that. So that's why like these main 2 drugs is really the common drug that people use to develop the microdelivery system. But we will see in the future, maybe if people find a lot of problems with other drugs, people will try the solution to develop the microdelivery system for other drugs, something like that. I hope this answers your question, Mr. Arlan. Thank you.
Thank you very much, Professor.
Okay, thank you, Mr. Arlan. Next, we have other participant who has raised her hand, Mrs. Sukmawati from Palangkaraya. Please, Ms. Sukmawati.
Thank you very much, Mr. Fadli Hussein. Can you hear my voice?
Yes.
Okay. This is nice presentation from Professor Dian Permana. I want to ask a couple questions, more than 3 maybe. Mr. Dian, are you with that?
Yeah, I'm okay, but can we just have a 1-by-1 session so I will not forget what you asked?
Yeah, okay, thank you. My first question is talking about the purpose of microneedle. It's what kind of medication? That's, I mean, it's for systemic pathway or this only in local pathway. But
Okay.
in your explanation before, it can be useful for diabetes, so it's maybe for systemic pathway. There is a big portion for the systemic pathway or the local pathway. My first question, sir.
Okay, so well, it depends on of the delivery purpose. So as I said before, that we can use the micro delivery system for both, like for systemic and for systemic and for local effects. So if we want to deliver the drug for systemic effects, so we have to make sure, because if we have a look the structure of the skin, so we can see in the skin there are a lot of blood circulations that can absorb the drug, right? So if we want to deliver the drug to the systemic part, so we have to make sure that the drug can reach the blood circulation. And if you want to make sure, if you want to make a local effect, it means that we have to make sure that the drug will stay on the skin and then doesn't reach the systemic circulations. So if you want to, can I? Okay, can I plug in? So I cannot. Oh, sorry. I want to. Oh. Oh no. I thought that we can see the. I want to share I want to draw something in the whiteboard, but I cannot do it. So, I mean, we can do both. Like if you want to make sure that if you want to for example, if it's like a soluble drug, the drug is soluble, for example, and we want to make a local effect like a skin infection. So we cannot directly formulate the drug into the microdose. Why? Because if we prepare the drug into the microdose and then we apply the microdose on the skin, and because the drug is soluble, the drug will go to the blood circulation and then it will give a systemic effect, right? So something like that. So if you want to make them locally, the soluble drug, we have to make them insoluble. Like we have to make them insoluble. Why? How? So we can coat them with the particles, for example, like a microparticle. So the soluble drug will be incorporated into the microparticle, and then microparticle will be staying in the microneedle. So when we apply the particle in the skin, because the drug is not that soluble, the drug will stay in the skin and they will have a local effect. So basically, if we say the proportion, I'm not saying the local is higher than systemic, both, because I can say like a 50-50 proportion between this kind of purpose. So because I mean it can effectively deliver the drug using through the local effect and also the systemic effect, something like that. I hope this answers your question. Sorry for really, really complicated answers.
Okay, thank you, Mr. Dean. My second question, because I never seen the microneedle before, I want to ask about the length of the microneedle. It's capable of same as the length of lancet needle from the microneedle. Something the needle is used for the checks like blood sugar and anything else, some cholesterol, about the length of the microneedles.
So the needle, if you have a look in the let me show you again. So here are the examples of the needles. So this is 700 micrometers. So here is 700 micrometers. So the main idea of the length of the microneedle is that we have to make sure that it can permeate the stratum corneum. As simple as that. So it's not as long as lancet needle. Lancet needle is really long, so it's bleeding and it can cause some pain. But the microneedle is not like that. So something like that.
Okay, Mr. My last question is that microneedle treatment directly released into the skin and then penetrates into the blood vessel, or there is a possibility to can made in like slow response or slow release formulation, like sustained release in the
Yes.
oral.
Yeah, of course, we can make a sustained release profiles. We can control the release by using different polymers, right? So if we use polymer for sustained release, the release of the drug in the skin will be sustained also. Or what we can do is we can incorporate the drug into the nanoparticle or into the microparticle, and then the nanoparticle and the microparticle will be including into the microdel, and it can control the release of the drug. So it's really possible to control the release of the drug using the microdel by playing with the concentration of the polymer that we use for the preparation. That's it.
Okay, thank you very much for the explanation. It's so clear.
Thank you.
Thank you.
Thank you, Mrs. Sukmawati. Next, is there any question from participant? Okay, please. Okay, Professor Tian, we are
Yes.
here right now in our auditorium. Joining with us today is our student, and now
Okay.
Our student will give you some question.
Okay.
Okay, please.
Thank you, Prof.
Ah,
Let me
okay.
introduce myself. My name is Siti Alicia Henga. My question is the dissolving microneedle type not harmful to the body when used for a long time?
Okay. Only one question? Should I answer it now? Okay. So, what's your name again? Sorry.
Siti Alicia Henga.
Alicia.
Huh?
Alicia.
Alicia. Okay, Alicia, thanks for the question. So, as I said before, that I was one of the participants of the clinical study of the microneedles, and then we were given the microneedles every single day for 14 days, and then our blood was taken, and then all of the inflammation marker were tested, like TNF alpha and everything, and we didn't find any significant effects. So, meaning that the application of the microneedle is safe. It doesn't cause any irritation, and it doesn't cause any pain, and also it doesn't cause any allergic effects of the skin. Because the polymer that we use is biocompatible and biodegradable, so it should be biocompatible. It shouldn't be causing any problem with the skin, something like that. Okay.
Thank you, Prof.
Okay, thank you, Alicia.
You're welcome.
Okay, is there any student to give question? Okay, excuse me, Professor. There is another student.
No problem at all.
Okay, please.
Thank you, Prof, for the opportunity. Let me introduce myself. My name is Yurika Tolua. My question is, can all medicine be microneedle made? Microneedle made.
Okay. Well, if you ask, can everyone make the microneedles? Can everyone make microneedles using all of the drugs? Yes, you can, but it is important, of course, to know. So before making the drug into the microneedles, we have to make sure what is the problem first, right? So if we have a problem with the metabolism, if we have the problem with the oral administration, yes, we will have to make them into the microneedles. But if they don't have any problem with that, we just need to, like for example, the oral bioavailability of drug X is really low. And we have to know why it is low. If the problem is the metabolism, so we can make them into the microneedle. But if the problem is the solubility, so we can just improve the solubility and then give them orally, something like that, right? So it is the first thing that we have to know. And then secondly, one thing that we have to know that the size of the microneedle is quite low, right? It's quite like small, and the dose it's quite low. Like if we have a high dose, like 700 milligrams or even 1 grams, so it cannot be made into the microneedles. So those should be 500 milligrams or less, something like that. So in order to put the drug inside the needles. I think this is the simplest way to answer the question of the students. Thank you.
Thank you, Prof.
Okay.
Thank you.
Okay, thank you very much, Professor Tian. I think it's there is more student. Professor, it's okay.
Okay.
Okay, thank you, Professor.
Okay, thank you for the opportunity that has been given to me. Before that, let me introduce myself. My name is Angel Priscilla Ahmad, and my question is how the potential of a microneedle drug delivery system can be developed in Indonesia. Thank you.
Well, it is really, really interesting question because I did it, so everyone can do it. So how it's possible? So it's really possible. So the thing is like we have to know first the like it is another topic because it's really long story how I create the tools to prepare the microdose. But if you ask how possible it is, my answer will be yes, it is really, really possible. We just need to modify some part. We just need to modify some tool, some instrument, but it's possible. Okay.
Okay, thank you, Prof.
Okay.
Okay, thank you. Uh, okay, I think there's more student. Okay, maybe committee.
Baik, thank you,
This is the last one.
Prof, for the opportunity. Introduce, my name is Safitri Yasin. My question is, when we have an active ingredients, what are the things we need to consider when selecting the type of microneedle?
Okay. So the type of microneedle is not about the drug characterizations. Okay. So the type of the microneedle is based on what kind of delivery purpose that we want. Like if you want to deliver the drug to the skin directly, you can use dissolving microneedles. If you want to control the release, if you want to if you want to help the permeation of the drug through this from the cream, from the serum, or from the patch, you can use solid microneedle. So actually, there is no specific drug that we can that we should know before we select the type of the microneedles, because the selection of the microneedle is based on the type of delivery purpose. It's not the type of the drugs, something like that, right? Thank you. She didn't say thank you.
Thank you, Professor. I think she's forget.
Okay.
Okay, there's one more, Professor.
Thank you, Prof, for the opportunity. Let me introduce myself. My name is Rahmatia Nyora. Microneedle is a new preparation that is very interesting for me. From of your explanation, that microneedle preparation can be developed for topical active substance such as antibacterial or fungi. My question is, can this microneedle patch preparation be developed for preparations such as acne patch contain active ingredient for antibacterial P. acnes?
It's a really long question, and the answer is yes, you can. So basically, like if we have a problem with the skin, it's not only skin infection like a normal skin infection, but also other types of skin infection, including acne, because you know acne is quite difficult to permeate. Because if we have a problem with the skin acne, the skin will be thicker, and if the skin is thicker, the drug will be difficult to permeate. So we can use the microneedle to help the facilitation of the drug to be inserted into the acne patch. So in acne part. So the answer is yes, you can.
Thank you very much, Prof.
Thank you.
Okay, thank you.
Okay. Thank you for the time and thank you for the opportunity. Let me introduce myself. My name is Srilansya Badu, and I have question for you. The question is, can microneedle medication be made for all disease?
Okay. It is really long explanation because you know before we choose the type of disease, we have to see the problems of the medication. So sometimes we don't have to see what kind of disease. Sometimes we have to see what the problem of the medication. For example, the oral bioavailability. The oral bioavailability is faced by a lot of disease. Say, for example, drug A, B, C, D, E, F, G. There are different drugs, but they have a problem with the oral bioavailability, and they can be used to treat a lot of problems. So as long as they have oral bioavailability and traceable delivery can be used to overcome the problem of that, so we can use that for microneedle delivery system. So if you ask what kind of disease, we cannot specify any specific disease for that, but it depends on the problems. You know what I mean? Like if the drug have a problems, no matter what kind of disease they are, we have to make them into microneedle, for example. But for the skin disease, it's really good for microneedle, something like that. So I'm talking the previous sentence. I'm talking about the systemic effect of the microneedle. Okay. Okay.
Okay. Okay, thank you, Professor. I think he's forget to say thank you to you.
It's okay.
Okay. Okay, I think Professor Dian, there is another participant who raised her hand. Maybe you know this one. Okay.
I know her really well. Okay.
Mrs. Julfi Ayu, please. Okay, Mrs. Julfi Ayu, maybe using microphone.
Assalamualaikum, my professor. Okay,
Yeah.
I am Zulfi Ayu, one of your student. We are very proud to learn directly from the first microneedle professor in Indonesia. I would like to say thank you for being willing to be speaker in this conference, Professor, so that our student can hear drug delivery system directly from you, my professor. My question, this conference is about diabetes mellitus. Maybe you can explain us about the microneedle for other disease. Thank you.
Okay. So I think I have I can show you some examples here actually. Where is the examples? Yes, I think it's better to show. Okay, so just in short explanation that in addition to diabetes mellitus, for example, so here is a lot of example for using microneedle for can everyone see the screen now? Okay. So I can show you several examples. The first one is we incorporated albendazole nanocrystal. So we make the nanocrystal first using albendazole, and then we formulate this into dissolving microneedle to avoid some parasite infection. This is parasite infection, albendazole, right? So here we can see the example of the needles, and then we also perform the pharmacokinetic study. Here we can see like we compare microneedle with the oral administration, and then we can see like the oral bioavailability is completely 8,000% higher compared to oral administration. So here we can see a really, really good bioavailability, improvement of bioavailability. Here also we incorporate like the first example for systemic application. This is for local administration. So here we can see like a doxycycline particles we use into dissolving microneedles for bacterial biofilm infection. So here we can see that the bacterial can be eliminated to the skin completely using the system. So here of the example. So I cannot explain this in detail because it can take 2 hours. So it is the needles, it's yellow because the drug is yellow. This is the examples of this skin infection that we create, and this is the profile of the drug, profile of the nanoparticles, and sorry. So that's it. So this is the example of why microneedles can be used for systemic and skin infection for local administration. And we have a lot of examples. So if you guys interested, if you guys want to see, you can open my Google Scholar, and then you can see some of the studies that I perform using microneedles. So that's it. Thank you.
I am very impressed. Thank you.
Okay, thank you very much, Professor Dian and Mrs. Zulfi Ayu. Okay, Mr. Arlan, I think. Okay, please, Mr. Arlan.
Very interesting material, Prof. One question. The previous speaker explained the potential of natural ingredient for oral antidiabetic. What about active substance from natural ingredients to formulate to microneedle some as extract? We have to purify, or we can use the extract? Thank you.
Well, yes. Well, why we have a problem? Why we develop a microneedle? Because we have a problem with the oral administration. So for the natural compound, first, the thing that we need to answer is, do they have any problems with the oral administration? If they don't have any problem with the oral, why should we make them into the microneedles? So if they don't have any problems, like we will find the problems after several years of consuming. A natural product is not as simple as that. It contains a lot of compound and a lot of materials or something that we need to concern. So if the question is, if we want to formulate them into the microneedles, like we have to make sure that they if we formulate the extract, we have to know all of the compound in the extract. So the problem in drug delivery system is we have to know the main compound. We have to know all of the compound. If it's not known, if it's like really crude, it is not well known, we cannot make them into the microneedles. But before going to that, we have to make sure that they have problem with the oral. So if they don't have any problem with the oral, why should we make it into the microneedle, right? So it's really complicated things, something like that. Thank you.
Thank you, Prof.
Okay, thank you. I think that's all, Professor Dian.
Yep.
We have reaching out time. Really nice presentation and nice discussion for today, Professor Dian. And maybe there is some certificate of achievement for Professor Dian from committee. Okay. Committee, is there any certificate of achievement for Professor Dian? Okay, maybe we are waiting, Professor Dian. Really nice presentation for today. You're on fire. And I
as usual.
I think we should invite you to give some guest lecturers in Poltekkes, Kemenkes, Gorontalo, Professor Dian?
Oh, I would love to. I've never been in Gorontalo before, so it's gonna be a really
Oh,
first time.
okay. You must going here for offline. Okay, committee, there is prepare for certificate of achievement. Okay, Professor Andi Dian Permana, this is the certificate of appreciation for your thanks for being our invited speaker at the third international conference in pharmaceutical and health science, and we hope we can join with you offline in the next opportunity maybe. Thank you very much once more, Professor Andi Dian Permana. Okay.
Thank you.
Next, I give back to the Master of Ceremony.
Thank you very much to our keynote speech, Professor Andi Dian Permana, MSI, PhD, APT, and our moderator, Mr. Fadli Hussein, SSE, MSE. From the depths of my heart, I do apologize for our mistake in presenting this event.
Thank you very much for your kind attention. See you tomorrow in the second day.
Wassalamualaikum warahmatullahi wabarakatuh.
Thank you, Professor Dian.
Thank you.